Good morning, everyone, and welcome to day 2 of the 3rd International Conference in Molecular Life Sciences at the Institute of Biochemistry, Molecular Biology, and Biotechnology. We will start off with session 3 of oral presentations. I would like to invite the chairpersons and judges for this session, Professor Chayanika Padumadasa, Professor in Chemistry at the University of Sri Javadanapura, and a visiting scholar of the Natural Products Research Center at the University of Arizona, and Dr. Ireshika De Silva, Senior Lecturer of the Department of Chemistry at the University of Colombo. We will begin the sessions now. You can invite the speakers first.
Hello to all those present here today. I am Tafani Salim, and today I will be presenting my study, Protein Ligand Docking Study for the Identification of Ligand Binding Sites and Novel Therapeutics for Breast Cancer. This study was conducted together with my mentors, Ms. Heishani and Ms. Ominda. These are the contents that I will be discussing today. To start off with, breast cancer is a complex heterogeneous disease that is caused due to the uncontrolled growth of cells in the milk duct and in the lobules of the breast. This results in a tumor which can then metastasize into any other part of the breast and even to other parts of the body. The current treatment for breast cancer includes hormonal therapy, chemotherapy, radiotherapy, and surgery. However, despite the wide use of these treatment options, breast cancer remains one of the primary causes of death in women. And this is mainly due to the tumor relapse and the resistance of the drugs that occurs in the patients. This calls for the urgent need of more effective drugs that can be used to treat the cancer. And naturally occurring phytochemicals have recently gained the interest for showing great potency as drug therapeutics. So the aim of my study was to identify potential and effective phytochemical ligands by carrying out docking studies in order to treat breast cancer. This slide outlines the materials that I used. There were 6 phytochemicals that I used, and 2 FDA-approved drugs were used as the control. The Autodoc was used to run the docking, and Chimera and BioVia was used for the analysis. Moving on to the methodology, the estrogen receptor alpha was the receptor used, and the 2-IOK receptor was retrieved in using RCSB and loaded in Autodoc. This was then prepared by deleting the co-crystal IOK, other heteroatoms like gold atoms, and deleting the water molecules. To this, the polar hydrogen atoms and Coleman charges were added as required in Autodoc. Meanwhile, the phytochemicals were analyzed for the drug likeness using Swiss ADME, and as mentioned, 6 phytochemicals were then used for the study. The SDF format of the phytochemicals was downloaded using PubChem, and this was then converted to the PBDQT format using OpenBabel. This was also then loaded onto Autodoc and was prepared by setting the maximum number of rotatable bonds. And since it was a blind docking that I carried out, the grid box that was generated was used to cover the entire area of the receptor, and the coordinates were given as shown here. Then Autodoc was run with 50 GA runs with a population size of 300. For the resulting RMSD table was then used to analyze the binding energy and the inhibition constant of the interactions. And as mentioned, Chimera and BioVia was then used to analyze the docking process on the interactions. Further, the docking protocol was validated by redocking the co-crystal IOK. And finally, the receptor to IOK was validated by using the Ramachandran plots. This was repeated for all of the 6 phytochemicals, and including the 2 FDA-approved drugs, one of which is given here. Moving on to the results. This table gives the binding energy, the inhibition constant, and the amino acid interactions that were involved in the interactions. The lowest binding energy with lowest inhibition constant signifies stable constant, and as can be here, camptothecin had the lowest binding energy. And there were many amino acid interactions involved, and leucine 525 was a hydrogen was in hydrogen bonding. There were many amino acid residues that were common to all chemical lists, as well as the control drugs, signifying that the ligands binding in the same binding pocket. This shows the docking process of my phytochemicals as seen in Chimera. And this shows the interactions of the phytochemicals using BioVia. This shows all the interactions that were involved. Moving on to the validation of the docking protocol, the IOK co-crystal was redocked as mentioned, and it gave a result of negative 10.39 binding energy with an inhibition constant of 24.19 nanomoles, signifying that this docking was validating. Moving on, this diagram shows the Ramachandran plots for the original ligand to IOK, as well as the redocked ligand after the blind docking was conducted. And as can see, there's a similarity between the 2 plots. And you can also see that many of the residues are in the red region, which signifies the most eroded. There were less in the white region, which is the disallowed region, which again confirms that the receptor is validated. So based on the results of the study, the most potent drug was shown to be camptothecin with a binding energy of negative 9 kilocalorie per mole and an inhibition constant of 254 nanomoles. Additionally, there were 2 other phytochemical ligands, namely azotoxin and pristinamycin, which also proved to be effective. And as mentioned, there were many amino acid residues that were found to be common in all phytochemical ligands, and these are listed here. This was also confirmed by the study that was conducted by Rasul and his colleagues, who also got similar amino acid residues in the binding pocket. So coming to my conclusion, the study proved that there are the phytochemicals which are proving to be potent against the breast cancer, and the most effective one can be concluded to be camptothecin, azotoxin, and pristinamycin. So with further wet lab experiments and other clinical trials that can be conducted, we can show that these phytochemicals can be used to effectively treat breast cancer. These are my references, so that brings me to the conclusion of my presentation. I would like to take this opportunity to thank my mentors as well as the organizing team for giving me this opportunity for presenting my research study. Thank you very much for your time. Yes. Hello, can you hear me?
It was a good presentation, Hapani. I have one question for you. So now, how did you choose the phytochemicals for your study?
I'm sorry, could you repeat that?
How did you select the phytochemicals for your study? What was the basis? How did you pick that?
Yeah, I went through some research papers to get to look at some phytochemicals, and that was mostly how I got my phytochemicals. Yes, through the research papers.
Yes, through research papers. On what basis, Tafani? What was the basis?
To show effectiveness against the breast cancer. No. So then I tested it with it using the Swiss ADME, and if it did show that it had some potency, then I went ahead with it.
Yes, so that was your aim. That was your aim or your goal. So to pick these compounds for your study, what was the reason? You did a literature search, yes. So what made you pick these over others? Because there are plenty. In the literature, what did you see about these? What made you pick these?
Yeah, they did show some potency against the breast cancer.
Okay, so that is lab work or computational studies? That was
computational. Yeah, that was also computational.
Okay, so then if so, what is different in your study? Now you are saying in literature, computational studies for these compounds are there.
Yeah.
So
not all of it, just some of it were there, and some I went with the. I just looked up for some phytochemicals, got a list of phytochemicals, and then tested to show if it showed the initial potency, and then I tested with the receptor.
Okay.
I had the same question, Chapani, because maybe you could have done before you selecting the phytochemicals, maybe based on the in vitro studies, you could have selected these phytochemicals. In that some of these you have just selected without knowing any activity or anything, right?
Yeah.
So there has to be a sound background as to why you pick these compounds. So, I mean, previously reported work can be a start point. Then, Tafani, you have to mention a little bit and then highlight what is different in your work. Do you understand?
Yeah. Yeah.
Okay. Okay, Tafani. So, thank you very much.
Okay. Thank you.
We can move on to the next one.
Today I'm happy to talk about isolation of an anti-cancer compound from manager of Veronica leaf and investigation of its potential toxicity. I am Ismail from When we talk about lung cancer, it accounts for 2.06 million cases in 2020, 1.8 million of deaths in 2020, being the second most frequently reported cancer in the whole world. Lung cancer can be divided into two broad subcategories, such as small cell lung cancers and non-small cell lung cancers. Of these, non-small cell lung cancers account for 85% of all lung cancers. Smoking, asbestos, polycyclic aromatic hydrocarbons, air pollution, usually things like these are known as risk factors for lung cancer. And currently available treatments are chemotherapy, radiotherapy, and surgery. But there are some limitations of this treatment. First thing is they have unpleasant side effects. Other one is cancer can develop resistance towards the treatment. Mangifera cervica is a plant endemic to Sri Lanka and known as Atambe in Sinhala. A slow-growing tree, they are edible fruits. An important thing is it has been used to treat cancer in Sri Lanka traditional medicine. Previous studies conducted IBMBB, two novel compounds against breast cancer to investigate cytotoxic effects of Mangifera cervica leaves. We collected them from Herbal area in Sri Lanka, dried them into towels and finally powdered. Then we carried out sequential extraction using chloroform, ethyl acetate, and methanol as well. Then we used sulfodamine V assay to investigate its cytotoxic activity on MCIH292 non-small cell lung cancer cells and MRP normal lung fibroblasts. In sulfodamine V assay, we placed 5,000 cells per day in 96 plates, wait for 27 Celsius, with 5% carbon dioxide and 5% air. Then we treated them with different doses of the extract, wait for 24 hours. We mixed the cells using trichloroacetic acid inhalation. Then we stained the cells using sulfodamine V. Then we stabilized the grit base, and finally absorbance was obtained using HT at 540 nanometer wavelength. We used drop pad prism 7 to calculate IC50 value. To isolate cytotoxic compounds from Mangifera cervica leaves, from the previous assay we identified that its chloroform extract is more active against MCIH292 cells. So we carried out large-scale extractions from Mangifera cervica leaf. Then we distributed that between 9 to 1 methanol water solution and hexane. Then we found methanol water layer is more cytotoxic to cancer cells. Then we dried that layer and we subjected that solvent partition using 4 to 6 methanol water solution and chloroform. Then we found chloroform layer is more cytotoxic to MCIH292 cells. Then we dried that layer. We subjected that to silica column chromatography to obtain some fractions which were cytotoxic to MCIH292 cells. Then we combine those fractions and subject to HPLC, high performance liquid chromatography, to obtain the final molecule named as A1X. Then we carried out colony formation assay. In that we seeded 500 cells per well in 960 plates, then incubated for 24 hours. Then they were treated with different doses of A1X molecule and observed for 7 days. Finally, we stained those cells using SRB dye and we counted cell colonies using phase contrast microscope. Then we carried out cell migration assay. For that, we seeded 200,000 cells per well in 24 well plates and incubate for 24 hours. Then we made the scratch using 2 microliter, 200 microliter pipette tips, and then we treated cells with different doses of A1X. Then we observed the wound closure just after treatment and after 24 hours. Then we carried out observation of morphological features using fluorescence microscopy. To that we feeded 6,000 cells per well in 24 well plates containing cover slip, incubate for 24 hours. We treated those cells with A1X compound, different doses of that molecule. Then we fixed those cells using 4% formaldehyde. Then we stained them with ethylene or ethylene bromide mixture. Then again we stained them with fused dye. Finally, we visualized them using fluorescence microscope. From those experiments, we obtained these results. We found that chloroform extract, out of all the other extracts we got from sequential extraction, chloroform extract was more cytotoxic to MCIH292 cells and less cytotoxic to MRP5 normal lung fibroblasts. Therefore, we carried out our studies using chloroform extract. After we separating chloroform extract using pollen chromatograph, we got 14 fractions cytotoxic to NCH292. Then we combined them and subjected to HPLC. From that, we obtained A1X molecule, which showed 5.46 micrograms per milliliter IC50 on NCH292 and 10.54 micrograms per milliliter IC50 on MRP5 normal lung fibroblast test. From colony formation as a result, we obtained P. We found dose-dependent reduction of colony formation efficiency in NCH292 cells, indicating our molecule can reduce colony formation ability in non-small cell lung cancer cells. From cell migration as a result, we obtained that results that show significant reduction of wound healing ability of NCH292 cells due to our molecule. This indicates our molecule can produce wound healing ability in non-small cell lung cancer cells. From fluorescence-assisted cell morphological studies, we found dose-dependent reduction of cell membrane integrity due to A1X molecule in NCH292 cells. And from the host dye, we found that chromatin condensation in these cells exposed to A1X molecule, and these results indicate that A1X molecule can trigger apoptosis in NCH292 non-small cell lung cancer cells. So we can conclude that A1X compound isolated from Mandipera leaves exerts significant cytotoxic effects on NCH292 cells and less cytotoxicity in MRP5 normal lung fibroblast. Therefore, this molecule is selectively cytotoxic to NCH292 non-small cell lung cancer cells, and it reduces colony formation ability and wound healing ability in non-small cell lung cancer cells. And this can exert potential apoptotic effects on NCH292 cancer cells. And from these results, we can identify this molecule has the potential to be developed into an anti-cancer drug to treat lung cancer. In future, we will be doing these things. We will be evaluating apoptosis using caspase 3-7 assays and gene expression analysis. We will be carrying out gene expression analysis to investigate apoptosis and epigenetic modification in lung cancer cells. Structural decision for this molecule is currently being carried out. This was supported by science and research in Sri Lanka. This is some of my references in this presentation. Thank you.
Dipun, good presentation. Can you hear Dipun?
No, I'm here.
Oh, okay, okay. So good, good work. I have a question. Now, from the leaves, you have prepared extracts. So then, how about chlorophyll?
It's like during chromatography. Now, in my presentation, I said that we carried out solvent extraction.
Yes.
In our solvent extractions, most of chlorophylls were removed into other solvents.
What do you mean by other? Now you
And
have used the chloroform extract. So now it's a non-polar. So, and I saw it, there have been green color also, right? The extract?
Yes, it was green color actually.
Okay.
But when we are doing the testing, we did the solvent distribution between hexane and ethanol water solvent.
Okay.
From that, most of those we observed they went to hexane layers, and that was not that cytotoxic to lung cells as well.
What do you mean by most of it? So still there was some
then
chlorophyll.
The chloroform also was kind of green.
Okay.
But when we carried out those chromatography and those things, most of them were
You need a separation.
Yeah, anyway, chromatography, of course, you can separate.
And other one is
yes.
from chromatography, from silica column.
Yes.
The first fraction is the one that had the cytotoxic molecule.
Okay.
And that actually didn't have any amount of chlorophyll.
Yes, after compound isolation, fine. It's just that for the extracts, you would have done some preliminary anti-cancer work. So then the chlorophyll would have interfered, but then your compound isolation is fine. So then finally you are getting a pure compound. Yes. So I know because sometimes even I find it very difficult to get rid of chlorophyll, but then it interferes.
But one thing we observed is matter. Now, we didn't include them because it is not a part of this presentation.
Okay.
We did the same thing with the Mangifera indica leaves as well.
Okay.
We also have chlorophyll. And chloroform actually in this experiment also they went to other layers as well. Hexane layer and those. They didn't show any cytotoxicity in H292 cells. So from those, I mean, we actually, as we separate them, we didn't worry much about chlorophyll.
Yeah. So I mean, that's fine because you have gone for compound isolation. It's just that the initial extract, I mean, there are lots of methods to remove chlorophyll, but I mean, I'm not very sure about how effective you can use activated charcoal, all these methods. But then sometimes even like phenolics go out, so it's a problem. Anyway, so I'm just a bit curious to know. Now you said this is used in the leaves of this Ethnogenesis, right? Ethnogenesis is used in Ayurveda to treat cancer.
It's not actually leaves, madam. Its bark is used in traditional medicine.
Okay.
Leaves, no one has done so far in a research.
Okay.
How about the bark?
Bark previously was a project carried out at IBMBB. Dr. Meran Nedrivit isolated 2 new anti-cancer compounds. They act on breast cancer, and they have been isolated. And this is the first time
leaves have been studied. Okay, Dipun. Okay, good work. Yes.
Good day, everyone. My name is Shumana Jaffer, and I'm going to be presenting on my research and its findings, conducted with the help of my supervisors, Ms. Hishani Mudarge and Ms. Rominda Ferreira. The research is on Alzheimer's disease and the protein docking of phytochemicals against its receptors. This here is an overview of the content that I'll be presenting today. Let us briefly look into what Alzheimer's disease is. Alzheimer's disease is a multifactorial neurodegenerative disease that affects the brain regions responsible for cognitive functioning and memory skills. It is one of the most common causes of dementia and is detected in individuals over the ages of 65. Early onset Alzheimer's is detected in individuals between the ages of 30 and 60. AD is mostly sporadic, with over 50 million individuals worldwide being affected by it, and this rate is expected to increase twice the amount by the year 2050. Moving on to the pathology of AD, the main cause of AD is yet unclear. However, there are a few hypotheses that explain the possible causes, one of which is the formation of beta-amyloid aggregates due to the abnormal cleavage of amyloid precursor protein by beta secretase, which is then further cleaved by gamma secretase. Another risk factor involved in the development of AD is the presence of apolipoprotein E4. Symptoms of AD include the loss of memory, speech difficulty, difficulty in rational thinking, and difficulty in performing day-to-day functions. As we look into the significance of this research, although AD is detected in individuals over the ages of 65, studies have shown that neuronal damages begin decades before the first clinical symptom is detected. There are currently no treatment methods to cure or prevent AD, and most treatment methods mainly focus on easing the symptoms. As can be seen in this table here, most of the FDA-approved drugs target acetylcholinesterase, as the researches on inhibiting beta-amyloid has mostly been unsuccessful. Targeting multiple elements that affect AD instead of just a single element could help develop an efficient treatment method to prevent or cure AD at an early stage. Therefore, the objective of this research is to identify the best ligand binding sites of AD receptors and to select the potentially effective ligands for treatment. The table here provides an overview of the materials required, which includes websites such as RCSD-PDB and NCBI Public. Let us look into the methodology into detail. The protein ligand docking was performed using Autodoc Vena version 1.1.2. Receptors selected were the beta-amyloid fibril 5-OQB and the ApoE4 gene 1B68. 3D structures of the receptors were obtained from RCSB-PDB in PDB format, while the 3D structures of the ligands were obtained from NCBI PubChem in SDF format, which were then converted to PDB-QT format using OpenPub. Preparation of the receptor using autodock tool was performed, which included the removal of water molecules and addition of polar hydrogen. The grid parameters were set based on the active and binding sites of the respective receptors obtained from various articles. The docking was performed for 10 runs using command prompt, and the outputs obtained were then further analyzed. Let us look into the results in detail. Both receptors were docked with FDA-approved drugs and a clinical trial drug, aziliragon, as control. Out of the docked process, the best binding efficiency was obtained for aziliragon with receptor 1B68. As we further look into the outputs obtained with the docking process of phytochemicals, binding affinity describes the binding strength between the receptor and the ligand. And the lower the binding affinity, the higher the binding efficiency. As such, the highest binding efficiency was obtained for both the receptors with hesperidine with the binding affinity of -7.5 kcal per mole. Docking of 5-QV with hesperidine showed 3 hydrogen bonds, while with 1B68 showed 1. The amino acid residues that interact with the ligands are mentioned in the table here, with the common amino acid residues being highlighted. Further visualization of the docking poses were obtained from Chimera. Out of the 10 runs, the best docked pose of 5-QV with the phytochemical ligand is shown in the figure here, and the docking poses, the best docked pose of the receptor 1B68 is shown here in figure 4. Further analysis of the interactions of the receptors with the ligand was obtained using BioVia Discovery Studio. Binding efficiency depends on hydrogen bonding, hydrophobic interactions, and van der Waals forces. And in the images here, you can see the amino acid residues that interact with the ligands, as well as the hydrogen bonding, as mentioned in green. This image here shows the interactions of the receptors 5-QV, and here you can see the interactions of the receptors 1B68. Further analysis of the ADMET property of the ligands was performed using Swiss Ad to choose the best efficient ligand to be used as a drug. Efficient treatment for AD requires a good blood-brain barrier permeability. The obtained results from Swiss ADME, Citrol and urolithin A were the only 2 ligands with a good blood barrier permeability. Although hesperidine does not have PDB formulation, researchers show that hesperidine do facilitate the blood-brain barrier formation. As well as there are a few researchers which show potential blood-brain barrier permeability in acetic acid as well. Out of the obtained outputs of the Swiss ADB, urolithin A showed the best lightness and pharmacological properties. Further comparing the results obtained in this study with previous studies, there were a few amino acid residues common with the previous studies for this QV, and it is highlighted here. And further, 1B68's results showed the most common amino acid that was found to interact with the ligand was arginine, while the most common amino acid residue found to interact with the ligands from the receptor 5-QV was phenylalanine. Although hesperidine showed a violation of Lipinski's rules, there are researchers that show that hesperidine has potential inhibitory properties, which still could be used as an effective drug. In conclusion, the phytochemicals hesperidine, acetic acid, chromanin, and urolithin A showed potential inhibiting properties against AD receptors. And out of these 4, urolithin A was the most effective ligand with the blood-brain barrier permeability. citral showed the least effective efficiency with the binding. And there were a few previous studies that showed effective binding of such as hesperidine and uracil A against acetylcholinesterase and base. And further research could help develop a multi-target treatment method. These are reference used.
Thank you, Jamana. She's in the Okay, thank you, Jamana. Can you hear me?
Yes, ma'am. Can you hear me?
Okay, same question that we have. What's the basis for selecting these five phytochemicals for your studies?
Yes. So the phytochemicals were chosen based on some of them were chosen based on the wet lab where they tested it on mice, and most that is the citral, and the rest were chosen based on wet lab experiments and computational methods for different receptors such as acetylcholinesterase and beta secretase. So in my research, I'm targeting it with beta amyloid fibril and apoE4 gene.
How about the availability of these compounds in Sri Lankan herbal plants? Did you check on that?
Yes, I did, and that is also one of the factors that's used. Like acetic acid is gotu kola. We get it from gotu kola, and we have pyrimidine and uratinase from pomegranates, and the hesperidine is from citrus plants. You get it from citrus plants, so I based that as well.
Thank you, Jamana.
Okay.
Thank you, Jamana. Good presentation. We can move to the next one.
I'm GVCP Lakshman from Department of Biochemistry and Development, Sabaragamu University, Sri Lanka. My submission number for abstract is 28. Here my topic, preliminary study on phytochemical analysis and Ecms fingerprinting of Sri Lankan traditional polyherbal drug, Panu. Let's have a quick overview. Objectives, introduction, methodology, results and implication, conclusion, future works, and final references and acknowledgement. These are my objectives. The approach was to determine different phytochemicals and their bioactivity of polyherbal formulation. Panu to determine the total content of aqueous extract Panu using the Pollensia caltava assay. To determine the antioxidant activity of aqueous extract Panu using the DBPH radical scavenging assay. Finally, GCMS limited to detect chemicals of ethanol extract of polyherbal drug. I would like to give a brief introduction about my study. Traditional medicine known as Phylogeny has been utilized in treatment of infected disease such as viral and bacterial as a potential alternative. Panu is a herbal formulation made of certain plant by indigenous practitioners in Sri Lanka to treat viral and bacterial infections. Although various traditional drugs are available, their low scientific background has become the main problem on incorporation in modern medical practice. Therefore, the study was expected to measure phytochemical background and bioactivity of the drug Panu. Here we will see the flowchart of my methodology in which lipolysis technique was followed to extract red sample. Then aqueous extract was assessed to total phenolic content and antioxidant activity through the Pollensia caltava assay and DBPH scavenging activity respectively. Finally, analysis will be done in order to exhibit the gallic acid equivalent and IC50 values respective tests. ECMS fingerprinting is carried out parallelly in order to detect the phytochemicals available in sample. Here, sample preparation. Sample preparation would be similar to the traditional procedure followed by the traditional practitioner. Here, 50 grams of dried sample extract in 100 milliliter water transported to the rotary shaker for 48 hours. Next, extract was filtered through the muslin layer. Filtrate was centrifuged at high speed in order to remove the debris. Supplement was freeze-dried at lower temperature for further uses. Finally, the exact concentration of aqueous extract was prepared for experimental purposes. Pollensia caltava method was used to was followed using the gallic acid as a standard range from 1 ppm to 40 ppm, along with the panu extract with exact concentration as 20 ppm concentration. Absorption was measured at 760 nanometer after reliable incubation period. Data were analyzed and represented to generate the calibration curve. DBP radical scavenging activity was performed for the panu extract ranging from 10 ppm to 1000 ppm, along with ascorbic acid as a standard range from 5 ppm to 50 ppm, in which working concentration of DBPH was 100 micromole. A complete procedure was performed under the dark environment to avoid the effect of light on DBPH reagent. Finally, data will be presented in calibration curve in order to calibrate the half maximum inhibited concentration. Here, next, the GCMS fingerprinting was performed in order to determine the phytochemical available in the ethanolic extract, because it is considered as a gold standard for the drug discovery. For that, the Shimatsu single quadrupole GCMS with mid-polar column was utilized. Here I have mentioned the condition reach of each part followed by the process in which a special injection volume and temperature were 1 microliter and 250 Celsius respectively. Run was done for the triplicate sample and detection was done under scan mode. Then data analysis. Under data analysis, results are given at the mean with standard deviation for 3 independent samples. Least square linear calibration curve and logarithm curve were given as 0.99 and 0.90 respectively. Bivariate correlation analysis was done using SPSS software using version 25 by considering 0.05 as a significant level. Here slide shows the results of TB, total phenolic content, in which absorption of gallic acid against each concentration presented following that calibration curve have been plotted with higher regression value. The TPC was achieved as 7.88 plus or minus 0.19 gallic acid equivalent. Here, results of DBP radical scavenging assay. DBP data were evaluated to calculate the radical scavenging activity of both ascorbic acid and panu extract based on the following equation. In order to plot both calibration curve by which IC50 for the ascorbic acid and panu was separately acquired. It was actually the IC50 for ascorbic was 37.42 microgram per milliliter, and while the EC50 for panu extract was 367.00 microgram per milliliter. And also the radical scavenging activity of the panu extracts has a higher positive correlation with the ascorbic acid. It was significant at 0.05 level. All these GCMS results, a total and chromatography were obtained, which results many chemical compound in different level, by which 4 different chemical compounds were extracted selected based on their peak of chemical place. Here I have shown their structures and available data regarding the bioactivity of the respective compound. First, phenol plus bis 1,1-dimethyl ethyl, which was considered as a precursor for the antioxidant for many researchers. And next, 1,4-benzene dicarboxylic acid, also known as a topical antifungal agent and antibacterial agent. And also phenol 2,2-methyl base dimethyl ethyl 4-methyl is also considered as a potential antioxidant. Finally, the benzene propanoic acid and the propanoic acid exhibit anti-inflammatory, antifungal, and antibacterial activity. This is my conclusion. Total phenolic content of panu is moderately low compared to the gallic acid. Panu extract was a significant effect on radical scavenging activity compared to the ascorbic acid. GCMS for ethanol extract of panu detect potential phytochemical generally involved in antioxidant, anti-inflammatory, and antifungal, antibacterial activities. Preliminary study on PANU indicate potent bioactivity, which evaluate the further study on cytotoxic study of the drug. These are my future work. But the qualitative and quantitative analysis will be performed for the PANU extract, and antibacterial and antiviral assay also will be performed for the PANU extract to evaluate the mechanism of action of the traditional drug. These are my references. And this is my acknowledgement. Here I would like to thank for everyone who helped me to this work. Thank you.
Chakura, can you hear me?
Yes, madam. Can you hear me?
Yes. Thank you for the presentation. There are several questions. Now, this polyherbal drug is used for antiviral and antibacterial infections. Am I
Yes,
correct?
madam. Yes, madam.
Okay, then why did you study the antioxidant activity?
Yes, madam. It's because normally both antioxidant and antiviral and bacterial effect is arise from the due to the available phenolic content of the plants. Normally, if I get a measure of any about the phenolic content about the plant extract, it's kind of measure for the antibacterial effect as well. And also, I have referred that there are some normally antioxidant. What antioxidant do is they remove the free radical from the medium. Normally, there are some bacteria they require that monoatomic oxygen to survive. Due to available antioxidant ability, they can't survive. So it is another example for that. That's the reason, madam. And also, it is proven by the madam my GCMS results as well.
Do you think the title reflects the work described in your abstract?
Madam, I edited it because earlier they have asked to edit that my topic.
Well,
I hope that my presentation topic is similar as my abstract topic.
the abstract topic is preliminary study on phytochemical analysis and JCMS fingerprints of Sri Lankan traditional polyherbal drug PANU. Is that what you wanted?
Yes, yes, madam.
I think it should be like total phenolic, antioxidant, and maybe phytochemical analysis by JCMS of this PANU ethanolic extract or something like that. Aqueous extract.
Do you have any idea about the composition of this herbal drug?
Actually, madam, in one slide, I have given the plants available, but actually the traditional practitioner given me the crude extra.
So it's a secret recipe, I suppose, right?
Yes, ma'am.
Is it a powder, right? Is it a powder?
Actually, they give us as a powder.
And I think what you have done is not a fingerprint, Daruo. So you
Yes,
have
madam.
identified several phytochemicals. So this is GCMS. So how about the MS? To which extent did this tally with the library compounds?
Actually, madam, we follow the libraries NIST 2017.
Yes.
Depending on that.
What was the percentage?
Madam, actually, madam, we take these phytochemicals according to the similarity score. So again, we are not quantifying it because we don't have any standards regarding phenolic compounds. So we just identify only the phytochemicals that give us the similarity score beyond 90.
Yes, that is right. Beyond 90%, right?
Yes, madam. Yes, madam.
So, okay.
Now, for this phytochemical analysis, you have used the ethanol extract. Am I correct? But for the studies, it is the aqueous extract.
Yes, madam. I use that. We use ethanol extract for the GCMS, while the water extract for the other two experiment. Because ethanol, normally the GCMS cannot run through the with the water, because they have recommended that water can affect the my column, and actually water are going to be oxidized column and the
Yeah,
part of
but
the
you could have freeze dried the water extract and then dissolved it in like ethanol or methanol. It
yes,
would have
madam.
been nice if you did it in the same solvent, so that there is some correlation.
Yes, madam.
Yeah, thank you.
Thank
Thank
you,
you.
Chatura.
Thank you, madam.
It's my great pleasure to introduce our keynote speaker, Raj Somadeva, somebody whom even the public know very well. With his programs telling us about our history and our origins and so on. Professor Raj Somadeva is a professor in archaeology at the Postgraduate Institute of Archaeology, University of California, and he has been in this field for nearly 33 years. He's doing a special honors degree in archaeology from the University of California, MPhil in archaeology from the TGIA, and also from the University of California, and obtained his PhD from Sweden, from the University of Uppsala, which also has many connections to Sri Lanka. And he studied urban origins in southern Sri Lanka. He received many professional trainings and field experiences related to aspects in archaeology, and currently serves as a fellow of the Sri Lanka Council of Archaeologists and a member of the World Archaeological Congress. He disseminates knowledge as a consultant in archaeology. He has obtained several awards, including top 10 in Sri Lanka Award for the category of academic leadership and accomplishment in 1998. Charles Wallace Research Fellowship from the Institute of Archaeology, University College London in 2005, and Vice Chancellor's Award for the most outstanding researcher of the Postgraduate Institute of Archaeology in 2023. He has directed over a dozen master's student research projects and many undergraduate projects in archaeology. He is the author of many books, including the Rupai Survey and Archaeology of Herbal Basin, rock paintings and engraving sites in Sri Lanka, History of Sri Lanka, and civilization. Apart from that, there are over 50 publications comprised of 4 papers, both internationally and locally, abstracts in international seminars, and articles in local journals and symposia. Currently, he serves as consultant in forensic archaeology of the mass grave recovery manner, and you can read his work from his webpage www.rajsomadeva.com, and if you search YouTube, you will find a series of presentations to Netflix, which are very, very intriguing to listen to. And he is also one of our collaborators whom everyone is working on ancient so many.
Professor Prasanna Galhena, the Director of the Institute of Biochemistry, Molecular Biology and Biotechnology, Professor Tenkon, Deans of Faculties, Professors of various departments, Heads of departments, dear colleagues and friends. When I received the invitation from your institution, I was felt by a certain uncertainty. The uncertainty was to decide what should be the theme of my presentation to this distinguished audience, consisting of professionals who are situated in an orbit that is far beyond my field of study, that is archaeology. Archaeology is fundamentally the study of humanity and its past. Archaeologists study things that were created, used, or changed by humans. They do this by studying the material remains, the stuff we leave behind, such as lithic tools, a simple hut dwellings, skeletons covered with gold jewelries, or a pyramid that majestically rises from the desert floors. Sometimes archaeologists study contemporary societies in order to shed light on those that perished in the past. Archaeology is practiced around the world by archaeologists who work with people from a wide variety of other disciplines to help answer questions about whom we are and where we came from. In doing so, archaeologists find evidence that shed light on what our future may bring to us. Hence, why don't we talk about ourselves? The way we all came from the path of an unperceivable history. The diversity of our genetic makeup. The history of the evolution of our cognitive advancements, including the thinking on engineering and medicine. All such are hiding by a fog of time. Anyway, it is a great pleasure to be here today to address you all. It is also an honor to me to invite to deliver the keynote speech at the 3rd International Conference on Frontiers of Molecular Life Sciences and Life Sciences organized by your institute. As we all are explicitly aware, we are biologically related to the anatomically modern humans. Anatomically modern humans are anthropologically known as the archaic Homo sapiens that was first appeared at least 300,000 years ago in Africa. This has become known to us through diverse lines of inquiry, including overall mitochondrial DNA diversity, deep Y chromosome ancestry, parasitology, paleoclimatology, and mostly on paleontology. It is accepted that the emergence of full-fledged modern behavior is manifested by several key indicators, including the introduction of language, music, dance, and the development of image-making skills, and so on. Let me focus on one among the many. It is biology. As we are all familiar with, biochemistry, molecular biology, and biotechnology are the subdisciplines in biology. It has continuously expanded their own academic pursuits among the other disciplines of human knowledge. It has been unveiled the mysteries of life on our planet on an unprecedented scale. Sometimes it studies the life spectrum from the parasitic bacterium like Mycoplasma genitalium, which lives in the primate blood, to the largest single cell organism in the world known as Culepra taxifolia. Let me now turn to the objective of my presentation today. My intention is to elaborate on the way that how archaeology gains intellectual benefits from the high-resolution biological sciences like molecular biology and genomics. The fresh avenues in mainstream archaeology, for instance, subjects like cognitive archaeology and behavioral archaeology are entirely dependent on the information derived from the biological sciences. And it shows how much such scientific disciplines have influenced our understanding of the past. If we look through historical sciences like archaeology from a global perspective, especially the population prehistory, what one can see is the co-evolving parallel trends in historical linguistics and genetic studies. The epigenetic origin of the modern linguistic phylum has shown its association with wide geographical regions. It is explicit that languages had drifted from place to place via human migrations, at least since the Neolithic phase. Migrations of Austroasiatic populations from the Far East towards the West has resulted in the spread of Tibeto-Burmese languages in Southeast Asia, and the substrate languages like Kusunda and Burusaski of the Munda family in South Asia are bold examples. It highlights the relationship between the linguistic makeup and the population migrations held in the Old World. The only reliable information pertaining to such human migratory waves brings to us by the biological sciences, especially the genetic sciences. Dispersal patterns of ancient languages have been considered as a strong indicator of prehistoric population distribution all over the world. Archaeological evidence provides parallel clues as complementary to elaborate the relationship held between linguistic dispersal and population diversity. Such diffusion occurred in a time which was far beyond the past, when before the written records emerged, and therefore narratives are absent about the prehistoric linguistic interactions held at that time. Most of the histories belonging to the diverse nations in the world have memories of such population migrations. Memories of migrations related to their ancestral populations appear in different forms. The stories pertaining to the peopling of Sri Lanka are also enmeshed by such a story that is strongly rooted in the public consciousness of their identity. The arrival of a gang who had North Indian cultural origin and their subjugation of the native, then being inhabited the island by them, is the major content of that story. The idea of population migration associated with mainland India brings us a sound theme to be analyzed scientifically. Any attempt to be focused on the study of the composition of the prehistoric population in Sri Lanka has to scrutinize the population dynamics in South Asia from a broad base of biological point of view. Perhaps it might be spatially incorporated further into mainland Southeast Asia. Analysis of the complexity of the composition of the lingua franca in mainland India is an important point of departure to formulate a conceptual framework on the biological inheritance of the prehistoric population in Sri Lanka. On one hand, the language family of Indo-Aryan derived from a Central Asian wave of linguistic influence that occurred 3.5 kilo years ago, dominated in most part of the northern part of mainland India, even including Pakistan and Bangladesh. On the other hand, South Indian Peninsula was dominated by Dravidian languages. This was a result of a high level of endogamy, triggered by the strict social boundaries and a high degree of genetic drift, encouraged by long-term isolation. The investigations carried out using mitogenome analysis of autosomal data and Y chromosome lineages on the population history of mainland India suggest that the maternal lineages emerged 55 to 65 kilo years ago, and a major population shift triggered in the late Pleistocene. Linguistic information provides a beacon to resolve the problems related to those characteristics. The inspiration of the rises and falls of such dynamics might shed a light on determining some of the dilemmas posited on the genetic inheritance of the prehistoric population by the recent archaeological findings in Sri Lanka. Before being too specific on the population prehistory in Sri Lanka, it is unequivocally important to survey the evolutionary phylogeny of the prehistoric population in the wider Asian regions, including South and Southeast Asia. While doing that, it is more important to rely on high-resolution scientific information, which is being updated continuously. Since 1960s, archaeological research in Sri Lanka had gradually embraced the scientific rigor of biology and biological anthropology at a very slow pace. The first attempt at the application of the methodology in physical anthropology can be seen in the case of the analysis of prehistorical human skeletons recovered from Balangudu, popularly known as the Balangudu man, recovered from Bellambendi Palace in Balangoda. The site of Bellambendi Palace is situated in Balangoda of the Ratnapura district of Sabaragamu province. The excavation was carried out in 1956. During the excavations of 1956, the remains of some 9 individuals were uncovered. However, the laboratory examinations of the bones had not been undertaken until 1960s and 1961, when the specimens were sent on a loan basis to the British Museum in London. The anthropometric and comparative analysis of human remains from Bellambendi Palace posed certain problems of interpretation that demand a more sophisticated analytical methodology, notably regarding the questions of the phenotypic affinities of the species recovered. Late Dr. P.E.P. Dharanekala, a well-known physical anthropologist, is of an opinion that was the supposedly Autochthonous Vedda of Sri Lanka possibly carries some proportions of the blood of Balangoda man, and he tended to designate the Balangoda specimens as proto-Veddoid. He further states, quoted, "The blood of Homo sapien balangodensis, however, exists in almost equal intensity to the blood of so-called Vedda, and also in Sinhalese, since appearance of the fossils from the Bellambendi Palace," unquoted. Dharanekala has insisted that the balangodensis show the combination of Neanderthaloid and Australoid traits that he called Proto-Australoid. He suggests that the balangodensis replace the people of Ratnapura culture, who are the true Autochthonous of Ceylon, and that the balangodensis continued to practice their distinctive culture until the dawn of the historical period in the 5th century BCE. He concluded the Vedda as a degenerate mixed population that inherited the racial elements of balangodensis and Sinhalese. The number of osteological specimens collected from Sri Lanka since 1827 and bearing the name Vedda on their label is impressive and exceeds in size the series accessible for many other Asiatic tribal groups of higher population frequency, and the amount of anthropometric data pertaining to both osseous and living Vedda specimen is not inconsiderable. The Balangoda skeletal remains from Bellambendi Palace constitutes a unique phenotypic pattern present in Sri Lanka at the dawn of the historic period, a time within the 2nd and 3rd millennium BP, while the balangodensis fall within a range of polytypic variation representative of post-Pleistocene Homo sapien of the Indian, Southeast Asian, Australian gene pool. The phenotypic pattern most closely resembling that the balangodensis is that of the Veddas in Sri Lanka. To understand the genetic composition of the native population in Sri Lanka, it is important to review the recent results derived from the studies conducted focusing on population genetics in the South Asian region as well. The greater Himalayan region, including the Tibetan plateau in the north and the Gangetic plain in the south, served as a principal prehistoric thoroughfare for the peopling of East and Southeast Asia. Several of the keys to understanding the ethnogenesis of human diversity in Asia include the father-tongue correlations, possible refugia during the last glacial maximum, and the hypothesis that language families may have arisen as a result of demographic bottleneck in prehistory. Ethnolinguistically informed inferences based on Asian Y chromosomal phylogeography permit a construction of episodes of ethnolinguistic prehistory which lies beyond the linguistic event horizons, that is beyond the time that empirically accessible to the historical linguistics. The origin of language families, which make up the hypothetical Uralo-Siberian and East Asian linguistic phyla, are argued to have lain in the northeastern corner of the Indian subcontinent. Several other Asian language families are shown to be tied to the Indian subcontinent. Early human population outside of Africa no doubt must have interacted with each other in parapatric or sympatric modes at various junctures in their long prehistory. There were numerous waves of peopling, and the genetic evidence is compatible with sustained and multiple migrations out of Africa through the Levant region over time. Evidence has been adduced of gene flow from the now extinct Denisovans into the ancestors of the people who ultimately settled Melanesia. As temperature and humidity increased after the last glacial maximum, the Y chromosomal haplogroups O split up into the subclades O1, O2, and O3. The three subclades can be putatively assigned to the three geographical loci along the east-west axis for the sake of argument and without any claim to geographical precision. Human genetic studies suggest that the paternal lineages N and O may have split in East Asia between 30,000 and 20,000 years ago. The spatial dynamics of the two haplogroups diverged greatly after the split, and the ancient Asian populations which bore the Y chromosomal haplogroups N and O are calculated to have undergone an effective expansion between 18,000 and 12,000 years ago. At the dawn of the Holocene in the southeastern Himalayas and the eastern declivity of the Tibetan plateau, haplogroups 3 gave rise to the ancestral trans-Himalayan or Tibeto-Burmese paternal lineages O3A and 3C. And the original Hmong Mien paternal lineage O3A and 3B as well. The bearers of the polymorphism O3A, 3C stayed behind in the area comprising northeastern India, southeastern Tibet, and northern Burma, which the bearers of the O3A, 3B lineages migrated eastward to settle in the areas south of the Yangtze River in China. On their way, the early Hmong encountered the ancient Austroasiatics, from whom they adopted rice agriculture. It has been proposed that the Y chromosomal lineage L, which shows a great diversity of subclades on the Iranian highlands, can be identified as a possible marker of a patrilingual dispersal of Elamo-Dravidian emanating from a region which included the Bactria and Margiana complex of late prehistory. One of these Y chromosomal L subclades appear to be correlated with patrilingual spread of Dravidian languages from the Indus Valley into South India. We may infer that, in part, the Baluk represent in situ descendants of the ancient Dravidians in Balukistan, who peopled the Indus civilization. The ancestors of the Baluk, however, opted to assimilate linguistically to the incursive Indo-Europeans and retain the originally predominant Elamo-Dravidian haplogroup L at the highest frequency. By contrast, the Brahui retained the original Dravidian language of their ancestors and consequently also inherited the lower social status of the subjugated Indus population. Ironically, the lower status connected with the retention of a pre-Indo-Aryan ethno-linguistic identity rendered the Brahui language community more prone to male-biased genetic contributions from incursive Indo-European group through hypergamy practiced by succeeding generations of Brahui women. Confronted with the overwhelming growing body of evidence in support of the father tongue hypothesis, Foster and Brentrow impute the spread of language families to emigrating agriculturalists who took local wives. This interpretation is a transparent attempt to support Peter Bellwood's and Colin Renfrew's embattled first farmer hypothesis, which seeks to ascribe the founding dispersal of language families to the spread of agriculture. Those collaborating with Renfrew's continue to seek Y chromosomal correlates for the spread of the agriculture horizon in Neolithic, even when the reasoning continues to be strained and the purported correlates are not manifestly evident. In order to buttress Colin Renfrew's widely doubted hypothesis of an Indo-European homeland in Asia Minor, Foster and Renfrew propose a correlation of Indo-European with the Y chromosomal haplogroup J2A. During the Neolithic horizon, the spread of farming was necessarily a sedentary and incremental process, which likewise must mostly have involved both sexes. Early farmers might only have been able to spread their language at time of great surplus and concomitant population growth, perhaps sometimes involving the establishment of agricultural colonies elsewhere. By contrast, the modern ethno-linguistic composition of Asian populations must be understood, at least in part, as having resulted from male-biased linguistic intrusions, whether motivated by conquest, land grab, or the urge to seek out new habitats. Biology as a discipline has an unimaginable potential to serve for solving problems we're still waiting to find answers to. It is not only confined to our present, but also can utilize to resolve some important issues related to history of mankind that is remain unanswered yet. This urges us to work hard on the rigid scientific frame of reference. Our knowledge of our biological identity still suffering ambiguity. As professionals of different disciplines, this is the time to work together for our country and the nation. Let me wish you all the courage to make it a reality. Thank you so much for listening. So I want to mention one thing. The contribution of biology is not only confined to our present. So it can be utilized to resolve some important problems, which belongs to the future. So my presentation today, the main objective is to elaborate the potentials now being emerging to merge the historical sciences with extremely complex biological sciences. So as archaeologists, we have different problems that we can't solve by alone. So we are asking different professionals from different disciplines to help us in different point of views. Especially in archaeology, we are dealing with extremely fragmented artifacts, no stories, no animations, nothing at all. So the human remains and also the artifacts they produced have to be animated to make a complete story for the sake of the future generations. So this is the important juncture. You all represent the biology, art science, but we are sociologists. Now we are in an important juncture to merge and bridge the different streams of knowledge into a common, to see a common goal. Thank you very much for listening to me.
Thank you very much for your specific presentation for today and sharing your time, knowledge, and expertise with us. In fact, the point that you have highlighted here is a timely requirement. We look at how we can contribute to the timely requirement that we can make the bridge between the archaeology and the modern science. But in fact, I think the IBMBB is making some contribution with the work that is taking place with Continental and One. In fact, the day I came here, I was really encouraging that kind of a work with the objective of which is not a what do you say, the profitable investment for us, but in fact it is our duty in order to make this strengthening on this field. So that is what I said, that the same idea was shared with Professor Continental. We need to really focus on this line of investigations, or perhaps as a service, and make a repository and how we can move forward. That is what exactly we need to look at. And probably we will not be in a position to get a, what you call the industry finance. But in fact, we can look at some alternative funding, perhaps which I was aware. We have a bit of a funding on this cultural triangle. That is one of the sources that we can really think of and make use of that funding in a very effective way and very meaningful way. So that is what we are focusing on. And perhaps I will give my plus support for this line of work, because I also feel that it is unexplored area. So with that remark, I thank you again for Professor Rajkumar for your presence here and making the keynote address for our International Conference of Frontiers of Molecular Life Sciences 2022. Thank you. Okay.
background is in linguistics, basically looking at more on the language situation in this country, and also as a linguist, I have interest in generally in South Asia. For my PhD and my doctoral work more focus on South Asia. So what I'm going to focus for this symposium is about just to revisit these debates about the origin of Sinhala. Some of you are aware of these famous debates about the origin and evolution of the language, and I would like to provide a kind of a historical linguistic perspective to this symposium. So thank you very much for bringing this aspect and also kind of bringing this diversity to this your institute. So as a colleague and as an academic at the University of Colombo, I'm very happy to bring this kind of perspective to this symposium. I think just I'm not going to elaborate all these things that we listened to Professor Somadev as well. As linguists, we kind of think about these language families. We know these language families are based on basically on genetic relation, not genetic in your sense, but genetic relationship with the languages. So that's one thing. Language family is it's a branch of historical linguistics. So looking at the genetic relations between languages. Sinhala, we know the language spoken by the Sinhalese is a Indo-Aryan. It's not a family. It's a subfamily of the Indo-European family, which belongs to the English and some other European languages as well. But it's an Indo-Aryan subfamily. It's also linked to other Indian languages like Hindi, Marathi, Odia, or earlier Oriya, and Bengali. But what is interesting is, from a geolinguistic perspective, our nearest relatives are not Indo-Aryan. Our nearest relatives are the Dravidian languages. When you say Dravidian, it's not Tamil. But I think 20 years ago, I compiled a book. I think Professor Somadev knows about this, a dictionary of Tamil words. But when you say Tamil, some of these, just take the word Kannadi, it's from Tamil. Kan means the eye, Adi means the glass. Kannadi is from Tamil, but we think it's from Tamil. But it's also related to Kannada, Telugu, and Malayalam. So when you say all these languages, Dravidian, then the Dravidian is not only Tamil, but it includes Tamil, Malayalam, Telugu, and Kannada. So we are the close relatives of Sinhala are not Indo-Aryan, but Dravidian. So it's interesting. But except there's only the Divahites Indo-Aryan involved. So there is a Dravidian, or you can say it is a Dravidian belt or a Hindi belt. So there's a connection between these two. So because of this isolation from the Indo-Aryan group, the Sinhala developed along some independent lines. So I would say as kind of separated from the other Indo-Aryan. So some linguists say it's a kind of a miracle. It's a kind of a linguistic miracle how the Sinhala survive as Indo-Aryan. Like at the Cornell University, Professor Gale said it's a kind of a linguistic miracle how the Sinhala language retain its Indo-Aryan character. Our close relatives are not Indo-Aryan language. So this is another area that how these as in island, maybe because of that, how this Sinhala retain its Indo-Aryan character. So this briefly about these relations or the genetic relations means from a linguistic point of view with these languages. So it's interesting to note that Dravidian, the South Dravidian, but except for the Brahui one, Professor Somadev mentioned, it's in the north. But all the other languages are South Indian languages are Dravidian, except the Brahui, which is in the north. Okay, so apart from that, there is another stock of Sinhala words we call Nishpanna. The 13th century Sinhala grammar we call Siddharth Sangarava. So Sangara, it's not a magazine; it's a treatise of grammar You say Siddharth Sangarava, someone can ask, is it still publisher? Siddharth Sangarava? No, it's not a Sangarava. It's a compendium of rules about that. It says there is a category of words called Nishpanna. Nishpanna, Tatsama, and Tadbhava. The Nishpanna means indigenous. Heladiva Palanipanna. Originated in this, say, originally, Heladiva Palanipanna. That is the definition given in Siddharth Sangarava. So there is a stock of words not related to any other Indian languages. Just take the word Kata, Bada, Uluva. So Kata, we just take the Uluva, is the Sanskrit word is Shirsa, Hisa. So Hisa is a Tadbhava, derived from Hisa. But when you say Uluva, there is no such other Indian connection. But I'm not saying, but maybe we have other not Indian, but other maybe Polynesian. Now, I think a few years back, I met a colleague when I was at a conference, attending conference in Vietnam. Academic from Indonesia. He came from Bali. So we had in the afternoon we had a chat. He said, in Bahasa Indonesia, they said ulu for head. Ulu for head. We say Uluva. But Uluva is not relevant, it's not equivalent to any Indian language, but maybe for other areas, the Polynesian line, Malaysia, Indonesia, Sumatra line. So that's a less research area in language studies in Sri Lanka. Not only Sri Lanka, but in the Indian country. But we have a stock of words, Kata, Bada, Uluva, Kakula. Kakula, we know the Pada and Pa. Bada is Udara. Right? Udara is a Sanskrit. But all these words, but there's all the names of our villages, your villages, which you say Piliandala, or Borleskam, or Oberal. There's no Sanskrit words in our place names. So all these place names and also names of our trees, flora and fauna, all called Nishpanna. Nishpanna means Heladiva Palanipannam, that is indigenous. Right? So that's when, so because of that, there is this composite character we need to look at how this is Indian languages and local inherited language. But at the same time, just to revisit this debate about origin of Sinhala, the dominant legend about the origin of Sinhala is this the story of this Vijay. Story of Vijay. We know this Mahavansa, an Indian prince. Perhaps somebody say a gang. Yes, okay, we'll accept the gang. The gang visited, settled down here, migrated in the 6th century. That's the legend or the historical what Mahavansa says. So it is accepted that Vijaya, the son of Singhabahu, was the founder of the Sinhala race. That is, it's a common fact. As school, that's not as expert, but as our school, even in our curriculum. So it says that Sinhala was introduced to the island by him and his followers. But what's interesting is, from a linguistic point of view and also from a geolinguistic perspective, so where's this Prince Vijaya's original home, the Singapore? The exact location has not been identified yet. There are different views about it. There are conflicting views about this region, which is the home of Vijay. So why this is a kind of a conflicting arguments? Both the historians and the linguists found it difficult with the exact homeland of Vijay. There are a few reasons, as I quote from Sana, maybe. One reason is, he says, the composite character of this myth. This myth or the legend is not only here in local chronicles, but in the other, some other South Asian evidence is also there. Narrated in locally also, is the composite character of them. And also the place name, this Lala or the Paraka and all these place names that appear in these legends and also in these languages have been identified and interpreted by different scholars in the chronicle. So that is one. So where's this place? Whether it is from eastern side of India or from the western side of India, or whether it is from Dravidian, the southern. So there are different views about it. They came from, whether it is from Bengal or from Orissa or from south. So this is. And the other thing is the composite character of this Vijay's retinue, the followers, whether it is one group of people or so. There are, I'm not going to go into detail about this. There are different views about this, but we need to consider this legend as well. And so at the beginning of this last century, that's very interesting because we need to revisit this debates about the evolution of language was occurred around, I think, in 1930s or 30s. But it is interesting to note that one of the early scholar that expressed views about Sinhala language was a lecturer in Oriental languages at the University of College, Reverend Rabukweli Siddharth. But that I'll be launching a coffee table book on Saturday on College House. I added a picture of Reverend Siddhartha. So this debate was in 1930s. Some of the scholars were attached to the University College at that time, faculty of, not the faculty, the Oriental languages. So these debates focus on the Indo-Aryan linguistic identity and the affinities in Sinhala, and not only Indo-Aryan but Dravidian. So that was whether there was a debate in 1920s and 30s whether Sinhala is a Indo-Aryan or Dravidian. Even one Sinhala scholar, W.F. Gunawardhan, one of the editors of the Sinhala dictionary, argued that Sinhala is not an Indo-Aryan. Sinhala is a Dravidian. His words are, Sinhalese is a Dravidian language with an Aryan wash. Aryan wash. So, but later on, because of the Sinhala dictionary and the historical linguistic work of Geiger and others, they proved historically this is Indo-Aryan. The correct is Indo-Aryan, but there is originally Indo-Aryan, but there are stock of words from Dravidian. So these debates was like started in 1920s and 30s. There was a group of scholars from the University College and from the dictionary and the Royal Asiatic Society. There was a big debate. So at that time, there was no such molecular biology or genetic evolutions about this. No such a discipline in this country. I don't know about the other countries. There was no such biological kind of approach to look at these migrations. And again, there's another argument about this, whether these early speakers of Sinhala, whether they were from east of India, Kalinga, they say in Kalinga in Oriya, in Odisha, or we say in Kalinga, or the west of India, or the Gujarat. So that was the debate, whether they're from Orissa, or the old name Utkala Desha, whether it's in the Indian anthem say Utkala, there is Orissa, Kalinga, or from the Gujarat. But there are different, when you come to this debate, there are the linguistic factors, then the genealogical kind of narratives about this Vijaya and all others. And there are some very interesting geographical locations we need to again revisit. We need to revisit these geographical locations. I had a chance some time back in 3-4 years back before this COVID in 2015, visiting Orissa, Kalinga. It's very interesting to look at the places that connected with the history, especially the story of this Danta Kumara and also from various the Kalinga kingdom. And the ethnic relations also important. And now we can add this biological kind of, especially genetic aspect as to this debate. Right. And what is interesting here is whether when all the scholars, especially the single scholars in 30s and 40s.
Recording in progress.
the sources that is available in the 3rd century about the Sinhala. There were several attempts by linguists to solve this problem of the original place of Vijay, or the original place of the Sinhalese, by looking at these historical traditions and the comparative studies of Sinhala. And the main argument was about the identification and the location of this Lal or the latter about the homeland of Vijay. This issue here, not moving. It's not working there. Right. Right. So I mentioned about 2 hypotheses. One is the Eastern, and the other one is the Western. The scholars who support the Eastern hypothesis with Sinhala, and also with these Ashoka inscriptions, Ashoka Lipi, they point out that Sinhala agrees more with the East Ashoka dialects. East Ashoka dialect, that is the Kalinga area, the Orissa, rather than in the West, and conclude that Sinhala originally was an Eastern Indo-Aryan dialect. So what's interesting is there were Rabukwala Siddhartha, who taught Sinhalese at the college house in 1930s, argued that, I quote from Reverend Siddhartha, if one studies Bengali and compare it with Sinhalese, one will surely say that Sinhalese is directly connected with the Bengali, the language. And again, if one would study Oriya, now it's Odia, now they call it Odia, and compare it with Sinhalese, he will say that Sinhalese language is nothing but a modified form of Oriya or the Odia from Kalinga. So that is the linguistic evidence provides for this Eastern hypothesis of Sinhala. Right? With Oriya, I don't know, but as far as I know, there is no such Oriya scholars in this country, Odia. When I visited Orissa some time back, I tried to study, but there's a different script, that's issue. Oriya script. But languages, some of the there are similarities between many. So what this Reverend Siddhartha says is very important, that it says whether it is Bengali, but whether it is a Bengali Oriya, is that it is eastern part of India. So that is one debate. And the early Brahmin inscriptions of the island also reveal that these migrants from eastern India form the foundation, the early settlements. That is from the evidence that we provide. There's the groups are also there. Another historical linguist, P.B.F. Vijayaratna, who studied the phonology of Sinhala, he says, he was one of the editors of the Sinhala dictionary, etymological dictionary. He says the early language shows that it must have originally belonged to some eastern tract of India by its salient eastern characteristics. This is by P.B.F. Vijayaratna. That is from his PhD in 1940s at the SOAS, University of London. So commenting on this debate was dominant in 30s and 40s. That was the main. So Gunawardhana and others, Reverend Jnana Prakasa. So they proposed it was a Dravidian, but when the Geiger's work and the historical linguistic work supported this Indo-Aryan character, but the debate still continues. Debate continues about whether it is Eastern or Western. The Geiger and Jayatilaka, the found editors of the dictionary, confirm that in this dictionary, this is available. The base seems to be So there are different views. The base seemed to be a Western dialect brought to the island by the first Aryan colonist, but this base overgrown with new elements. That's very interesting. Overgrown with new elements imported into Ceylon at various times, probably already soon after the colonization, and from different parts of the continental India, chiefly from the east, that is Kalinga, or they say in Kalinga. Yeah. So that's supported by the views of the Jayatillaka and the Geiger, the two main scholars in this debate of origin of Sinhala. So they say it's a base seemed to be a Western dialect, but it was other elements, base overgrown with new elements. The base overgrown with new elements imported to Ceylon at various times. So this is interesting. And there was a strong belief in medieval Sri Lanka. The location of Singapore was Kalinga, the ancient Odia. But some time back, for this, for a volume published in Orient and Black's Form in India, I think last year it was published. I wrote an article on origin of Sinhala and Kalinga Lanka connections, Kalinga Lanka affinities. As I quote, this reference to that, Kalinga Lanka affinities. But these connections between these Singapore was Kalinga. So that is the debate about this Eastern and the Western hypothesis about the origin of Singapore. From a different perspective, again, about the recent work about this, I'm not going to refer to these genetic aspects of it. Now, as I mentioned earlier, this Kalinga, you can see the map, the Kalinga or the Kalinga area. This is the Marga and all this. They were Nisankamalla, Marga, right? They all come from, they were from Kalinga. And I also say that Guru Gomi, not a relative of mine, but I'm Sandu, but Guru Gomi also. My students asked me in my first year that Guru Gomi was from Kalinga, Elua. Samaj was from Kalinga, from Polonor period. But this ancient Odisha, Singapore was from the Kalinga, ancient Odisha. From different aspect. And there are recent studies, that is, maybe some of you are aware of this work. I don't know how this is acceptable in your area, but as a scholar in the field of linguistics, I'm also interested in these, looking at these other aspects. The genetic affinities of Sri Lankan populations, is a Gautam Kumar Shastriya found that 25% of the genetic makeup of the Sinhalese population was contributed by the Bengalis. But maybe later on, the presentations in this symposium will provide more kind of input to this debate. And a very interesting book, I don't know whether are there any readers who have read this work. It's available, I don't know whether it's online available, but it's very interesting work by Sanjeev Sanyal in a recent book, not recent now, it's in 2017, The Ocean of Churn, how the Indian Ocean shaped human history. Now the studies about, even in the languages we call Indian Ocean studies, Indian Ocean languages, or Sinhala, Divehi, or Indian Ocean languages. They are not talking about the South Asian, it's a more Indian Ocean. In a recent book, The Ocean of Churn, how the Indian Ocean shaped human history, Sanjeev Zan, very interesting book, remarks the symbol of the lion that is so important among the Sinhalese, equally considered as a revered among the Odias and Bengalis. It's true. You know, if you go to the Sun Temple in Orissa, Sun Temple, there are so many, the Singha, especially the symbol of the lion, is very common, both in Orissa and also in Bengal, especially the Sun Temple. It's one of the, I think, archaeologically important place. So apart from this linguistic lines perspective, there are other areas that we need to look at. How this Indian Ocean shaped the human history and the connection between populations and languages. So there is another aspect to this story about this. That is the Dravidian, or for us, as Sri Lankans, the Tamils. The Tamils of Sri Lanka, also close to Sinhalese because of they were like separated from the as island, we are separated, but this close proximity to each other, historically, linguistically, and culturally. In my 2001, my dictionary of Tamil words in Sinhala, I pointed out that 20% of our spoken Sinhala words are Dravidian, Tamil or Dravidian. Like you say Udau, Padi, Amma, Tata, and all this, especially the spoken Sinhala. Even Reverend Siddhartha also supported this, pointed out this, that there is a large, kind of not large, it's a kind of a percentage of the words in spoken Sinhala that we use in the day-to-day language, even all the kin terms, Ayya, Amma, Amama, and all, from Dravidian, that is Tamil or from. So this is also interesting to look at how, especially the core vocabulary of core vocabulary. The core vocabulary means our kin terms, day-to-day words, right? All these words. When there is a consonant doubling, say kappam, aitam, vannam, or when there's a double consonant doubling, it's a Tamil word. That's the one way that we can identify whether it is Tamil or not, whether it's an Aryan word or not. So again, referring to this article, this mixture of these revealed that Sinhalese of Sri Lanka have higher contribution from the Tamils of South India, and also compared with the Bengalis of Northeast India. So it's interesting to see this, how now the early debates of Sinhala. So we are now in a position to support these hypotheses from you because of your work on this genetics. So genetics studies meets linguists and can come up with all these debates are basically more on historical linguistic data. So thank you very much for your, sorry. It's not working, sorry about this. All right, sorry. So about this, our presentation. So it is one area is deeply embedded in the history of Sinhala language and people. So we need a kind of a more comprehensive study about these linguistic affinities between Sinhala language, that is about the early phase of its development, and the languages of Eastern India. But unfortunately, most of the departments that focus on languages in this country, especially the linguistic, we don't have a linguistic department in Colombo, but in Caledonia and other departments, and also language departments are more, they are basically, we have Sinhala departments and other Tamil departments, but we would have scholars working on, say, Oriya or Odia or Bengali. We don't have any Bengali scholars or Odia scholars working, looking at the both language history, and also look at from a comparative linguistic perspective, bring comparative linguistics, and also from historically to bring these languages, Indian Ocean languages and all. And there's another aspect that which I mentioned earlier when I mentioned about this Oluwa. We need to look at the Polynesian line, that is Indonesia, or I mean, say when my friend I met in Hanoi, visiting a conference from this, he was from Bali, from Indonesia. Then he said Ulu is for head. Now we say Ulu for the roof tiles, but it's Uluwa. That's again Uluwa. So what is this say? What is it? Is interesting to look at these not only this Eastern India, but the other the Sumatra Polynesian line of language. There's a stock of words also. Maybe some of these what you call the Nishpanna words may be from that area. So that's one of my hypotheses. Whether I've not done any serious work on this, but I'm trying to look at I can remember about many years ago, when I was an undergraduate, I thought of writing a dissertation on the Nishpanna words. That was my supervisor, Professor JB, told me, Sandhukam, you can do a study on Nishpanna words, but less work has been done on Nishpanna words, that is about this locally. So it's interesting. So thank you very much. If you have any questions, I'm happy to answer. But thank you for bringing this linguistic perspective for this conference. Thank you very much.
Thank you very much for the excellent presentation on looking for the very important aspect of our association of single language, but is not well defined, and probably looking for the roots and then perhaps on the association. So I think this presentation is open for discussion. Any questions?
No, even I have not done any study about this the Divyadana, but there are many similar sources are there. But apart from going before Mahavansa period, so there are many sources, but also even the Mahavansa provides many other aspects are there. So there's need to look at the historical, the authenticity of these records of life. So only from as a linguist, my argument is that all these are, but there is some literary sources are available. So I mentioned about the literary sources like not only Sinhala but even the Sanskrit, and also especially like sources in the other languages like Odia, and especially Odia and Tamil sources. So, but that are mainly on Sanskrit sources that we look at, Pali and Sanskrit. But yeah, obviously you are completely right, because say these we need to support, kind of provide a kind of input from those records as well, historical records. But I don't know whether any study that the work conducted to look at, to combine both historical and literary sources. The major resource is the Mahavansa, but there are other sources as well. But not only local sources, but there are many other South Asian, especially the Indian sources as well. It's a very interesting area to bring from history, historical records of both India and Sri Lanka, about the ancient India and Sri Lanka, need to bring to this debate of origin, the evolution of language, and the migrations, that is, but all these, but Divyadana and all the Mahavansas, it's all about the migrations, it's all about the migrations. It's about how these migrations work at the beginning of languages, and how this mixture of both language speakers of different languages. Yeah, yeah.
What is there? So I was reading about it, there is some paper published an article under my name, my dictation, something which was not written by me.
Right.
So he saw this and he emailed me.
What was the article?
The article was, I think, that we all like what is family kind of.
Right.
So they had
genetically one family.
Yeah, some of his blogs they had copied.
Right. Yeah.
That was the standard of journalism. So this person, he saw it, he emailed me, and then he said he had been in Bengal something or for some time. And then he was visiting friends' houses and so on. In one house he had seen this calendar, which are like a kind of thing under space. On it. And then they have related that this is the
Vijay story, yes.
So it is something embedded in that culture, I think. That they say that he came from there, so I do not know. So that is something that we don't.
It's interesting, yeah. So these like even like in look at the kind of legends in Orissa, right? I had a chat, but I was just there for 1 week at that time. Just try to speak to had a chat with some very learned people there. It's very similar stories, similar legends, like about this, not only about this Vijaya, but about these colonizations and the migrations that even this early, they record this in the Buddhist literature about this Tapasu and Balluka. So Tapasu and Balluka, they were came from Orissa, Odia, from Kalinga. Tapasu Balluka, so they were two merchants. That is that the Buddhist record says, and they were like I don't know, but I can remember the period. I would say the story of Tapasu Balluka is there in Kali. Time of the Buddha, yeah. So just Buddha, yeah. So after enlightenment, so what I don't know whether this records it says whether it's local records, but they were from Orissa, from eastern part of India. But as a linguist, I would look at any recent work that I know you are working on these peoples and the migrations and the languages. So any recent input to support this Eastern hypothesis, but you have now we discuss about the Eastern and the Western from a genetic point of view. Is there any support that we can provide to this, whether it is for Western or the Eastern? What is this? Because from linguistic point of view, both schools are strong. Now it's because it's Eastern and the Western. Because it's the Western hypothesis about the origin, it's about because of Geiger and the dictionary found editors. And the Eastern is also supported by many scholars. And there was another argument, this what you call the Hela, or the original language, or Hela. It's like this Nishpan, as the 30th century grammar says, Heladiva Palanipannam was like the indigenous words. The Kumaratunga says Deshi, or the indigenous. So what are these? What is the stock? What is the linguistic character of this stock of words? Right? When you say Anguna Kola Palace, the names of all the tenses, no relevance to any Indian language. So where this come from? This stock of words. That is what we call the pre-Vijaya vocabulary. Pre-Vijaya vocabulary. Even our place names. Except for these recent place names, but all the place names in Sri Lanka. Right. Any other views on that? Right. Okay. Thank you. Right.
We can think of doing much more contribution on that we find in the roots of the language, the support from the genetic perspective. We are in the symposium, Tackling and Evolution of Languages. Our second speaker is Professor Daineswar Shobi. So let me introduce Professor Shobi. Professor Shobi is a professor in zoology at the Department of Zoology, Nalanda State University, India. He earned a PhD from University of Tartu, Estonia in 2010, and he has previously earned a master's degree in science from VBS Purvachal University, Jaunpur, India. Shobi specializes in biological anthropology, including population genetics, human migration and adaptation, DNA studies, medical genetics, forensics, and COVID-19 related issues. He has the Estonian Biocenter at the University of Tartu, Estonia, as a senior scientist. He was also visiting scientist at the Sangha Center, United Kingdom. After finishing his doctoral study, he led collaborative projects involving South and Southeast Asian genetics. He is also leading projects funded by the National Geographic Society USA and GenPo USA. He has published over 20 research papers as first author, 9 papers of second author, and co-authored over 75 ISI papers and 4 book chapters on human population genetics. His work has been published in many scholarly journals such as Nature, American Journal of Human Genetics, European Journal of Human Genetics, PLOS ONE, and Scientific People. He also served as a featured associate editor at the 10th anniversary Journal of the PLOS ONE. During his career, he has received many awards, including Most Productive Researcher Award in 2020 in 40 to 55 years age category in the Institute of Science PHU National Field Award. And in 2021, Rotary Club, Varanasi, India, and also felicitated by Honorable Home Minister, Government of India for outstanding work in the Indian PhD in 2019. Currently, his research involves studying various kinds of uniparental and biparental genetic markers and tracing the role of migration, drift, and selection in East or South Asia by using available computational tools and developing new ones. Over to you, Professor.
Thank you very much, sir. Thanks for your kind introduction. I'm very happy to be here, and I'm grateful to Dr. Rana Singhe and her team for inviting me for this talk. So the previous talk was very interesting, and that is really a hard task actually for geneticists even to differentiate the Orissa and West Bengal, because these two regions are very close with each other, even in geographically as well as genetically. So let's see the how future goes with that work. But today I'll be presenting about the recent migration which has happened to South Asia in last 2000 years. And these migration are Jewish migration, then Parsis migration, as well as the migration which has went out of India, for example, the Roma, the European Gypsy population. So how these migration is being shown by the geneticist or genetic studies, that how these migration has the imprints of genes, which clearly shows that these migration has happened recently. So the first story is all about the Siddhis. The Siddhis, they have been brought to India by the slave trade from Africa by the Portuguese people around 15th to 19th century. So if we look to the principal component analysis of the Siddhis, this is the Indian cline, this is the African, and the Siddhis are in between Indian and African, but they are more closer to African comparing to the Indian population. And here if we see all the three profiles, for example, like autosomal, the mitochondrial DNA, and Y chromosomal profile. In autosomal, the Siddhis has something like 65 to 70% of the African ancestry. The remaining is Indian ancestry. Similarly, for the Y chromosome, they show 70% of the African ancestry, 30% Indian. But for mitochondrial DNA, they show more South Asian ancestry, which suggests that there is a sex-specific admixture of these population to South Asia. And their migration has happened. Probably they were the Bantu speakers. They have been brought by the Portuguese speaker to India. And second work was related with the Roma people who have gone from India something like 1200 to 1500 years back. And these Roma population has been shown by some of the studies that they are more related with the people of the northwestern part of India. So we have done study related with the Y chromosome, because one of the Y haplogroup, that is Y haplogroup H, which was also present among the Roma population, and because H is very specific to South Asia, only present into Indian, Pakistani, Sri Lankan, Bangladeshi, and Nepali population. So it means that H is indigenous to South Asia, and any lineage out of the India, out of the South Asia to other part of the world usually suggests that this is migrated from South Asia. So in this case, we have found out that the northwestern region indeed showed that the population of Roma showed a very closer haplotype similarity, whereas some of the population which are related with the scheduled caste and scheduled tribe population, Roma showed the closest affinity. So this study has showed that Roma is very closely related with the northwestern part of India, as well as the scheduled caste and scheduled tribe population of India, more with the tribal populations. So here we have completely mapped the Y chromosomal H haplogroup profile. We showed that it has originated somewhere in South India around 25,000 years back, segregated to different parts of India and other parts of South Asia. And further, there was one branch which went out of India something like 1,400 years back. And the autosomal analysis for Roma population showed that there are two populations, Dharkar and Kanjar. These are also the gypsies of India. They showed the most closest similarity with them. Our third story was with the Jewish population of India. The Jewish population, they're supposed to be migrated from Middle East something like 2,000 years ago, but there are two groups. One is present into the Maharashtra and other into the South India, into the Kerala, Cochin. So these two groups has migrated at two different time periods. Molecular data also suggests the same. So the historical data and molecular data, both of the data, usually they are in very much coherence with these kind of migration. So in this principal component analysis, the Mumbai Jews, they are more closer to their neighboring population. Cochin Jews make two different clusters. One is related with the Indian clinical groups and more with the Southern groups, whereas the other European Jews, Middle Eastern Jews, Caucasian Jews, they are more related with the European and Caucasus populations. And their admixture analysis shows that they are overwhelmingly related with the South Asian population. However, there was some traces of Middle Eastern, like this dark blue like ancestry, and this ancestry connects them with the Jewish population, as we can clearly see that this ancestry is only present into the Jewish population, not into their neighboring populations. And if we look to this Middle Eastern specific distribution of the ancestry component, it is more into Middle Eastern, whereas there are few groups in India, for example, Mumbai Jews and Cochin Jews, they have a higher amount of this ancestry. This shows that this ancestry did not come just by the allele distributions. It is due to a migratory pattern which has migrated from somewhere here to here. So the time of admixture was also very much coherence, and this time of admixture shows that around 37 generations ago, the Mumbai Jews has migrated, and around 53 generations ago, the Cochin Jews has migrated to India. And here, the Jews and the Indian cluster, both of the cluster is being present into this Jewish population. And also we have found out that the mitochondrial DNA and Y chromosomal ancestries, mitochondrial DNA is more related with the local South Asian population, whereas the Y chromosome is connecting them together because of the haplogroup J2, connecting them together with the Middle Eastern populations. And our fourth story was related with the Parsi population. As we all know that Parsi live mostly into Mumbai, the central part of India, the western part of India, also some in Kolkata, some present into the Pakistan. And I don't know whether they are present into the Sri Lanka, but mostly they are into the India and Pakistan. So these population, they say that they have migrated something like 1200 years back to South Asia. And these migration is also present into their molecular data. And you will be surprised to know that the real Zoroastrian ancestry is now being retained by the Parsis, whereas their original homeland, which is in Iran, is usually admixed with the other population. So this 7th century, because of the Islamic conquest, the population of few people, actually there were 7 groups, they have moved to South Asia, and there was a local king. This says that they have gone to the local king for the shelter. Then local king has sent a glass of milk, and they have put sugar into this milk, telling that we will be culturally mixed with you people. We will not make any problem. Just we need the shelter. And this is how the Parsis, they have included to South Asian population, and they are actually one who has made the modern India. So the principal component analysis, where we compare various population, here we see that the Parsis, they are in between. Iranian population are here, and this is the Pakistani population, this is the Indian population. So so the Parsis are in between the South Asian groups as well as Iranian groups. It means that they share both of the ancestries which are present in India and Iran. This is again testified with the migration, which shows that they have a huge amount of Iranian ancestry. This is the local South Asian ancestry, whereas among the South Asian population, this ancestry was absent. So we estimated the time period, and this time shows that around 50%, 56 to 70% of the ancestry of Parsi is being driven by the Iranian groups, whereas the local South Indian group is usually given them around 30 to 50% of their ancestry. So interestingly, we found out that when we compared with the Iranian Neolithic population, the population which has lived around 10,000 years back in Iran, and the population which is living now, the Parsi population, they are more related with Iranian Neolithic population comparing to the modern Iranian population. This suggests that the modern Iranian population has gone a very severe admixture with the other population. And here this analysis shows that how much genetic ancestry is being received by these donors. And here these are the donors, these are the recipient. And here we see that Gujaratis has given a high level of ancestry to the Parsis, whereas Parsi has not contributed to the Gujaratis. So this is like, this also shows that there is a symmetrical with the Parsis. And runs of homozygosity shows, because this population is small, so runs of homozygosity shows that this population is highly inbred comparing to the other Indian population. And the generation time is something like 20 to 38 generation, which perfectly matches with their migration from Iran to India. And here we see that their paternal ancestry is more related with the Iranian population, whereas their maternal ancestry is more related with the Indian population. And here clearly see that the ancestry pattern of the maternal and very distinct from each other. An ancient sample of all the three groups, Mumbai Jews, Kerala Jews, and Parsis, they are very clear, and they show that the South Asian ancestry, Iranian ancestry, and then the steppe ancestry is present among all the three populations. So here we conclude that Siddhi, they are most recent African migration, which have arrived from Africa as a slave trade. The scheduled caste and scheduled tribe population of northwestern India are the ancestors of European Roma, and the Middle Eastern related ancestry is present among the Indian Jews, whereas it is absent into the local population, whereas it is more in the Indian Jews, it is less. So it means the Indian Jews have admixed very highly with the local population, whereas Parsi retained their ancestry. So the Cochin Jews has migrated in the 5th century, whereas Parsis has migrated around 1200 years, 7th century, around 1200 years back. So what we have suggested that because of the initial religious conversion, the population of Jews has increased, but then later they started following the endogamy, and this is how the Jewish population got admixed with the local population. And in case of Parsis, that mixture with the local South Asian population was comparatively minor, and the migration was male mediated. There were more male population comparing to the female population in both of the cases. So I would like to acknowledge people from Estonian Biocenter, University of Kolkata, from Deccan College, the Wellcome Trust people, CSIR, CCMB Hyderabad, Sanger Institute people, and Professor George Wandering, who is the only linguist in our group and who has contributed a lot in such studies. So thank you very much for your kind attention. I'll be happy to take your questions.
Thank you very much, Professor Sobi, for very interesting presentation. Now the forum is open to questions.
Professor Chobe, thank you very much for that very interesting presentation. My question is not related to your today's presentation, but some of the work that you did with tribes which are mentioned in Ramayana. Could you please elaborate a bit on that?
I could not hear. I'm sorry. Could you just speak a bit louder?
I'll come to the podium. Professor Chobe, thank you very much for that very interesting presentation. My question is not related to your today's presentation, but a paper that you published on some of the tribes mentioned in Ramayana and those tribes which are living in India yet at the moment. Could you please elaborate a little bit on that, on your findings to this audience?
Yeah, there were 3 tribes, Kol, Bhil, and Gond. These 3 tribes, they have been mentioned several times in Ramayana. So what we have done, we wanted to know whether these tribes are related with the local population or they have some very unique kind of ancestry pattern. So we have compared their ancestry with their neighboring population, and we found out that they are the population who actually make the basal root, the basal foundation of all South Asian population, and each and every population of South Asia, whether it lives in Sri Lanka, whether it lives into Nepal, whether it lives into even up to the Myanmar, all the population, they have some amount of ancestries which has been found in these tribal populations.
Thank you.
Any other questions, please? In the absence of questions, we will thank Professor Chobe in our usual manner. Okay, we are coming to the last lecture of symposium 4. The lecture is delivered by Professor Kamini Tennakoon. Let me introduce Professor Kamini Tennakoon to the audience. Professor Kamini Tennakoon is well known to the scientific communication of Sri Lanka for her dedicated contribution to various branches of science and especially molecular life sciences. Professor Tennakoon obtained an MBBS with second class upper division in 1979 from the Faculty of Medicine, University of Colombo, and her PhD at the University of Sheffield, United Kingdom, under the Commonwealth Scholarship Scheme in 1982. She served as the director of the Institute of Biochemistry, Molecular Biology, Biotechnology from 2007 to 2012. Until her retirement, she served as the Senior Professor of Molecular Life Sciences at the Institute of Biochemistry. She began Her research carrying reproductive physiology and extended her research focus on molecular genetics of cancer, mitochondrial DNA and ancient DNA, population genetics, natural product development, and identification of biomarkers for hepatocellular carcinoma. She has supervised many masters and doctoral students and authored nearly 80 publications published in international peer-reviewed journals. Her excellence in research was duly recognized, and she has received many awards, including the Presidential Research Award in years 2001, 2005, 2010, 12, 15, and 16, Merit Award for Scientific Excellence in 2011, 13, 14, Suspect Award in 2016 by the National Science Foundation of Sri Lanka, and Senate Award for Research Excellence in 2016, 17, 18, and 19. She has also received several research grants from national and international funding agencies. Professor Tennokon is a member of many international national organizations, scientific societies, and served as an evaluator, assessor for many journals, institutes, local funding agencies, and professional organizations. So let me introduce the Professor Tennokon, and I would like to welcome Madam for your presentation.
Thank you, Prasanna, for that rather lengthy introduction. If I had an idea, any idea that you were going to read all of that, I would have stopped it. But anyway, so I feel a bit of a draw because there's eminent archaeologists, eminent linguists, and an eminent geneticist online. The other two are on site, and I'm like jack of all trades, but master of none. Like when it comes to this presentation, so I title it as "History Meets Genetics: A Glimpse into Genetics of Sri Lankans." And just to put everyone in the same level, just that we can use both uniparental and biparental genetic markers. Kanchan, how do I get the pointer here? Without an external mouse, I'm lost.
Oh.
Do you have a point? Or just show me how to find it. That's a movement point.
Oh, okay.
If you can find something else. Never mind, I'll try to do it without a point. Okay, that's okay. So the uniparental markers are the mitochondrial DNA, which is exclusively maternally inherited, and the Y chromosome, which is passes from the father to the son. Mitochondrial DNA passes from the mother to both sons and daughters, but only the daughters will pass it to the next generation. Then in our nuclear genome, we will have the nuclear genome will be passed down from the rest of the chromosomes will be passed down from both parents. And there we can identify what are referred to as single nucleotide polymorphisms, where there, as you can see that two sets of letters written at the bottom, where the first set has CCGT, other one CAGT. So that's a change. A single nucleotide has changed, and we have millions of these. These can also be used to identify people. And the autosomal STRs are short repeats. You can see that the first one shows CAG, the bottom one shows CACA. So these are the repeats, CAG. So one person may have 5 repeats, another person may have 6 repeats. Like that, these also can be used to identify people. Since I'm going to talk about mitochondrial work, I'll just give you a little introduction to the mitochondrial genome. Mitochondria, we have hundreds of mitochondria, hundreds of thousands of mitochondria in our cells, and it has its own genome of circular double-strand DNA comprising of 16,569 base pairs in the human. And it has an area which is referred to as the control region, which acquire most of the mutations, and the mitochondrial DNA can acquire mutations and they last because unlike the nuclear DNA, the repair mechanism is rather deficient. And the rest of the area of the mitochondrial DNA is referred to as the coding region. So since the control region acquires most of the mutations. This is used to identify variations between people, but the coding region mutations are also important. And this diagram shows you how the mitochondrial DNA is inherited exclusively down the maternal line. You can see the black lady is transmitting it down to the bottom line. The other ladies have stopped having daughters, so their mitochondria lineages are not transmitted down. So what we see in our contemporary populations today is what has been passed down, and what we have lost is not seen in our contemporary populations. Because I'll be using these terms saplotypes and haplogroups, just to tell you that what a haplotype is, that is, it's a set of DNA variations or polymorphisms that tend to be inherited together. We know that DNA is made up of the nucleotides, and at the bottom in the diagram, you can see a string of letters, and those two are different, and so those will be two haplotypes. A haplogroup, on the other hand, is a group of haplotypes which will share a common ancestor with a common mutation in all the individuals. And mitochondrial DNA is divided into a number of haplogroups. The L haplogroup is considered the original haplogroup, from which several others have arisen, going from L0 to L6, and of which L3 is considered to have migrated out of Africa. Africa is the continent where the Homo sapiens are thought to have evolved. And as they migrated out, this acquired further mutations, and one group was identified as macrohaplogroup M and the other as macrohaplogroup N. And then M is also having further subgroups, and I'm only showing the R and the U haplogroups here, and which comes under N because we will be looking at those. So-called mitochondrial Eve, which is not a single person, but obviously a group of people, they were thought to have lived in Africa about 130,000 to 200,000 years ago. But new evidence from Egyptian fossils tend to put these, according to some scholars, this time of mitochondrial Eve's existence to back even 300,000 to 500,000 years before present. So this L1 to L6 has arisen in the African continent, and the L3 is the one which migrated out. You can see the L3 in the middle square, and that has migrated out, giving rise to M and N, and it's considered to have happened about 55,000 to 90,000 years ago. Again, the new evidence adds another 100,000, approximately another 100,000 years to this. As they migrated out, the mitochondria DNA keeps on acquiring mutations. So people who acquire certain mutations would be grouped into a particular haplogroup. So we have, this has led to the notion of continental specific mitochondrial haplogroups. If you look at the bottom diagram, you can see that Africa, Asia, and Europe are shown. And in the European oval, you can see the H haplogroup. So if we find the H haplogroup in Sri Lanka, we will say that that person is having a European ancestry. Similarly, in the Asian one, you will see G, and then we will say that if you find it in Europe, you will say that the person is having an Asian ancestry. And these people who left the African continent, there are two migratory routes that people believe that they have taken. One through the Fertile Crescent, the Levant, the northern route, which is shown in the red arrows, and then they populated the rest of the world by having various stopovers here and there. And then the other one is what is referred to as the Beachcomber route, which is shown in the dashed line at the bottom, which has been indicated by the blue arrow there. And there's quite a number of evidence from, including work from Professor Thangaraj that confirms the southern Beachcomber route. And whether this was the single dispersal or several dispersals is not sure, but it's about 60,000 years ago this route also is considered to have been taken by people. And if people moved out of Africa and went along the coastal route to populate the Indian subcontinent, then it's obvious that Sri Lanka also could have been a favorite destination. Just to put the picture make the picture complete, we know that we have a diverse population in Sri Lanka and the majority are Sinhalese. And this diagram shows some pie charts in different parts of the country, and you can see Sinhalese are mostly in the southern parts of the country. The blue color shows the Sinhalese, and the north, and in the north is predominantly Sri Lankan Tamils, and in the east you can see that more or less equal proportions of Sinhalese, Sri Lankan Tamils, and the Moors, who are shown in green color. So orange color shows the Sri Lankan Tamils, and they are also found in the western province as well as in the upcountry. The yellow color shows the Indian Tamils, who are recent migrants who were brought to Sri Lanka by the British. We have some evidence and history of human settlements. If you look at if you go from the bottom of this slide, we have archaeological artifacts which tells us Sri Lanka has been inhabited by humans approximately from about 125,000 years before present, but there's no skeletal evidence for that. Skeletal remains of Balangoda man that Professor Raj Somdeva mentioned in his keynote speak is about 38,000 years before present, dated to that time period. And tools associated with these groups are now dated to about 45,000 years before present. Then we also have some data from botanical side that there was evidence of cultivation of barley and oat in the Horton place, which goes back to about 17,500 years before present. Then we have this legend of Ravana of the Ramayana fame, and it's a legend. We do not have any archaeological evidence or historical records as such, but there are many places in the country named after the characters of associated with the Ravana legend. And as Professor Chobe just clarified, that some of the people who lived in those times, that seems to be forming the base of they are mixed with all South Asian populations. In that column there's a little green part which I have expanded. That is our historical times where we have the recorded, our history is recorded. And we heard about the migration from North India about Prince Vijaya in Professor Sandagomi's talk. And that's about 5th or 4th or 6th centuries BC. Then the same Chronicle Mahavansa records about South Indian Dravidians coming to the country from 2nd century BC. And we have the Muslims as traders coming to Sri Lanka from 7th to 8th centuries AD. And Malays were brought to Sri Lanka when we were occupied by, part of Sri Lanka was occupied by the Dutch. And that's from about 17th to 18th centuries AD. Of course, there's also a story about, it's recorded that there were movement of people from Malay area that, and they integrated with the original inhabitants of the country, and they cannot be identified as a separate group. Whereas these people who came in the 17th to 18th century, we can identify them as a distinct group. Then, of course, the Indian Tamils were brought in the 19th century, mostly from Tamil Nadu, by the British to work in the estate and do other hard work, because the natives in the country were not cooperating with them and they were not willing to do that hard work. So I will not go through this because we learned a lot about the arrival of Prince Vijay, and this is what I was asking, that the fact that Sri Lanka was called Sinhala Dweepa and people were Sinhalese long before Vijay arrived is a school of thought which is being put by some people. And one of the books, the people who proposed that, they cite several references, and I had read about it, but only recently I read the Vyavadan, so this is the first time I'm putting that in my slides, because until I read the original source, I didn't want to put it. So that's another story. Of course, we had a major cultural influence during the Emperor Asoka's time, and Buddhism was brought by Arhat Mahinda Thero, and Mauryan Empire extended almost all over India, except the little bit which is not shown in the light blue color. And few months later, Arhat Mahinda's sister, Arhat Mahinda is son of Emperor Asoka, and the daughter of Emperor Asoka, Arhat Terni Sangamita, brought the sacred bow sapling, and with her came many people from many clans, nobility to craftsmen. Of course, from the time of Vijaya, there were very often these kings, people who came, they probably didn't think much of local women. They brought women, princesses from there for their coronation and so on, and also for their councils and so on. The women were brought from India, from different parts. There's not a single area. So some of the nuns who came with Arhat Terni Sangamita, they have been derobed and given in marriage to those nobility who came. So again, they wanted to propagate the mitochondrial lineages, I guess. Then, of course, we have this 4th century BC, it's not BC, I think it's, I can't see that. Anyway, this was also mentioned by Professor Sandagomi, and we know that sacred tooth relic was brought to Sri Lanka by Princess Hemamala and Prince Danta, and they came from Kalinga. Of course, this map of Kalinga, which also I took, it shows only a small area, whereas I think you had a bigger area in eastern India as Kalinga. Of course, then we also see that there was Buddhism in Sri Lanka before the advent of Arhat Mahinda. There's the story of 3 visits of Lord Buddha to Sri Lanka in his lifetime. And then Arhat Sarabu, a pupil of Agrashravaka Arhat Sariputta Thero, he took the cervical vertebrae, that is the Griva Dhatu, from the funeral pyre of Lord Buddha. The story goes like that, and then brought it to Mahayangana. That's said to be the first place visited by Lord Buddha. Then, of course, those are the stories, but then there's a stone inscription in Duguvegala Tamankaduwa, which says Sangarakita Thero, a pupil of Agrashravaka Arhat Sariputta Thero, is said to have been teaching Naga children. So who are the Sinhalese? Well, all our chronicles talk about 4 tribes who were natives in the country: Yakka, Naga, Deva, and Raksha. How did they get here? Did they come along the southern beachcomber route? Did they come through the northern route and then through mainland India? Or did they take a southeastern route, as I will show you later on? It's not a very much accepted one, but there is also a possibility of a southeastern route. It's very likely that the contemporary Sinhalese are an admixture of those who lived in the country at the time. If Vijaya or a group of people came from North India, those migrants, as well as subsequent migrants from both North and South India. Then who are the Sri Lankan Tamils? Then again, it's very likely that they are an admixture of people who were natives in the country and migrants from southern India. And there are the records of numerous Indian invasions. They came from different localities of India, southern India, Chola, Chera, and Pandya, the areas that you can identify with in the map at the bottom. Not only that, IPKF, we think of only IPKF, but local ruling family members, when they wanted to oust the ruling king, they got help from India from time to time, and they brought armies from there. So what is happening in the contemporary situation is nothing new, it's only history repeating. Then, of course, we have the Adivasis, and they are, I'm sorry, I can't get this line out, and it's thought that there are various schools of thoughts as to what the origin is. It's thought that they are the descendants of prehistoric humans in Sri Lanka. Okay, thank you. That is the Balangola man and whoever lived before that. Some people think that they are descendants of Vijay and Kuweni's children. That was a story that we learned when we were children. if they were descendants of Kuenese children, then we would expect them to have, all of them to have Kuenese mitochondrial DNA. We do not know what she had, but it has to be probably one particular type of mitochondrial DNA. Of course, the people themselves have, I read in some book that they said that Kuven is one of the Lokuamas and what not the Lokuama. So that is not, there would have been many others. Then of course there's the story of the thought that they are descendants of Proto-Australoid and Proto-Negroid. These are the people who went on to become Aborigines of Australia as well as inhabited islands like Andamans and so on, and migrants from tribal groups from India. Since I mentioned about Muslims, the Malays and the Indian Tamils, I will not elaborate on them, and I will just go through some of the early studies before getting on to our own studies. So this diagram, which is from a publication made using gene frequency data for 18 protein and blood group loci from published data by Roy Chowdhury et al. In 1985, shows that the Sinhalese cluster with other Indian populations, Bangladeshis and so on, and they are closer to the Afghans and Iranians rather than to the so-called Mongoloid populations, the Nepalese, Bhutanese, Malay, Chinese, and Batak. So these are protein and blood group loci data. Then, of course, there's this paper from Mastana et al. In 2007, where they describe evidence from allopolymorphisms. This is again looking at some markers in autosomal DNA. And they studied 121 Sinhalese from Colombo. We do not have any information about how they, they would have reported that they are Sinhalese, whether there were mixed marriages, mixed ethnicity marriages in their families, whether there was any contribution, we have no idea. And this analysis had shown that a greater contribution from Bengali when 3 parental groups were used. Bengali contribution to Sinhalese remains strong, 50 to 66%, followed by Northwestern, and rest contributed by Tamil population. Now let me take you through some of our work, and where we looked at mitochondrial DNA in contemporary Sri Lankans. We looked at maternally unrelated Sri Lankans. We asked them to recall whether there are mixed ethnicity marriages as far as they can recall, the minimum recall being at least for 3 generations. And in our first study, we had Sinhalese, Sri Lankan Tamils, Vedda, Moor, Malay, and Indian Tamils. That is the work that Rwanda did. And then we have also data from 2 other studies which were carried out for other purposes, but we used them also to look at our population distribution. And that one had 121 Sinhalese and 21 Sri Lankan Tamils, and the other one had 60 Sinhalese. The first 2 actually we looked at the non-coding region or control region of the mitochondrial DNA, and we used Sanger sequencing. And the last one where we looked at 60 Sinhalese, that is where we did next generation sequencing, so that it also included mutations in the coding regions. When we look at the 2 main macrohaplogroups, M and N, that is the 2 to which L3 got divided, all haplotypes belong to out of Africa M and N macrohaplogroups derived from L3. So we all came from Africa. And Rwanda's work demonstrated that if you look at M and N haplogroup distribution in these different populations, Sinhalese, Sri Lankan Tamils, and that's the 2 first and the second columns, and the 3rd column and the 4th columns are Muslims and the Malays. They tend to have similar proportions. M is little more than the N, but they tend to have similar proportions. Vedda, which is the 3rd column, you can see that they have a lower proportion of M haplogroups and a higher proportion of N haplogroups. And the Indian Tamils, who are the more recent migrants from southern India, they are having predominantly M haplogroups and very little that belong to the N macro haplogroup. This is data from that Amali has compiled from 675 Sri Lankans. It's still unpublished and paper is under review. And I looked at the M and N distribution again in these three categories. Muslims and the Malays, there were only very few additional samples, but there were more samples for Sinhalese, Sri Lankan Tamils rather than the Indian Tamils. And the same pattern continues. The Vedda are having a lesser number of M haplogroups and more of N haplogroups. Rwandi looked at the phylogeographical distribution of haplogroups in her study, and the blue color here shows South Asian or Indian mitochondrial haplogroups. They are common in India and South Asia. And you can see Sinhalese, Sri Lankan Tamils and Vedda are having something between 63 to 66% of South Asian mitochondrial haplogroups. Malays and Muslims are having about 90%, and the Indian Tamils are having about 95%. What is interesting is that there is a considerable presence of West Eurasian haplogroups among the Sinhalese, Sri Lankan Tamils and Vedda, people who have been living in this country for a longer duration of time. And we also have a Southeast Asian contribution, and that is seen more among in this group among the Sri Lankan Tamils and little bit among the Sinhalese, and not among the Malays, Muslims, and the Indian Tamils studied in this cohort. Then we have an East Asian contribution, which is shown in green color, and that is seen mostly among the highest prevalence is among the Veddas, and then some prevalence among the Sinhalese and the Sri Lankan Tamils, the Muslims, Indian Tamils in Sri Lanka, and so on. And a South Pacific contribution, which is seen among the Malays and the Sinhalese. And this is again from Rwandi's data, where she looked at how closely these groups are related. Veddas tend to cluster further away from the other groups. Sri Lankan Tamils are much more closer to Sinhalese than to the Indian Tamils. And the Malays and Muslims are also, they are closer to each other and closer to Sinhalese and the Sri Lankan Tamils. Now, when we say the M haplogroup, within them there are many M macro haplogroup. Within them there are many other haplogroups. In this data, I took all the data from the three studies that I described, and we looked at the prevalence of different haplogroups, common haplogroups. There are many M haplogroups going to some 70 odd. And you can see that although they belong to M, their distribution, when you look at a lower level of higher level of resolution, then they are different. You can see the Indian Tamils having a very high prevalence of the M2 haplogroup, which has a found age of about 52,000 to 64,000 years. And we know that these are the people who were brought to Sri Lanka in the 19th century. So just because they have an old haplogroup, that doesn't mean that they have been in a particular geographical location for that long, but it's just that they are carrying without any mutations that particular haplogroup which was derived at that time. The Sinhalese have M65, which is the grayish column, which is the highest prevalence among the Sinhalese. I will come to that M65 in a minute. And this is just to illustrate that distribution of these different haplogroups within them is not the same between these different ethnic groups. So we can say we are the same, we can also say we are not the same. And these red dots indicate the places where this M65 has been, M65A has been described, which we find we find that to be the commonest haplogroup among the Sinhalese. So it's found in India, Pakistan, China, Tajikistan, Ladakh, Myanmar, China, and so on. You see a red dot in Syria, and that is actually from an ancient, not very ancient, but old skeleton, and it's thought that it's not found in the population there. They found it in a skeleton, and this led to the belief that this was actually from Indian merchants who, Indian merchant who was buried in that locality. So when we find skeletal evidence also, we have to be very careful as to how we interpret, because it could be from a visitor. Then when we looked at the distribution of R haplogroups, again you can see the similar phenomena where between groups there are differences. Among Indian Tamils we did not see any. Vedas, interestingly, had two main haplogroups, two main R haplogroups, R5 and R30. And this is the prevalence of U haplogroups. Some of the U haplogroups are of Indian origin, and there are others which are of West Eurasian origin. And Indian U subtraits are more prevalent among Sri Lankan Tamils, Moors, and Malays. Vedas, on the other hand, the West Eurasian U7 haplogroup, which is shown in red color, this was highest, U7A was highest among Vedas. It was second highest among the Sinhalese, and it was absent in Sri Lankan Tamils. Now, West Eurasian haplogroups, it's said that in Indian studies that Indian caste populations have about 17% prevalence versus 7% prevalence in Indian tribal populations. So how on earth that something which is found in Indian caste populations got into our Veda population? And this is again about U7A, and the map shows the areas where U7 is predominant. It is of West Eurasian origin, and it was shared with Sinhalese, Moor, and Malay, and was found predominantly among Veda people. And the R5 subtrade, which we found among Veda, and it is also is thought to have arrived from Near East and Middle East to India, and that is also more frequent among caste populations. And this was found among Sri Lankan Tamils as well as Moors. And R30B2, which was found among Vedas, that was another very interesting haplogroup. This is found in the South Asian coastal region, but low frequency. A skeleton from Rupkund Lake in Himalayas also carries this particular haplogroup. This is quite common haplogroup among our Veda people. That was not found among Sinhalese, but was found among Sri Lankan Tamils and the Moors. Then I want to share with you some other interesting findings that we had. We found certain haplogroups. So you can see that these haplogroups, they don't, there are further subclades. And these three haplogroups, U2E1, A1, we found just one Sinhalese person to carry that. This has been described in an ancient mitochondrial genome from Ukraine that belonged to the late Neolithic culture, the Copper Age, 3350 to 3200 BC. U4C1 was found in two Sinhalese, and it's reported from Central Asia and has been found in steppe ancestry skeletal remains, early Bronze Age, late Copper Age to early Bronze Age, 3100 to 2800 years before present. H6A1A was also found in two Sinhalese, and this is derived from H6A. It is assigned to an ancient mitochondrial genome from Poland associated with coded ware culture, that is 2400 to 2300 BC. So it is plausible that these microgenomes from Central Asian origins migrated either through India or through West Eurasia. So when we summarize, we have mostly South Asian or Indian mitochondrial DNA haplogroups among Sri Lankans. That supports historical evidence of female migration from India or through the beachcomb, or a more or less simultaneous colonization of both Sri Lanka and India, either through the beachcomber route or from Southeast Asia. I did not describe Southeast Asian route because that is not one of the most popularly accepted theories, but some people think that the terrain would have been more difficult and people would have moved towards Asia and then to Southeast Asia and then colonized India and the Indian subcontinent, but it is less accepted at the moment. The West Eurasian haplogroups among the Sinhalese, Sri Lankan Tamils and Vedda suggest early migration of women carrying these haplogroups into the country. And the predominance of Indian mitochondrial DNA haplogroups among Malays and Muslims support the notion of their largely male migration and marriage to local women. So these Arabian traders, they didn't bring women with them, they married local women. So our mitochondrial DNA evidence for Sri Lankans show that our maternal ancestors have come from the out of Africa mtDNA lineage. They have either come from India, West Eurasia, Southeast Asia, East Asia, or South Pacific, or we share common ancestors with these populations. And there's a considerable genetic admixture in contemporary ethnic groups in Sri Lanka. And the Adivas is clustered separately from other groups, suggesting a distinct ancestry. Now, if we look at the genetic lineages and historical perspectives, as I said, we have come out of Africa, we have come from West Eurasia, India, East Asia, Southeast Asia, and then populating Sri Lanka. Indian female migrants, our recorded histories have evidence of female migrants from India. Southeast Asian female migrants, whether they came on their own or whether they were forcibly brought, there are stories about wars being fought and princesses being brought from Southeastern countries like Burma and so on. I'm sure Professor Raj Somadeva and Professor Sandabhumi can elaborate on them more than me. I remember reading about some of these. And from East Asia, did we have female migrants? I do not know, but there are stories about Buddhist nuns going to China and so on. Whether Chinese women came to Sri Lanka in the ancient times, I do not know. But we somehow or other we have East Asian lineages. They could also have come to South Asia, East Asia, or India and come to Sri Lanka. West Eurasian female migrants, and I have not come across any direct migration of women from West Eurasia to Sri Lanka. Of course, if you watch that Ravana, the teledrama, they showed the women merchants coming and dealing with Maninaga. So I do not know how true those stories were, but obviously we have a West Eurasian ancestry. Whether they came through India or whether they came directly through the beachcomber route, we do not know. But genetic evidence tells us that we have a West Eurasian ancestry in our population. And South Pacific female migrants, there's again no record to be seen. I have not come across. So that's why I have put question marks. I cannot say no because I don't know. Of course, these could also be if people moved from along the beachcomber route, if they populated India and Sri Lanka and then went on to Southeast Asia and Australia, the South Pacific, then maybe these are the remnants. We do not know. Or maybe these are more recent introduced lineages. So that's how we can at the moment relate the historical evidence to the genetics. But the two presentations which will follow, the two pre-papers, will talk about more about our recent findings. I did not want to include them here and do. I mean, I want to do justice. To the two PhD students, so they will be presenting after the symposium. So I would like if Professor Sandagumi and Professor Raj can stay and listen to those as well. So these are the people to thank. Professor Karunanayaka initiated these studies, and Ruandi was the workhorse for the first study, and the others, we took data from Lakshika and Joanne. They were working on mitochondrial DNA in relation to cancer, and Amal is working on ancient DNA, and Anjana is also working on Vedda and contemporary populations. And we had support from National Research Council, National Science Foundation, Sarek Sweden, and Dr. Tangaraj and Dr. Niraj Rai helped Ruandi in these various analyses, and they continue to collaborate with her, and also now Professor Chobe. And there were many people who assisted with sample collection to make these studies a success. Actually, I wanted Ruandi to make this presentation, but she forced it on me, and I have told her this is the last time that I'm going to be talking about this, and she's the one who should be talking about this work. Okay, thank you.
Okay, thank you very much, Madam. It was an excellent presentation on, perhaps it is eye-opening. So looking at our interest and the migration pattern and how the genetic mix-up is giving the clear indication of what is our origin. So the presentation is open for the discussion. Any questions?
So can I have a question?
Yes.
Wonderful, madam. This was a very great talk, and with the background information you have given to all of us, that was very nice. I wanted to just make a comment that I think adding the Y chromosomal information is going to tell a lot more comparing to the mitochondrial DNA.
We really want to do the Y chromosomal work, but we are looking for funds, so that is why Ruan had been looking for funds for several years now.
We are ready to help if you are planning for that. We can include our funds for this kind of studies.
That will be very great. Ruan, you have to take it from there because without Y it's not complete.
Yeah, maybe you need to send your students here. They would learn few things, and they would also do some of the studies. Yeah.
Sure. Thank you.
And then some of the mitochondrial DNA which you have showed, they are really interesting, and I think some of the complete sequence set would be very much useful to do.
Yeah,
like M65 and then other haplogroups. Even R30s are very interesting to explore.
Yeah. So you will hear a little bit about complete sequences from Amali and Anjana, who will be doing the oral presentations now immediately. And I very much would like you to stay on and give your inputs for that.
Sure. Thank you. Thank you.
Thank you for bringing this history and then this together. So what I'm interested about this, as I mentioned earlier, about this Hispania, work of Hispania. Is there any basic kind of evidence from Southeast Asia? Also, like not recently, but in 1940s, the scholars from the Hela House, like people like scholars like Abu, they pointed out these kind of African, like Madagascar kind of connections to Hela. So, in basic kind of play of Madagascar, and also from this, but I believe it's more from this East Asia, Indonesian side, this fund that I want to do later on. Any kind of genetical evidence to prove that early kind of population
Have you included Madagascar?
I have included 4
Indonesian sequences in your presentation. Yes. Madagascar, no, not Madagascar. We have to do an analysis and see. I think that is Just now, huh? Just now.
Madam, small, please, my university, I'm asking. Now, there's a saying of this, the kings getting these queens from Kalinga.
Not only Kalinga,
Yeah.
Kalinga or someplace, no,
Yeah,
they sent
yeah,
some.
So the problem, and most of the cases, they suffer from the subfertility or infertility that was reported. So how you justify that is what you see in the mitochondrial composition, what we are having, and how we form. Is there any justification of what the history says? Fine.
We can't get into that shit, no. There are infertility and then of course, but there was, so there is a reason one. I'm sure they are very fertile, they have very fertile women in Sri Lanka, but maybe they wanted to always import us. Maybe they are more beautiful, we do not know like this. Now, you get a lot of beauty films from India and so on. I don't know.
But again, I'm looking at the Y chromosome.
Yeah, that is very important for us to do.
Yeah.
Because we need to look at the autosomal as well as Y chromosome. Y chromosome one will tell us how the paternal life.
Exactly. So thank you very much for coming to the phone for the interesting and very elaborative presentation, and then so we will show our appreciation as usual.
I think this is the idea trying to enhance the research on genetics and global population, because people are trying to strangle each other at this time. So we want to show that the common lineage and common origin to increase the community.
Unfortunately, it doesn't work because somebody first said we all are different, therefore you are the piece of the land. Then the same person said we are similar, therefore you are the piece. Whatever you say, it's for other reasons.
Online?
There's only two.
No,
yeah.
Become recorded?
Next, we have the fifth session on the oral presentations. We would like to invite the chairpersons, judges for this session, Dr. Meran Edrivira and Dr. Kanishka Senathilaka.
So welcome to the oral presentation session 5, population genetics and bioinformatics. There will be 2 presentations. The first one will be done by Amali Fernando on maternal genetic affinities of Sri Lankan, prehistoric and modern population with the population in the Eastern world. So you can come to the presentation.
Greetings, everyone. When understanding population genetics, you can either use autosomal genetic markers or uniparental genetic markers. So in uniparental genetic markers, you can study genetic variations in chromosome, or you can understand the maternal inheritance by looking into the variations in the mitochondrial genome. So in our study, we are looking at the variations in mitochondrial genome to understand the maternal genetic affinities of Sri Lankan prehistoric and modern populations with the populations in the Eastern world and Australia. Now, why are we using mitochondria to understand maternal genetics is that mitochondria, which we know as the powerhouse of the cell, is actually an organelle that we inherit from our mother. So its maternal inheritance, high copy number, lack of recombination, as well as high mutation rate make it a perfect genetic marker, a uniparental genetic marker for us to understand an individual's matrilineal kinship. So how we're going to do this is when we have the aligned mitochondrial sequences, we look into single base changes or single nucleotide polymorphisms. So when a set of or group of these SNPs are inherited together, we can call these haplotypes, and we can understand the haplotype distribution in a population using these group of SNPs, right? So when these haplotypes are very stable, they can be further categorized into haplogroup. Now, since we are focusing on the mitochondrial haplogroup, this is an illustration of the mitochondrial haplogroup diversity in the Sri Lankan population. As you can see here, the Sinhalese and Sri Lankan Tamils have a similar profile, but the Sri Lankan Tamils and Indian Tamils, they don't share the same profile. And if you focus on the Adivasi population, their maternal profile is quite different to the rest of the Sri Lankan populations. So why do we focus on these mitochondrial haplogroups? That is because mitochondrial haplogroups are known to be geographically restricted, meaning when studying mitochondrial haplogroups, we also get to study the population genetics and the different affinities that these populations might have with the rest of the population. So this diagram illustrates the migration map of modern humans is constructed using the mitochondrial haplogroups. So the current accepted theory is that modern humans are originated in East Africa and they migrated out of East Africa through different routes to settle down in different parts of the world. When doing that, one of the suggested routes is this coastal route, which actually passes through Sri Lanka, and it is also one of the that reach Australia. Now, moving on to the historical evidences that we have to prove human settlements in Sri Lanka. Sri Lanka has strong evidence for human settlements by 125,000 years. And also we have evidence for anatomically modern human settlements by 34,000 with the skeletons of Balangoda man. Now this evidence is not just the oldest skeleton in Sri Lanka, also oldest skeleton for South Asia to prove the evidences for anatomically modern human settlement. The same cave that produced this skeleton, which is Fahiandana, has produced other microfossils and microlithics, and out of which one of the most famous or most significant findings is the oldest bow and arrow technology outside Africa, which is dated around 47,800. Now, collectively, we have evidence to prove that Sri Lanka did have prehistoric human settlements before the arrival of Prince Vijaya. Now, Prince Vijaya and the successive Indian migrations are affiliated with the origin of Sinhalese ethnicity, whereas the prehistoric settlement evidences can be affiliated with Adivasi. Why? Because the Balangoda man is studied to be sharing morphologies with the Adivasi group as well as Australian Aboriginal community. Now, this is where our interest comes in, because we want to understand the maternal genetic affinity of prehistoric and modern Sri Lankans, focusing on Sinhala and Ratha Adivasis with the ancient and modern populations in Eastern world and Australia. Now, to do this, we used complete mitochondrial sequences. The Sri Lanka was which also included 2 prehistoric individuals. Then these populations were selected to represent the eastern side of South Asia. Then we have populations selected for Southeast Asia, and then China, Papua New Guinea, Australia, and also Africa were selected as the outgroup. So this made up a total of 69 sequences. These 69 sequences were then aligned using the revised Cambridge reference sequence via muscle, and we constructed the phylogenetic tree through MEGA. Now this is the started tree. Then Ratugala Adivasi in green and the outgroup in red color. As you can see, we can cluster this tree into 3 separate clusters. So let me go through each cluster one by one. At first, in the very first cluster, we have 3 of our Sinhalese sequences clustered together along with our 2 prehistoric sequences. So apart from that, when we look at the haplogroup clustering, we can see 2 of our one Sinhalese and 1 prehistoric individuals are clustered as M35A. Then we have 1 Sinhalese as M45, then another as M6A1A, and our other prehistoric individual as M18A. So apart from these sequences that have clustered together with the reported haplogroups from our current Sri Lankan samples, we have both Andamanese sequences clustered together with this in the first cluster. Then we have, so in our study, we used 4 Indonesian sequences, 2 of them representing a patrilocal culture, 2 of them representing a matrilocal culture. Here, the 2 sequences are from each culture. And then the 2 ancient Chinese sequences and 1 Indonesian ancient sequence, all 3 have clustered here in the very first one. And then out of the 6 Chinese sequences, 2 of them are clustered here. And also finally, out of the 3 modern Aboriginal sequences, 1 of them are clustered here. So moving to the second cluster, here, all 5 of our Adivasis are clustered in the second one. So looking at the haplogroups, we have U7A from 1 Adivasi sequence. So U7A2 is actually a haplogroup that is popular in the Near East. But the rest of the 4 Adivasis were all reported with R30B2A. You can see here. And then apart from the haplogroup clustering, 2 of the modern Aboriginal sequences and the ancient Aboriginal Australian sequence were clustered here with the Adivasis. Then the 2 Nicobarese sequences again clustered Adivasis. Then the rest of the 4 Chinese sequences are also here. And again, Indonesian, 1 patrilocal and matrilocal came here. So lastly, we have our final Sinhalese sequence clustering separately, which is reported as M5A2. Now, in conclusion, with the different affinities we saw, we can say there is a potential maternal gene flow between Sinhalese and South Asian and Southeast Asian. And when focusing on Ratugala Adivasi group, we can say a potential maternal gene flow between Ratugala Adivasi, Nicobarese, and Aborigines, or a common source population. So that's the end of my presentation, and I would like to thank Venerable Vellampitiya Sumana Dhammathera and Dr. Nadun Vikrama Ratna, and also Mr. A. Chavilanga for assistance in collecting Ratugala Adivasi samples, then Dr. Mansa Rangavan and her team from University of Chicago for their immense support in our ongoing study, as well as the financial support from NRC. And last but not least, my ever supportive supervisors for their constant support and guidance in my PhD project. So these are my references, and thank you.
Presentation now open for questions. I think you can come to the podium now. Yeah, okay, sir, that's one question. Okay, rational reasons for selecting sequences from different populations.
So there were 2 rationales. First one was when looking at different populations, we looked into their frequencies in mitochondrial haplogroups, and we selected the most frequent haplogroup. And apart from that, we also looked into the same haplogroups that were reported from our study and checked if those populations have that and included both.
Any other questions? So in the absence of other questions, let us thank the presenter. The next presentation will be done by Anjana Valkala on mitogenome diversity
in Sinhalese, an indigenous weather population in Sri Lanka.
Hi, I'm Anjana Balikala, and my study is mitogenome diversity in Sinhalese and indigenous weather population in Sri Lanka. Sri Lanka is an island which is located in the Indian Ocean, southern to the Indian subcontinent, and it has witnessed population movement since prehistoric period. So this is a map of human dispersal routes across Asia during late Pleistocene. So during latter dispersal, which was around 60 to 30 kilo years ago, during this period, South Asian region was inhabited by anatomically modern humans, and Sri Lanka is one of the lands which gives clear archaeological evidences, especially the skeletal records, which dated back more than 38,000 years. Since then, Sri Lanka witnessed many population movements, and especially due to the strategic location of Sri Lanka in the Indian Ocean, it connected the western and eastern part of the world, and hence Sri Lankan shores were very popular among international traders. So many population migrations occurred since protohistoric period, and this has led to enhance the genetic diversity of Sri Lankan population, increasing many ethnicities, cultures, religions, and traditions. Many historical manuscripts have reported different population movements. According to Mahavansa, the first migratory group was Prince Vijay and his followers during 5th century BC. So majority of these migrations were male dominated, but however, some few female migrations were also repeated in between. Especially for the crowning of Prince Vijay, a Kshatriya princess was brought from Madhurapur, India, and other women as brides for Vijay's troop. And also for the coronation of Prince Panduvas, a Shaki prince was brought to Sri Lanka. And along with Sangamita, 3rd century BC, women who belonged to 18 castes. During the period of Tamil invasions and also due to international relationships, many female migrations were occurred. And also we know Sri Lanka has been ruled by 3 European governing systems, Portuguese, Dutch, British. So during this period also, European women were introduced to Sri Lanka. So when we talk about ethnicities, Sinhalese is a major ethnic group in Sri Lanka. So according to the statistics in 2012, Sinhalese comprised 75% of the total population. So they believe that descending from the North Indian Prince Aryan Prince Vijay and his troop who arrived to Sri Lanka during 5th century BC. But present day, scientists have found out those North Indian populations are more likely to be an admixture of native country and North Indian invaders. However, we cannot say that only this Prince Vijay story, but there could be a possibility of Sinhalese to be descending from the indigenous populations who inhabited the country before the Prince Vijay. So we consider the Adivasi Vedda population. According to Mahavansa, the Vedda population was originated by Jivahata and Disala, who were the children of Yaka, Queen Wayne, and Prince Vijay. And when we talk with the Vedda population, they say they are straight away descending from the Yaka who inhabited the island before the arrival of Prince Vijay, and they have a maternal ancestry to Queen Wayne. So we don't know that these stories are true or having a relationship to each other. But however, according to the scientific studies, there are other suggestions as well. Some studies have reported that Adivasi Vedda population have an ancestry to prehistoric Balangara culture, which spans between 40,000 to 12,000 years. And especially the skeletal analysis and some genetic marker studies have proved this theory. And also there are some studies indicating that Vedda populations are related to South Indian tribal groups and also to Southeast Asian populations. So having this knowledge, we wanted to understand the maternal relationships and evolutionary adaptations of the Sinhalese population. So we Sanger sequenced 31 complete mitochondrial genomes, 16 from Sinhalese, 4 from Dambana, 11 from Henanigala. Actually, the Henanigala Vedda population belonged to the Dambana clan, and some of the individuals were relocated in Henanigala in 1986 due to Mahabali irrigation project. Then the sequence were aligned with the revised Cambridge reference sequence using Unipro Eugene software, and the variants and their positions were confirmed. Mutations were then analyzed with MPOP, haplogroup, and Phylotribule 17 databases. Index and analysis of molecular variants were calculated, and the evolutionary history was in MegaX software. So according to the results, high maternal diversity were observed in both populations. So this is the haplogroup diversity in Sinhalese and the haplogroup diversity in Vedda samples. So even the sample number is very low, we observed high maternal diversity in both populations. And also we observed higher variance within the population. And in individuals, M haplogroup was predominant, and in Vedda population, U and R haplogroup was prominent. So this is a mitochondrial DNA haplogroup tree and distribution map. Here you can see how haplogroups are distributed among all over the world. So when talk about Sinhalese individuals, majority of the haplogroup we observed is M, and here you can see the M haplogroup is prominent in South Asian region. So we can say the individuals, the majority of the individuals we analyzed are having ancestry to South Asian region. But when we talk about the Vedda population, again, our haplogroup is common in South Asian region. But when we consider the U haplogroup, it is very prominent in West Eurasian countries. So with respect to Vedda population, we can say the majority of the individuals we analyzed coming either from South Asian, having a maternal ancestry either to South Asian region or West Eurasian countries. So in addition, we identified 5 novel haplogroups. So this is current haplogroup given by haplogroup database, and these are the novel haplogroups suggested. So we know when a particular population is settled down in a specific geographical region, they adapt to that environment. So for this adaptation, their genome changes, the mutations occur in their genome. So in 2018, there was a study done on Jammu Kashmir population in India. And in that paper, authors have reported haplotypes shared by at least 2 individuals, unrelated individuals, were considered to constitute a new haplogroup. So in this paper, we also did the same. Individuals at least share, at least 2 individuals share the common variant. We assigned a new haplogroup, and we suggested an extension to the current haplo tree. So this is the evolutionary relationship of the samples analyzed by MegaX software, and we constructed neighbor joining tree. So these are the novel haplogroups we suggested. Here you can see some of the Vedda individuals having closer ancestry to Sinhalese individuals. So in present day, Vedda population is highly intermixed with neighboring Sinhalese and Tamil population. So these kind of relationships are very common in Vedda population in present day. So in conclusion, we can say the presence of distinctive haplogroups may indicate early migrations and evolutionary adaptations of the 2 populations in Sri Lanka. So these are some of the references I used. So this is this study is a part of my MPhil PhD project. So I would like to thank my supervisors, Dr. Ruwandi Ranasinghe and Professor Kamini Tennekon, for giving me continuous support and guiding me throughout this period. Thank you very much.
Presentation is now open for questions. there are no getting a conflict. What are the genetic studies conducted to show that we can have people come?
Okay. So actually, when we talk about the scientific studies, we can talk about the comparing the skeletal records, skeletal remains of the population. So it was done by actually the pioneer archaeologists, Professor Daraniagala and Kennedy. They have found out that the population having an relationship to Balangoda culture. But when it comes to genetic studies, some genetic studies have done on based on blood genetic markers in early 90s. So there, in 1986, Ellepola and Vikramasinghe has done on blood genetic markers on the population, and they have found out they have an ancestry to Balangoda culture, and also it goes around at least 12,000 years ago. And also when it comes to there's people say that they're having a tribal ancestry to the tribal populations in India. So in 1986, 83, Roy Chowdhury has done on tribal comparison on the population and tribal population in India. They have found out that Irula population having an closer relationship to the population in Sri Lanka. And also when it comes to Southeast Asian ancestry, in 1983, Vikramanayaka has done on genetic marker study on 6 Vedda population in Sri Lanka, and they have found out, they're telling that Vedda population has an ancestry to East Asian groups. But when it comes to genetic studies, today you could understand based on our studies where the population is having haplogroups related to tribal populations, and also haplogroups related to Southeast Asian groups. Yes, I went because I wanted to. I read a lot about it. Yeah.
Perspective, like look at the language from language here. Call that kind of a pure,
yes,
kind of pure language, kind of pureization of a language. So look at the early work on that time, like the Mark.
Yes, sir, I was.
And also there's a separate study on the language science in English. And also that this pure picture of like that was the language and it is and pure,
yes,
pure language, kind of a functional language.
Yes.
So that is the kind of picture of the population and your study in the linguistic kind of picture, the other groups, yeah, or also there is a linguistic perspective also. There is a architecture which is called
yes.
But some of my students did work in Japan about how languages change. Thank you for giving
So that concludes the oral presentation. presentation, session 5. And thank you for very interesting presentation. And we must actually thank researchers and Dr. particularly for merging with molecular biology with the history. And I think it is an area where you are very difficult to find funding. So thank you very much for the interesting presentation, and let's conclude with session 5.
Thank you to the chairpersons. Next, we will have session 6 of oral presentations looking at molecular genetics and genomic medicine. We invite the chairpersons, Professor Jagat Veerasena and Dr. Meran Edrivira.
Molecular genetics and genomic medicine. Preliminary study on mitochondrial genetic variations in sporadic dissemination of Sri Lankan Tamil, Sri Lankan Moor, and Sri Lankan Sinhalese population.
I will be presenting on my research looking at a preliminary study on mitochondrial D-loop variations in sporadic breast cancer patients of Sri Lankan Tamil, Sri Lankan Moor, and Sinhalese populations. What is mitochondrial DNA? Mitochondria are organelles that are found in the cell, and they are higher in number in metabolic cells. They contain their own DNA and are unique in that sense. This DNA is double-stranded, circular, and helical in structure. The light strand is called the L strand, and the heavier strand is called the H strand. The DNA consists of two major regions, the coding region and non-coding region, that is also called the D-loop. The D-loop consists of 3 variable regions and 2 PC tracks. Although this is the non-coding region, it has a significant role in the regulation of transcription and replication of mitochondrial DNA. Mitochondrial DNA exists in close proximity to reactive oxidizing species and hence are prone to acquire variations. Leigh syndrome and Leber's hereditary optic neuropathy are no mitochondrial diseases. Non-communicable diseases such as diabetes, cardiovascular disease, and cancers are also associated with mitochondrial DNA variations. What was of most interest to us is the work done on identifying a biomarker to predict cancer at an early stage and to also predict drug response as well as cancer progression. However, we must remember that mitochondrial DNA variations are population specific and variations can occur within and among populations. So this made it important for us to study the Sri Lankan population to correlate with the global findings. If we look at the Sri Lankan population, it consists of many ethnic groups. The Sinhalese population is the majority, accounting 24.9% of the population, while Sri Lankan Tamil accounts for 11.2% of the population, and Sri Lankan Moors account for 9.3%. If we look at cancer in the general population, we see that breast cancer accounts for 26% of cancers diagnosed in females in Sri Lanka. It is still the major cancer in females. So what did we do to obtain our data? We received ethical approval from the Colombo Medical Faculty, and we recruited 35 patients of Sri Lankan Tamil ethnicity and 31 patients of Sri Lankan Moor ethnicity. We obtained their consent, interviewed them to determine their medical history, and blood was drawn by a medical professional. Those with a family history of cancer were excluded from this study. Healthy controls from a prior study at IBMBB were obtained from each ethnicity. Miller's sorting out procedure was used to extract genomic DNA, and the non-coding region was amplified using primers 23 and 24. The purified product was subjected to Sanger sequencing, and the sequences were compared against the revised Cambridge reference sequence using Mutation Surveyor, and Fisher exact test was used to determine the significance of these SNPs. We plotted the negative log p-value versus the base position of these SNPs. We identified variation 146 and 489 to be significantly associated with cancer in the Sri Lankan Tamil population, while variation 480 was significantly associated with Sri Lankan Moor population. In regions 16,000 to 16,569, 16,129, 618, and 16,223 were significantly associated with cancer in the Sri Lankan male population. If we cut variations that show positive association and compare them against our previous published data on the Sinhalese population, we see that in the Sinhalese population, there was almost no difference between patients and healthy controls. If we look at 16,223 variation, which was observed in 24 Sri Lankan Tamil and 18 Sri Lankan Moor patients, we see that 14 and 8 patients were classified with invasive carcinoma, and 8 patients of each ethnicity were classified with Ki67 levels above 10%. Ki67 is a proliferative protein and is associated with metastasis. It is particularly interesting that this variation was observed in 2 instances of invasiveness. The variation 80T to C was only observed in Sri Lankan Moors, where 4 patients were classified as invasive ductal carcinoma, and variation 16,129 was observed in 8 Sri Lankan Tamil patients, where 2 patients were classified as having lobular carcinoma. These 2 variations did not show any association with the clinical condition. Now, if we look at what all this data means, variations did not show significance in the Sinhalese, but they did show association with disease in the other 2 ethnies. If we look at these variations in other populations and studies, we see that 16,223 was observed, however, was not significant in breast patients of Malaysian ethnicity. 16,223 and 489 was shown to affect transcription of mitochondrial genes, resulting in increased respiration. This is significant because increased respiration is linked to a higher metabolism, and we know that cancer cells display a higher metabolism. If this transfers to germline variations, then we could see an interesting observation here. 16129 and 16489 was observed in just 7% of breast cancer patients in the study by Losanov et al. 1623 again was associated but did not show significance in colorectal cancer patients population. Variation 146 T to C showed association with poor survival of hepatocellular carcinoma, although it was not in cases of breast cancer. Interestingly, variation 16189 is associated with a variety of metabolic disorders. We know cancer is classified as a metabolic syndrome, and in that instance, it is also interesting that variation is associated with cancer patients of Chinese and Indian populations. Variations that we observed are all transitions. Zheng et al. Also showed that transitions in mitochondrial DNA are most likely due to be errors by polymerase gamma, the key replicative polymerase in mitochondrial DNA. With this, we can conclude that these variations show association with disease in this study sample. However, we must verify these biomarkers with individual populations, and this study gives evidence for that need. A follow-up should be done with the controls, as we may have detected variations in the DNA, but they may not have been diagnosed yet. A follow-up will help us to determine their overall health status. Studying specific types of cancers, especially somatic cancers, will help us to look at mitochondrial DNA variations and their implication in the tumor tissue. Mitochondria does not work alone. It works together with nuclear encoded mitochondrial factors, which also play a role in disease. Studying factors such as polymerase gamma and its association with cancer will also help us to get a better idea as to how the nuclear and mitochondrial factors work in tandem. Thank you.
Is now open for if you find any sample having variations of 16228 who is going to do this subjective, then how do you relate the online improvement?
So the first question, we did have of the samples, 22 of the Sri Lankan Tamils shared both 489 and 16223. And those were the same two variations that had been identified to in mitochondrial hybrids. So that's not really connected to the germline, but it could give us an idea as to what happens physiologically. So that's the answer to the first question. Second, germline in relation to somatic. Studies have shown that the germline can be a precursor to the somatic variations that could cause disease. In addition, they've also seen that the germline variations can be sustained within somatic tissues, while some might be selected out. How that happens is not really clear yet, according to the data that is available. But I think that is, like I mentioned, studying, following up with patients would really be helpful in that sense, because we may have either detected the variations early, or the variations may have moved away as well, because again, mitochondrial DNA variations in blood is seen to sort of get selected out sometimes in rapidly dividing cells. So it's a precursor, and it does get sustained in somatic tissues, but in the blood it could get sort of transitioned out.
Are there any diagnostic tests you are using?
Not at the moment. Not at the moment, sir, but there is a lot of studies, a lot of diagnostic mutations that are coming out from other studies. And it's not really, for example, some of the variations that were found in the South Indian population to be significantly associated with disease was not associated with the Sri Lankan population, with any of our populations. So it's, even if they do come up with a diagnostic test, I think it does need to be verified for our population first before it can be used definitively. And there's also the other side of mitochondrial variations would be working with the nuclear variations. So we need to look at those factors as well.
I'm Umayal Branavan, lecturer attached to the Department of Obstetrics and Gynecology, Faculty of Medicine, University of Colombo. I did my PhD in polycystic ovary syndrome. I'm here today to present my research on the role of KIS1 KIS-PEP GPR54 system in the development of polycystic ovary syndrome in Sri Lankan women. So let's begin with the background. Polycystic ovary syndrome is a common endocrine disorder of women of reproductive age. It's a lifelong condition, usually from puberty, and leads to many reproductive and metabolic implications. Diagnosis of PCOS based on Bottram criteria, which requires the presence of 2 out of 3 clinical features. The first one is oligomenorrhea, that is irregular menstrual cycle. The second one is clinical and or biochemical hypoandrogenism. And the third one is polycystic ovary on ultrasound. However, diagnosis of PCOS in adolescent requires all 3 clinical features to be present. The clinical presentation of PCOS include irregular menstrual cycle that results in infertility, and hyperandrogenemia leads to acne, hirsutism, and hair loss. The most importantly, PCOS is commonly associated with insulin resistance and obesity, and insulin resistance results in acanthosis nigricans in some cases. So this is a diagram showing the difference between normal ovary and the polycystic ovary. In the normal ovary, you can see only one follicle, that is primary follicle, develop and they mature and release the egg. But in polycystic ovary, multiple follicles develop and they never get mature enough to release the egg. So this results in follicular arrest and irregular cycles in PCOS. So the pathophysiology of PCOS is poorly understood, but the study says it's a complex trait that results in interaction of genetic and environmental factors. So the genetic factors can be polycystic ovary, hypoandrogenemia, and insulin resistance. The environmental factors can be prenatal and this also results in PCOS in some cases. So all these results in disturbance in hypothalamic pituitary gonadal axis. So when the hypothalamic pituitary axis is disturbed, it results in increased GnRH secretion, which leads to increased LH pulsation, and which leads to abnormal secretion and regulation of gonadotrophins, that is LH and FSH hormones. So this increased secretion and action of ovarian steroid hormones results in subfertility. The most important thing is this elevated LH and FSH ratio is a useful biochemical diagnosis tool for diagnosis of PCOS. So this is a diagram showing the pathophysiology of PCOS. As I said, genetics, obesity, lifestyle, intrauterine abdominal exposure all can lead to PCOS. All these factors can stimulate the release of GnRH from hypothalamus, and GnRH hormone stimulate the release of LH and FSH hormone. which ultimately lead to increased androgen secretion. This increased androgens can cause the symptoms of hirsutism, acne, and alopecia. The another thing is increased LH also acts on ovarian theca cells and produce more androgens. So this excess androgen results in follicular development and leads to follicles and polycystic ovaries in polycystic ovaries. So recently, peptide known as kisspeptin was found. So this kisspeptin is synthesized from the gene called KIS1 gene. This kisspeptin acts by binding to its receptor known as GPR54. So these GPR54 are found in the neural
molecular surveillance of artemisinin resistant K13 mutation
its receptor, kisspeptin stimulates the release of GN1 from hypothalamus. So our main study
was seen in the nucleotide codon position of 1000
and their association
consensus
with
sequences
in Sri Lanka.
for all isolates was done using bioedit
So the rationale
software.
for the study is a PCOS is a
Thereafter,
major health problem of women of reproductive age, and the community prevalence of PCOS is 6.3 in Sri Lanka. So as I said, the SPG axis plays a major role in the development of puberty, and PCOS is commonly manifest from puberty. So the genes involved in HPG axis and their association with PCOS need to be studied. However, the many studies focus on genes of obesity, insulin resistant, and FSH and LH hormones. There are no studies that were carried out in the genes of KIS1 and GPRF54, which is the starting of HPG axis. So we mainly focus on these unexplored genes of HPG axis, that is KIS1 and GPR54, and their association with PCOS. Most importantly, the genetic risk can be identified well before puberty. So identifying the genetic cause at the early stage will enable early treatment management and reduce the severity of symptoms. So our aim of the study is to identify the role of KIS1, kisspeptin, GPR54 system, the development of PCOS. So this is a diagram showing our methodology, and we totally recruited 165 subjects. Out of the 165, 55 were cases and 110 were controls. We extracted 10 ml blood from each subject and separated serum, and serum was used for the ELISA assay of serum kisspeptin to determine the serum kisspeptin levels. And then we extracted DNA from EDTA blood, and we outsourced those DNA to carry out next generation sequencing of GPR54 and KIS1 genes. So this is a table that shows the results of clinical and biochemical characteristics of our study population. And you can see that serum kisspeptin levels were significantly high in PCOS subject. We also found serum kisspeptin levels were positively associated with PCOS with the odd ratio of 1.85. So this table shows the results of next generation sequencing of GPR54 and KIS1 gene. And we found total 5 SNPs in GPR54 gene. Out of these 5 SNPs, 2 SNPs were novel polymorphism. We also found 2 SNPs from KIS1 gene. Then we carried out binary logistic regression analysis to identify the risk factor for the development of PCOS. So after adjusting the confound factor, we found only serum kisspeptin levels were significantly associated with PCOS. We conclude kisspeptin can be a risk factor for the development of PCOS in Sri Lankan women. And we found there were no association between KIS1 and GPR54 gene metabolism and PCOS. To conclude, we concluded that Sri Lankan women with PCOS exhibit significantly higher serum kisspeptin levels than the controls. And these kisspeptin levels are positively associated with PCOS in Sri Lankan women and can be used as an early marker of PCOS. And we also found 2 novel polymorphisms in GPR54 gene, And genetic variations in GPR54 and KIS1 genes are unlikely to be associated with PCOS in Sri Lankan women. The heterozygous array of our novel polymorphism in GPR54 gene were associated with higher serum protein concentration, suggesting a potential role in the etiology of PCOS in Sri Lankan women. However, further studies should be carried out to confirm this finding. Thank you.
Online. Can you hear? Yes, Kumar, can you hear?
Yes, I can hear, but the volume is low.
Can you hear us now? Are you there? Are you there?
Yes, I am there.
Okay. Yes, Kumar, I have 2 questions. Yes, Sumayal, I have 2 questions for you. Just general questions. Can you please tell whether you have come across any drug which can target this signaling axis? That is KIS1, KIS15, GPR54. That is my first question. Second one is, are there any records available that shows the role of this signaling axis in the development of any other diseases other than PCOS? That is my second question.
Okay, in Sri Lanka, we found this is not the only genes, two genes I studied. I have studied totally 11 genes. So from this 11, I found only the obesity genes were associated with PCOS in Sri Lankan women. And the KIS1 was studied only related to PCOS because of this, it plays a major role in the development of puberty. So the puberty starts with the HPG axis, onset of HPG axis binding to the kispeptine binding to its receptor. So this, since the PCOS commonly manifests from puberty, these KIS1 genes are commonly studied in PCOS.
Right. The second question, role of this pathway other than PCOS? This KIS1.
No, I think this KIS1 gene is commonly studied in the PCOS. I haven't come across any. There's another disease called central precursors puberty, that is CPP. It has found some major association of KIS1 and GPR54 genes with the CPP, but in PCOS, there's not much study done. So that's why we selected the whole HPG axis gene in our PhD.
Right. Thank you, ma'am.
Thank you very much. Next presentation is the prevalence of PNPLA3 polymorphism in a form of non-alcoholic steatohepatitis related hepatocellular carcinoma patients in Sri Lanka. and say its stage in summary.
Hello everyone, I am Soumya Samarasinghe. My presentation topic is prevalence of PNP-LA3 polymorphisms in a cohort of non-alcoholic steatohepatitis related hepatocellular carcinoma patients in Sri Lanka. Hepatocellular carcinoma is a primary liver cancer arising predominantly in patients with chronic liver disease. The below image shows a large, well-vascularized solid tumor in the background of healthy liver tissue. Major risk factors for HCC include alcohol addiction, chronic viral hepatitis infections, dietary toxins such as aflatoxins and aristolochic acids, and non-alcoholic fatty liver disease, which our study is focusing on. Non-alcoholic fatty liver is the most primary form of non-alcoholic fatty liver disease spectrum, which is characterized by the fat accumulation in more than 5% of hepatocytes without any inflammatory activity. Fatty liver can progress into a much severe form, non-alcoholic steatohepatitis, which is histopathologically characterized by hepatic steatosis with the necroinflammatory components with additional characters like fibrosis and hepatocellular ballooning. NASH can progress into fibrosis or cirrhosis where hepatocytes are being replaced by scar tissue, which can form HCC characterized by a thick fibrous tissue comprised of malignant tumor cells. Also, a significant proportion of NASH patients are directly progressing into HCC without having any fibrosis or cirrhosis stage, and that is of major concern. Genetic polymorphisms plays a main role in predisposing NASH patients directly into HCC among the other risk factors such as obesity, insulin resistance, hepatic lipotoxicity, increased iron accumulation, and advanced age. When considering the genetic polymorphisms predisposing to NASH HCC, patatin-like phospholipase domain containing 3 or PNP-LA3 and its variants have shown close relationships with liver diseases. PNP-LA3 is highly expressed in the liver tissue and shows activities such as triglyceride hydrolysis activity, trans-esterification activity, hepatic triglyceride efflux, and lipid droplet remodeling. Also, PNP-LA3 gene variants have shown strong associations with a large spectrum of liver diseases, including steatosis, liver injury, NASH, HCC, cirrhosis, and fibrosis. PNP-LA3 gene is located in the long arm of chromosome 22, and among its polymorphisms, we have selected 3 most common single nucleotide polymorphisms for our study. First polymorphism is PNP-LA3 RS738409 C to G variant, which substitute isoleucine to methionine at the position of 148 in its amino acid coding region. Second mutation is PNP-LA3 RS2281135 intron variant, which changes a guanine into an adenine. Third mutation is PNP-LA3 RS2294918 missense variant, which changes lysine into glutamine at the position of 434 in its amino acid coding region, which changes adenine into an guanine. The C to G non-synonymous variant has shown reduction of hepatic triglyceride hydrolysis activity in the PNP-LA3 protein. The G to A intron variant has shown associations with hepatic triglyceride content. The A to G missense variants have shown associations with NASH and NAFLD severity. The objective was to analyze the genetic background of Sri Lankan NASH HCC cohort with respect to PNP-LA3 gene polymorphisms. After getting the ethical clearance to represent the Sri Lankan NASH HCC population, we have obtained a cohort of 48 NASH HCC patients from the co-investigators clinic in North Colombo Teaching Hospital, Ragam. The NASH HCC patient did not show any significant alcohol intake before, and they did not show any family history of medical diseases except NAFLD. They didn't have any infective hepatitis screening, and they had CT or MRI reports showing typical enhancing patterns for HCC. Then the patients were informed about the research, and clinical and demographic data were collected using investigator-based questionnaire, and informed consent was obtained from the study participant. Blood was withdrawn from the study participant, and blood samples were separated and stored in appropriate conditions. DNA was extracted from collected blood, and primers were designed for the short-range PCR and as well as for the SMP analysis. And primer sets were optimized. Then short-range PCR was carried out, followed by PCR purification and quantification. Then primer extension-based SMP analysis was followed, and results was analyzed using appropriate softwares. The polymorphism, 79% short heterozygous genotype. When considering G to A polymorphism, 29.16 short homozygous genotype and 4.16 short the heterozygous genotype. When considering A to G polymorphism, 64.58% short homozygous genotype and 25% short heterozygous genotype. Moving to allele frequencies, when considering the C to G polymorphism, the risk allele or the G allele showed a high frequency of 0.79, which was statistically significant. The G to A polymorphism showed a low frequency for the risk allele or for the allele showing a 0.3. Considering G polymorphism, it showed a statistically significant risk allele frequency showing a value of 0.9. When comparing the observed risk allele frequencies with other population studies, when considering C to G polymorphism and A to G polymorphism, similar high frequencies for the risk alleles were observed in other population studies. When considering G to A polymorphism, low frequencies for the risk allele were observed in other population studies. Finally, I would like to wind up my presentation by denoting some demographic characteristics of Sri Lanka NASH HCC cohort. When considering the age distribution, males who are aged in between 65 years to 69 years had showed the most prevalent for NASH HCC in the Sri Lankan cohort. When considering the BMI categorization distribution, nearly 30% of the NASH HCC patients were pre-obese, 40% were of normal weight, and 19% were of obese BMI. When considering diabetes status, nearly 60% of the NASH HCC patients were having diabetes as a comorbidity. Therefore, I would like to finalize my conclusions. Our cohort of patients has a significantly high frequency for PNP LA3 RS73 8409 C to G variant and for PNP LA3 RS2294 918 A to G variant, and a high frequency of the same variants has been shown in the population studies. 2281135 G to A variant, which was less prevalent in our cohort, had shown same low frequency in our populations. Majority of the NASH HCC patients were male and their age above 65 years. Diabetes and pre-obesity were prevalent metabolic risk factors that were observed in Sri Lanka NASH HCC cohort. These are some of my references, and thank you so much.
Presentation is now open. We use primer extension
It's not an allele-specific allele-specific detection method. It's just a short-range PCR, just followed by a single. We are detecting a change in a single base, and it's like single base detection, like single base change detection method. Yeah. Yes, sir. Actually, I have designed the timers for the 10 strand, like if that you are asking, like it's not actually allele specific. That's what I'm saying. Yeah.
Design from the
yes. Yes, 3 primers. Yeah, I'm like designing the primers just before the single nuclear polymorphism at the 3 primers. So in the detection step, it's detecting the next base. Therefore, it's extending at the 3 primers. Yeah.
next presentation is molecular surveillance of Parkinson's resistant K30 mutation in imported Plasmodium falciparum cases diagnosed in Sri Lanka.
Molecular surveillance of artemisinin-resistant K13 mutations in imported Plasmodium falciparum cases diagnosed in Sri Lanka. Rationale: artemisinin-based combination therapy that rapidly clears parasites from blood is the drug of choice for treatment of uncomplicated Plasmodium falciparum malaria. Emergence of artemisinin partial resistance has led to delayed parasite clearance, resulting in day 3 parasitemia, and if the partner drug is resistant, this can result in treatment failure. Initially, this artemisinin partial resistance was reported in Southeast Asia. However, now this has spread to countries in other regions also. The importance of molecular surveillance of artemisinin resistance has been highlighted by the demonstration of a strong correlation between point mutations in the Kelch protein propyl domain K13 and delayed parasite clearance following treatment with ACT. At present, there are 10 validated mutations in the Plasmodium falciparum K13 propyl gene. This map shows the countries where molecular markers of PFK13 mutations have been detected so far. So therefore, the objective of this study was to determine the molecular epidemiology of K13 genotypes in imported Plasmodium falciparum patients diagnosed in Sri Lanka during the phase of prevention of re-establishment of malaria. Methodology. Ethical approval for this study was obtained from the Ethics Review Committee of the Sri Lanka Medical Association. The study setting consisted of the Central Laboratory of the Anti-Malaria Campaign and IBMBB, University of Colombo. Sample collection was performed from April 2014 to December 2017. And the study population consisted of patients referred to anti-malaria campaign for malaria confirmation, that is, those tested under passive case detection, and also the asymptomatic high-risk individuals that were screened under active case detection were also included. All Plasmodium falciparum confirmed patients detected from these categories were included for this study. So the laboratory work, initially, extraction of DNA from blood samples of the Plasmodium falciparum patients was done. So this was done on the first day of diagnosis prior to initiation of treatment. So there were 83 such Plasmodium falciparum patients. Then a nested PCR assay was performed to amplify a 849 base pair fragment in the Kelch protein propeller domain in chromosome 13. The gel electrophoresis of this amplified product was done using a low melting agarose gel, and the PCR products were excised from these agarose gels, and the purification of this excised product was done using the Visa gel and PCR cleanup system from Promega USA using the manufacturer's instructions. Thereafter, cycle sequencing reactions using applied biosystem big dye terminator V3.1 cycle sequencing kit was done. The purification of the extension products was performed using ethanol EDTA sodium acetate precipitation method, and capillary electrophoresis of the purified products was performed using an ABI 3500 DS genetic analyzer. The analysis of electrophoregram and sequences and preparation of consensus sequences for all isolates was done using BioEdit software. Thereafter, the comparison of consensus sequences to determine the mutations was performed using the reference sequence tree-driven isolate using NCBI BLAST. Results. So this slide shows the outcome of the sequencing performed for the K13 gene among the 83 Plasmodium falciparum samples. Out of the 83 Plasmodium falciparum patients, there were interpretable sequences in 72 samples. Out of these 72 interpretable samples, 62 had the similar sequences to the 3D7 reference allele. Importantly, one sample had a sequence with non-synonymous mutation. That was R561H, a validated marker. Then there were 9 synonymous mutations also. So this slide shows the position of non-synonymous mutation and the synonymous K13 mutations that have been shared by 7 isolates. As you can see, the non-synonymous mutation was seen in a sample coded as P146, which had ATP instead of GTP at the nucleotide position 1682. 7 of the 9 synonymous mutations were seen in nucleotide codon position of 1644. This slide shows the details of the patients harboring the non-synonymous mutation and the 7 patients harboring the same synonymous mutations. The patient with the PCR sample coded as P140 had come from Rwanda, which has this the validated marker, that's a country that validated marker is common. And even though the parasitemia is low, that patient had parasitemia even on D3, indicating delayed parasite clearance. So this, the non-synonymous mutation with the D3 parasitemia indicates artemisinin partial resistance. So in conclusion, we can say the presence of the validated marker R561H and persistent parasitemia on day 3 confirms artemisinin partial resistance. So this is the first report of artemisinin resistance in Sri Lanka. So this findings show the risk of importation of K13 mutations to the country. And therefore, this highlights the importance of molecular surveillance to prevent the occurrence and spread of drug-resistant malaria in Sri Lanka. Acknowledgements. Financial assistance from the NSF grant is greatly acknowledged, and the staff of the Institute of Biochemistry, Molecular Biology and Biotechnology, and the director and laboratory staff of the anti-malaria campaign headquarters are also acknowledged, and especially the patients for their cooperation is acknowledged. Thank you.
Now open for can you hear?
Can you hear us?
Hello, yes.
So this may not be related to the work you carried out. So if in case if we detect artemisinin resistant patient, so how do we manage the disease?
Even though there is a delayed parasite clearance, if the patient parasitemia is cleared, after that, that would be okay. But we have to monitor that that particular genotype is not transmitted to the country. So we have to take preventive measures. Other than that, partial resistance is a big threat, but the patient can get the parasitemia cleared. However, if the partner drug is resistant, then it would be an issue. So out of another second line drug should be given to that patient so that all patients actually will have to be monitored until parasitemia is cleared. So that is been done at the moment for all falciparum patients to ensure parasite clearance, total clearance.
Thank you. We will move on to the next.
Hello everyone.
Next presentation is detection of hotspot region of WT1 gene in response of 45 BST events in Sri Lanka. The presenter is
To be here today. I'm Arambage and I'll be sharing hotspot regions of WTO in a cohort of 6 in Sri Lanka. Sex phenotypes, male and female, in which males have 46XY genotype, while females have 46XX genotype. Genes involved in SOX9, SRY, WT1, SF1, so and so on. When developing a healthy baby boy, the initial potential gonad is developed into a fully functional testis via a process known as sex determination. Involvement of WT1 protein, SF1 protein, for the regulation of SRY gene, which finally results in upregulation of SOX9 gene, is important for this. In our research, we focused on WT1 protein, and this diagram shows the initially biopotential gonad developed into the involvement of WT1 is encircled in red, which shows the involvement of WT1 steps in multiple steps in sex determination. A condition known as differences or disorders of sex development could arise due to malfunctioning of these biochemical pathways or due to a genetic reason during sex determination. DSD are congenital conditions in which development of chromosomal, gonadal, or anatomical sex is atypical. Mutations in transcription factors, signaling components, and epigenetic modifiers can result in DSD. Gonadal phenotypes may range from gonadal dysgenesis to varying degrees of overtestis. Diagnosis of DSD starts from blood typing, measuring androgen levels, serum DSD levels, LH-FSH hormone levels, endoscopic examination, pelvic ultrasound, MRI, laparoscopy, or assessment of gonad, size of phallus, height of urinary meters, and screening of genes including WT1, AR, SF, SOX9, and others. DSD can be categorized into three main categories: 46XY DSD, 46XX DSD, and overtesticular DSD. In our research, we focused on 46XY DSD. These patients are genetically male and they may have ambiguous genitalia, palpable gonads, scrotal folds, inguinal bifid scrotum, perineal urethra, subfertility, hypospadias, micropenis, and failure to develop secondary sexual characteristics. These diagrams show the newborns where the most appropriate sex of rearing is not immediately clear. The importance of checking the WT1 gene in setting of 46XY DSD. DSD are often the first symptom of WT1 mutations in male patients, and mutations in WT1, SF1, SRY, SOX9, and MAP3K1 are the most frequent molecular causes of 46XY DSD. WT1 gene, the extended form is Wilms tumor suppressor gene 1. It has two synonyms, WIT2, WT33, and is located in chromosome 11 p13 band. It's crucial for the development of the early forms of kidney and for the formation of gonads. The gene has 11 exons and it was first isolated in 1990. It has two alternative splice sites and codes for WT1 protein, which has transcriptional regulatory domain and C-terminal zinc finger domains. The mutations in WT1 gene are concentrated in exon 9 and 10. They are usually missense and nonsense, and these mutations can result in malfunctioning or complete loss of function of WT1 protein, and some mutations are lethal. Homozygous mutations in this gene can be found in 15% of the Wilms tumors. This diagram is a graphical representation of all the exons in WT1 gene, in which exon 8, 9, and 11 code for the zinc fingers of the protein. In our research, we concentrated our focus on exon 9 and 10, where the most mutations are occurring. The protein WT1 has 449 amino acids. The transcription regulatory domain is as N-terminus, and the zinc finger domain is at C-terminus. There are several isoforms, and it is capable of homodimerizing. There are many targets of WT1 protein, including SRY, IGF2, NPHS1, EGFR, TAX2, and amphiregulin. There are diseases associated with WT1 gene: Wilms tumor, Dennis-Drash syndrome, Fraser syndrome, WHGR syndrome, Nickel syndrome, and nephrotic syndrome. Wilms tumor results as the WT1 gene mutations and accounts for 90% of total pediatric renal cancers. Most cases are unilateral, and 10 to 20% somatic mutations are observed in sporadic Wilms tumors. The estimated percentage of Wilms tumor with WT1 mutation is 10 to 15%. These are the signs of Wilms tumor. Our work is as follows. We started by extracting blood DNA and Sanger sequencing. The sequence files were analyzed using the softwares mentioned here. We amplified exon 9 using one primer set, and the product size was 508 base pair. The second set of primers amplified exon 10 and part of intron 9 overlapping completes the complete intron 9. This is the gel image of PCR products. We have detected one benign variant in 3 of the patients by mutation survey software demo version. This is the reference WT1 sequence, patient DNA sequence, and the variant position is denoted by a red arrow. One pathogenic variant was detected in one of the patients. So this is the patient DNA sequence, the reference sequence, and the variant position denoted by a red arrow. This is the summary table of the detected variants during our research. 3 patients had intronic benign variant, 1 patient had missense pathogenic variant. Patient 1, patient 3, and patient 6 had all these variants, while patient 3 had the pathogenic variant present. So the multiple sequence alignment of the patient amino acid sequences is shown here, where the reference sequence of the protein is mentioned on top and below the patient amino acid sequences. The observed pathogenic variant is located at arginine 467 amino acid of the protein, where the mutated position of the patient 3 is detected and is denoted by a red arrow. The detected pathogenic variant always defines 3D structure of the WT1 protein, and due to this, the steric hindrance of the zinc finger domains is observed, and the altered sequence cannot bind with the consensus DNA sequence that it is supposed to bind, and may result in mismatch of the transcript factor. And it is enabled to homodimerize due to this mutation. So this diagram shows the normal WT1 bound to DNA. Most commonly observed variant position is denoted by purple amino acid. So this is the zooming in of the same view, where we can see that the arginine is interacting with DNA. So the detected pathogenic variant is the most common and is a hotspot where it changes the wild type positively charged arginine into neutral tryptophan. In this diagram shows the mutated variation of the protein in which interaction of the DNA is denoted here. So the pathogenic variant detected in patient 3 was in heterozygous condition in this cohort. The patient was diagnosed as having nephrotic syndrome. The normal WT1 protein can be synthesized in this patient, but is at risk of developing Wilms tumor. Previously reported non-pathogenic variant in intron 9 was detected in 3 patients, and there are no variants in exon 9. And none of the patients had family members with DSD. So the patient having the hotspot mutation must be followed up until adulthood for symptoms of these diseases, and use of gene panels for detection of DSD-related genetic mutations is suggested, while study of WT1 mutations in a larger cohort is also encouraged. I have used these references for preparation of this presentation. Thank you very much for your attention, and I'll welcome any questions you might have. Thank
you. The presentation is open for questions.
What's the sample size?
So we used 7 patient samples.
So, your next step is to do the lab cohort. So, what kind of cohort is select?
So initially we selected the total of, we targeted for 20 patient samples, but due to COVID situation, there were very less attendance. But however, we have collected the samples and it will be sequenced hopefully tomorrow, all the 20 samples.
So what has the presentation or you're going to do it in larger cohort?
If it is possible, we would also like to do, but thank you.
Now we will conclude the cohort. I hope the findings will be thank you for sharing your findings.
Rudolph. So over to you.
So thank you very much for the presentation. I hope that you can hear me well.
Yes, we can.
Thank you. First of all, I would like to thank for the invitation. It's really great. I was listening to the previous presentations and to see all the progress and all the work that's being done. Nowadays, you may remember when we started collaborating, it was the initiation of bioinformatics, but now I see that most projects are using bioinformatics and move with terms and tools and terminologies without any issues. So that really shows the progress of the area that I'm usually representing. I'm very, very happy to see that. I'm less happy that I would not be with you there because of last moment issues. I also can tell you that even if I had been there, I got a terrible post-COVID issue here. So I'm being in bed 2 days. So you can hear my voice is not perfect, and I'm not very clear in my brain. I get tired at the moment. But anyway, I will do my best and give my presentation. And I hope if you have questions that you can make them. So as the title said, it's kind of a holistic view of what is being done at the moment and what will be done in the future in the life science area. I make the travel or the journey from precision agriculture to personalized medicine. And I hope that the message will be more clear with the coming slides. I start nowadays because I've been in several seminars where people discuss the issues of having climate change, and you all know COVID, which is a zoonotic disease. Those zoonotic disease will increase in the future, but not many of the talks point the major reason to what is happening. And the major reason why this is happening is population growth. In the 1800, you can see there in the beginning of the curve that we were not that many, not even a billion people. If you even went down to the times where the Romans were around, there were no more than 100 million people living in the whole planet. So the planet could sustain us in a perfect way. We could live in harmony. We were not overusing resources, and we did not exterminate animals and water and all that. But in the end of the 1800s, with what was a technicality, I mean, the Industrial Revolution, we just exploded. And from that time to now, population has just increased exponentially. And even though there are people that are more optimistic than others that say that this will reach a plateau, I must say that I cannot see that plateau yet, because it's still increasing and has to do with many reasons that we do not have time to discuss today. But that is the case. So we had to deal with the increase of the population. We have to find solutions. I'm not a negative person. I want to, on the other hand, not too blunt for things, because then you cannot solve them. What we have problems, or what we need as humans to live, to exist, the first thing is water. Without water, no life. I mean, not insects, no. Something happened with the presentation?
Your presentation is small. Can you make it bigger?
Yeah. Okay.
It's me that move it like that when I'm talking, and then I put it big when I want to show the results.
Okay. Yeah. Okay.
It's not a normal PowerPoint. Sorry for that. And I'm looking at you guys on the audience, so your reaction. So that's the reason I saw that you were reacting to something strange on the presentation. Anyway, what we need to survive and what life came from is water. A part of water, we need food, and the food needs to be complete. We need different things. We need carbohydrates, we need vitamins, we need fibers, we need a lot of, and depending where you live in the planet, you have different needs. We are adapted, so no one can say this is the perfect plate for you. This is the perfect plate for some kind of default. But when you want to select the food that is really the best for you, you need to think you as individuals, or you as a nation, or you as an ethnic group. Because let's say, I will show you in a moment what I mean by that, because all this needs to be done scientifically and in more precise way. This food needs to be produced. To produce food and to have all the life that we have as we know it, and you may be even more aware of this than many others like us in Europe, we need energy. Without energy, you cannot transport yourself, you cannot make communications work, and you cannot produce food. At the moment, we humans, we are using mostly fossil, I would say, natural resources. And also all of this, if you can see in the curve, started in the 1900s. What are we using? Yeah, we're using coal, we're using oil, we're using gas. When was all this produced? Well, it was produced many years ago when the first type of life that existed, organic life in the planet, that went down to the ground, and it took more than 300 million years to convert all this vegetation to what we today call gas, carbon, and oil that we are using. But you will see in a moment what was produced in 300 million years of Earth history, we have consumed most of it in a few hundred years. And this is amazing. And if you consider the population growth and that everyone wants the same quality of life, we need to start thinking. So what are the issues that all of this is creating? Well, water is becoming more and more scarce, even though some countries have a lot of water, but many countries are having less and less. Deserts are just growing all over the planet, in every continent. Energy is causing the climate changes that we see. That means that ices are melting, that the corals are dying, that a lot of crops are not growing anymore as they used to. Crops, I mean, not only crops, but for instance, wine production in countries like France cannot be kept in the same way as they did in the past, because the climate has changed so much that they're moving up north, and every year more north, the vineyards, to produce in the same way that they did before. We have misused antibiotics in such an extent for humans, but also for production of food, that we have a big problem with antibiotic resistance. Many diseases that were extincted are back in force, and usually multi-drug resistant. Food is becoming a health issue, because we are eating more and more very unhealthy food, and we are eating more and more the same food globally. We are adapted to certain type of food, but now with globalization, we eat all more or less the same, and we get the same preservation elements in the food that are really messing up our microbiota. We have a lot of new emerging zoonotic diseases, and this is not because there are new zoonotic diseases is only due to the fact that human populations are moving more and more in areas where animals never had contact with humans, and they are living there in harmony with viruses, because it took for them a long time to adapt. But as soon as humans enter, the viruses consider that the best habitat, because humans, we are so many and we travel all around. So you all know COVID now. I don't need to explain zoonotic diseases, but this is one of the first in a long queue that we are going to surprise us in the coming years. We also have a lot of augmentation of social diseases, and social diseases means diseases that are due more to how we change our habits than anything else, and I will mention some. So just to mention the water here, I said that water is becoming scarce, and it's becoming scarce because water is not used only for drinking and for agriculture, but also, which is controversial, I mean, is contradictory both. We want to save the planet from climate change, and doing that, we do not want to use more fossils. And one of the solutions we have is to use hydropower. And we use hydropower as this is an example only of things that are going on. We get the issue that in Ethiopia, our friends, they want to have a good life. They need energy. They cannot pay the prices that oil and coal cost, so they are building dams for the Nile River. What happens when they do that? Well, they get a lot of electricity, but the countries that are down on the Nile, they will not get enough with water for their agriculture. It will create very big conflicts in the future, if not already, wars. So we need to think about these things. We need to understand that this will become even more, and we need to be prepared before it happens, not when it's too late. So after thinking about this, the energy, what is interesting here is that energy, the amount of years that we have left with, as I mentioned before, all the fossils that were created by Earth during 300 million years is only 154 more years in the way that we consume today for coal. And for natural gas is 52, 53. And for oil is 50 years. Okay, most of us will not be around, but we have to think about next generations. Maybe if we are clever and create systems that we do not need so much oil, we can have no problems with this. But this is a problem, and that shows that the planet has limited resources to offer us humans, and we use things without taking any considerations. We use that, and that's it until it's ended. We never learn. Climate change. Everyone is talking about the Arctic melting, the Antarctic melting. It has to do, will do a lot of things. Will come with floods, with tsunamis. Many islands will disappear in the world. But still, there are people that see this in a positive way. So nothing has negative or positive. So we need to be aware of this and discuss why it's not only negative. Well, because already today, transportation and global transport of goods and food are much cheaper to transport through the North way, that means through the Arctic, than all the way passing Sri Lanka and then the Suez Canal. It's much longer way, and with petrol prices, is incredible amount of money that they do, that now that the ice is melted and is more free. These are things that are ongoing and will create conflicts and will make changes. This is one of the reasons we need to think about renewable energy in the world. Hydropower is one. I mentioned that can create problems. We can also use a lot of wind, but wind also destroys a lot of habitats. You cannot put winds everywhere. And if you're on an island, you can kill all the the fauna, I mean, all the birds and others, because they crash in these things, and they make noise, it destroy. So it's not always the solution. It's good to have hydropower combined to wind. That means you put wind power supplies in the sea, close to the sea, and then you put these systems that use the waves of the sea to produce energy, not just to dam rivers and do that kind of stuff. Solar power, it should be more, but it's not yet. It's been very expensive. Prices are getting down, but still it's not the solution. So how do we solve all of these issues? I think that we have to see at nice solutions. We have a lot of new technologies. We can use them to really escape all the things that I've been showing you until now, very negative ones, because there are positive things. So what do we do then? Well, we can go to what and our health as much as we think, because we have what is called the food-gut interaction. And that means that we, in our whole system for digestion, we have so many different types of bacteria that those bacteria, if you count the amount of genes that they have around 30 million genes. We humans, we have around 20,000 genes. With all the splice variants, it may be 150,000 variants, but it's not even close to what the microbiome living in us has. So it's much more complex. These guys are the guys that do the job every time you eat. So the connection between you are adapted to certain food, you eat, this is processed by bacteria. The composition of the bacteria have been the product of many, many hundreds of years, sometimes thousands of years of adaptation to certain places. But we travel and we got problems with our stomach, and then gives a terrible, as even they call it that in some places, cocktail of antibiotics. So you get well in your belly. But what you do then, that you destroy the microbiota that was made for you, for your living conditions where you're coming from. So you get a new microbiota. And that can lead to diseases when there's an imbalance, when you go back to your country and you start eating the same food. So it's a lot here that we need to understand. A lot of work has been done. So nowadays it's even known that there are many bacteria that are almost not non-detectable in the microbiota, that are the most important members of the microbiota that you have. It's like saying that you have an orchestra. It's not the people in the trombone, it's not the people playing the violin or playing the cymbal that is the most important, it's the director. So sometimes the directors are so few that when you kill the director, you get an orchestra that will sound, but it will not sound as good as when you have a director. So when you use a lot of antibiotics without knowing what you're doing, you can get a microbiota without director. We need to find out which variants are directors. So when you get this mess, try to get them back. Why is so important? Well, because recently, I mean, all of you that have some medical studies, you know that there is the peripheral nerve system and the central nerve system, but most of you have never heard about the enteric nerve system. It's a new type of nerve system that is around the intestine and around all the organs that have to do with digestion. And this system is directly connected to the brain. It's not going through the other systems. And that means that the bacteria are communicating with our brain. So if you are now kind of getting to sleep, it's not just my fault. It's because your microbiome is telling you that it's working very hard, it needs to rest, and goes directly to your brain as a signal. When you're hungry, it's the same. You get, even some people get very, very hungry when they don't have food, because the microbiome is telling them, we need food, we need to work here. So there is a communication. And if you doubt that, I can tell you that when you are sick, what do? if you have a headache, you take acetyl salicylate, don't you? Or paracetamol. It's a chemical, but that goes to your brain and stop the pain. You can get other medications, chemicals, stop pain because block nerve systems. What do you think all this metabolism, that metabolic products that the bacteria produce do with your body? They do a lot of things. We need to understand that. We know very little about this. Anyway, this is what is going on, and this is why we need to know not only your genetic background, but we need to know what and how does your microbiota looks like to have personalized medicine. You may miss why some people have diabetes, not only because you look at the genes and if there are any variations or SNPs in your patients that have diabetes and the others not, because maybe there is a mess in the microbiota that makes the difference. And this microbiota may cause an epigenetics change in the genome of that person that cannot be seen by looking at SNPs because the nucleotides do not change. It's at another level that the change occurs. So epigenetics was discovered in reality in plants, but later there were many examples. Here I have one example that when a woman smokes, this can go to the baby, and the baby can get another setup of I would say metabolic pathways, and not only the baby in the mother, but also the offsprings can get this. And this was discovered during starvation in when it was the Second World War, and it was in Leningrad that the people were starving, and some mothers got pregnant during that time. And their kids, when they were born, because they were in starvation mode, they can metabolize fat much more and more efficient than any other person. So when the war was ended, these people got children and grandchildren, and they all had the same change in the metabolic pathway for taking up fat. This was discovered a few years ago, but it was the product of a war. Now, these are what were the classical examples. And you may ask, why is Eric telling us this? We live in Sri Lanka, not in Europe. Well, because you may even seen this. I don't know if you have seen this in there, but it's a discussion if dialectical and social things related to colonialism, these famines change the metabolism for the generations to come in many areas of South Asia, which make people in those places to have much more diabetes and also cardiovascular diseases than before, or areas that were not having these famine issues. So there were all these famines in India. There was 84 million people that died because of starvation. They saw in 43 the worst famine, and you may have now all the effects of these epigenetics in many big areas of populations. So when you're looking at SNPs, you may not see this, but if you sequence and look for epigenetic changes, you may see this very clearly. It's just a thought, and I would say that this is collected to all the actually epigenetics effects. So as you can see, this was my talk. I know that you may have some questions. This is how Uppsala looked a week ago. Now we have snow again. So if you have any questions, and I hope that I didn't talk too long from what I was supposed to talk, but I got kind of confused with the cut in the middle. So please, you have problems.
Eric, as usual, very, very interesting talk. I'm sure we have some questions from the audience. any others joining online?
If not, I mean, I hope that the talk was more inspirational. So you go and get all of these ideas and see what the different, because I talk about many fields, and those are fields of research. What is the most important thing is that when you decide for what you want to do, because you need to go in the detail methodologically, but you cannot forget the whole spectrum of things that are outside what you do in your research, and to put that into that context, because that's the only way that we can solve the big issues in the future. And it's how people is doing nowadays with all the collection of big data and then analysis using AI.
Sorry, I have a question.
Yes, carry on.
Yeah, so I was interested in this slide that you showed with the soil microbiota, and I wonder if this is to what extent, how close are we to changing the soil microbiota, like with some sort of probiotics applied to crops, and how would this be applied, and would this be considered organic, and also would it need regulatory approvals? I wonder how far along we are with all those questions.
I think that first of all, we need to understand what is making the difference between having a good soil and a bad soil. And all that work is being done, is very exploratory, but in many places, people know this already. And you can buy already kilos of bacillus of different types that you can put in the roots of your plants. There are plantations for instance, that I visited in Brazil, where they use this bacillus that kills certain type of insects, do not harm the plants, the opposite, and nothing else. With that, the plantation could reduce the use of insecticides by 60%, which is a lot. Then you do not kill all the other good insects that are needed there. So nothing is a 100% solution, but it's always a help to get to the goal, which is to not use insecticides. You have to go step by step. Every time we find a solution, we get another problem. And not always things that look like the solution are the solution. Like the example that I usually mention, when the first humans got to the island of Mauritius, they brought with them rats because the rats jump out the boat. And then people said, "Oh, this is bad because the rats are eating all the eggs of that and the birds that we want to collect." So they put cats to kill the rats. But the cats started eating more birds than rats because they could jump higher and eat those. And then people say, "Oh, then we put dogs." And the dogs kills the cats, kills the rats, but start killing all the other animals. And after a while, with humans' intervention that was so clever, there was almost no fauna in the island of Mauritius. And you all know about the dodo bird. So what I want to say by this is that we usually can find new solutions, but we have to really make very thoroughly tests before jumping into the conclusion that this is a solution, because it could create bigger problems than they were before. It's just my opinion, but
thank you.
Any other questions from audience here, live audience? Maybe something that is relevant to Sri Lanka is this whole issue of organic fertilizer. I don't know, Eric, whether you heard about the government suddenly trying to switch completely to organic. So you were talking about, the precision agriculture, and in that we, as far as I'm aware, we actually do give the nutrients in the form of the chemicals, right?
Yeah.
yeah, how does this kind of thing apply there?
I don't want to interfere with local politics, but as I understand, the issue is kind of delicate because sometimes you, as I said, you put cats to kill the rats, and then you get bigger problems. So what happens usually when you do things very, very rapidly is that you can get into trouble. I think that the idea to use natural fertilizers and organic is a good idea, but you need to really know how things look like in the country and place that you're doing that, because it's not the same everywhere in the planet. Some places need a lot of certain chemicals extra, and some not. I mean, in the jungle, for instance, in Brazil, things grow so incredible well, but when you cut that and you try to put any kind of crops, they grow once and then they don't grow anymore. Why? Because the only thing that was keeping all the life in those jungles were the different levels of plants that were helping each other and having cooperation with each other. So when you just go for monoculture, you deplete the land immediately because the soil in those places were not that good. But only because it's growing very well doesn't mean that it will grow well forever, because it's nature that is so clever that put things together. So that's what I mentioned in one of my slides, that you need to find the perfect combination of plantation to put together, and then you get a really good organic growth. But it's difficult, and it's not always from the very beginning that will give you the best harvest.
Thank you. How are we for time?
I guess we are past the time.
Yeah, so I think maybe if there are no further questions, let us thank Eric as usual for kind of broadening our horizons in the application of bioinformatics and also for the multidisciplinary kind of approach he has, which is always very, very unusual, especially in our parts of the world, and very refreshing. Thank you very much, Eric, and for getting up early, I guess, I'm not sure, yeah, and hanging on for, even if our times were not matching quite well. Thank you very much.
Thank you a lot for inviting me. It's always a pleasure. And I must mention again, I see such an incredible progress and a lot of more and new enthusiastic people. I'm really happy to every time to see that. That is better and better. So congratulations to you too.
My presentation is very much interdisciplinary in nature with majority of work, I touch here my own work or work I collaborated. And can you hear me? Hello, we can hear. Okay, yeah. And also, like I was trying to come in person to deliver this lecture. Unfortunately, the traffics and number of other reasons I could not make it to Colombo, and I'm got stuck in Gaul. I'm trying to assemble as the best possible to give this seminar. And the title of the talk is Biofortification of Cereals with Zinc. And I also explore the opportunity to improve zinc loading into the grain. My talk mainly focus on the zinc loading into the grain. And a lot of people not aware of what is the biofortification, and also fortification is the fortification we discuss in many areas, and like food fortification as mainly defined by WHO and FAO is the deliberate addition of essential micronutrients such as vitamin and minerals to food in order to improve the nutritive quality of the cereals and pulses and food, etc. It can be done by food manufacturers or by government agencies as a public health policy, aiming to reduce the number of people suffering from dietary deficiency in those nutrients. That has been already identified as the hidden hunger, is globally recognized as one of the biggest problem at this stage. Most often fortified foods include cereals and cereal-based food, milk, dietary products, and fats and oil and snacks, so on. So goes on. But the lot of people talk about the fortification, addition of micro or macronutrient into the food. But my talk mainly focus on the biofortification, that means genetic or breeding intervention and a mechanistic understanding to intervene and improve the grain micronutrient status, and especially focusing on the zinc because the area is too big. I want to more focus on the one nutrient and go into the mechanistic side, and then move into the more biofortification, the breeding, etc. And the micronutrient deficiency in the world, as I said, is one of the biggest problem. And 6 billion people is suffering from micronutrient deficiency. The micronutrients, etc., like iron, zinc, iodine, vitamin, it goes on. But if among them is iron deficiency is the biggest problem in the world, and then next to iron deficiency, zinc deficiency is the biggest problem. Nearly 2 billion people suffering from zinc deficiency. And there are a number of physiological and metabolically related issues related to the human health, and is some of the things identified. You can see in the slide that the reduced growth, reduced immunity, loss of appetite, weight losses, and the number of sterility and etc. There's many metabolic related disease are linked to the zinc deficiency, and therefore it is essential to really to address this issue. And when come to the 1/3 of the world population from zinc deficiency are going through diseases or disorders, and the zinc deficiency is responsible for 0.8 million child death annually. This is nobody knows these numbers. Actually, this is very predominant in this part of the world. And zinc deficiency is acute is a rice-eating countries, like specifically in Asia. These are the major reason that why we really need to address this issue. And to address this issue, we really need to know the fundamental behind the disease development. And as said, zinc involves number of metabolic related disease, like zinc is the second most abundant micronutrient in the human body, and which is a co-factor of 300 enzymes, help to build health, immune system, maintain sense of smells and taste, and so on. It goes on, and involved in protein and DNA synthesis. Essential for growth of body tissues, blood clotting, wound healing, and so on. And so, as said, the large, nearly 70% of zinc human intake coming through cereals and pulses. The depletion of cereal and the zinc in this food system is the biggest culprit for the development of zinc deficiency globally. And I mainly work on these two crops, the wheat and rice. And again, this table published by the Nutrient Data Library, United States Department of Agriculture. And you can clearly see the grain chemistry of rice and wheat and the macro and micronutrient composition. And this is from large collection. And I highlighted few important the micronutrient component that we closely looking at. And the protein is also important. Like if you see the rice compared to the wheat is nearly half of the protein in rice. And the iron is again 3 times lower in rice compared to wheat. And zinc concentration also nearly 3 times lower in rice compared to wheat. Selenium, it is again like 4-5 times.
You're muted.
Just give me a minute. Okay, I'll reconnect again. Oh, jeez. Is it all right now?
Yeah, it's okay.
Okay. And sorry for the interruption. And so again, this is a 2 comparison, and this is you can see the fundamental problem what we have today here. It's not for us; it's a whole subcontinent issue. And what happened after Green Revolution? So right now we eat our rice is a very high yielding rice, and the harvest index is nearly 1. Like a total biomass, shoot biomass to grain biomass ratio is nearly 1. And because during the breeding process, you can see this graph, and we improve the more we got, the more carbon we got into the grain than the shoot, and we develop a very efficient system. And we achieved this goal because of the population pressure we had. We had nearly 4 billion people in 50 years back. Now we have 7 billion on the earth, and it's going to be 9 billion another by 2050. So this is the fundamental thing, like we need to feed the population. What International Rice Research Institute and also other rice researchers globe, what they did is after Green Revolution, we improved the rice yield potential. Those are the key innovative science went into the breeding programs. And one is a dwarfing the technology. So when they got the dwarfing gene into the new breeding programs and develop a disease and insect resistant varieties, and also we address the grain quality aspect, some of the quality aspect, and we brought later on the abiotic stress resistance, and now recently we got the hybrid the technology to improve the yield potential like that. So now our yield start to plateau, we cannot push any further. However, and the population is growing, we really need to work around here to get our yield up as much as possible while maintaining the quality characteristics. And for example, I will take this this experiment done in Rotham State Agriculture Experimental Station, and there's one of the oldest agriculture research institute in the UK, and they had a huge archive of grain sample, 200 years old, and you can clearly see how the grain yield changed. This jump happened after the Green Revolution. However, so what the micronutrients, if you see, for example, zinc, for example, iron and copper, and also manganese, we can clearly see the same time the micronutrients, especially iron, zinc, and all, that gradually, significantly decrease. And so this is the very recently published this work in a experimental medicine and biology journal. So further, when we look at our soil in the whole world, the MAPO zinc, there are some areas the zinc deficiency is predominant. For example, in Asia there is a zinc deficient, and also Africa, and there is a large heterogeneity of zinc distribution in the world. So this also influenced the zinc concentration in the grain and pulses, and we recently published this work in Nature, and the climate change is one of the big culprit that reduce the grain ionome. The whole micronutrient composition alters, especially the CO2. When the CO2 concentration increases in the atmosphere, this nutrient, especially zinc, iron, protein, in a wheat and rice and pea, except C4 crops, all of the cereals and pulses is substantially decreased. So this is a kind of a multiple factors influence the accumulation of grain and micronutrients, especially I'm talking about zinc and iron and protein, etc. And in summary, what I want to say is that the micronutrient deficiency in human is very much due to the plant breeding for high yield potential after the Green Revolution was one of the factor that contribute to reduce the grain micronutrients. And again, this 1 and 2 is interlinked, is the extensive use of modern rice varieties. It also is responsible, and the soil, when you take the soil, in the global heterogeneity of the zinc distribution is another factor contribute for the lowering the micronutrient, especially zinc, in our food system. And then finally, so we were the first to report this story that rising CO2 dilute the micronutrient in the grain, especially zinc and iron. We published this work both in Nature and Science very recently. And if we want to maintain a healthy diet, especially the micronutrients, the iron concentration should be around 4 to or 48 milligram per kilogram of rice. The zinc concentration should be 60 to 72 milligram of rice. So at this stage, the concentration this is actually World Food Program recommended the recommendations. At this stage, these are much lower in our rice and wheat system. So that's why we really need to address this issue immediately rather than delaying this issue. Again, like this is the pathway to address the micronutrients in the grain. This is how to improve the increase. And I don't want to go into the all other details. I'm mainly working on the pathway of biofortification, genetic biofortification, and conventional breeding, and other related parameters. And also we cannot ignore the fundamental mechanism of how this nutrient taken up and move into the shoot and take into the grain is very essential. That is, there is a huge knowledge gap in that space, and we also work on that space to establish the fundamental mechanism to get this nutrient back into the grain in the endosperm. And so this is we need to know about the soil chemistry. We, as I said before, the nutrient uptake and transport mechanism, and how they transport into the grain, and the grain physical and anatomical characteristics is required to fully understand to improve the grain micronutrient density in the future. And biofortification, as if you take directly, this is a biofortification is an idea of breeding crops to improve nutritional value, as I highlighted before. It can be accomplished using a conventional selective breeding or both genetic engineering. And the biofortification is distinct from conventional fortification, is that focus on increase the nutrition value of plant food with the plants, or when plants are growing, and the biofortification is viewed as the most feasible approach to address the global malnutrition of zinc and iron deficiency. And some of our data, again, this is the collection of wheat and Sri Lankan rice germplasm we put into test. And what we see is that in the figure, the graph 1, you can see this is a global collection of genetic diverse collection of wheat cultivars and wheat grain samples. We look at, there is a large variation in grain zinc concentration, which is nearly compared to the lowest, it's 100% the zinc can be seen in some of the cultivars we identify. It's similar to the wheat. The rice also said there is a large genetic variability in zinc concentration, and it is very unique to Sri Lankan germplasm, because I have looked at and tested and seen in other literature, this kind of variability is very rare. What we learn is from this, that we can take some of the genetics, what we have for the biofortification breeding in the future. At the same time, the mechanism is everything. Without knowing mechanism, we cannot address this issue. So once the zinc taken up from the soil, and it take through the root system and transfer into the xylem and the phloem, and so it's also the accumulate in the source tissues, and for example leaf and shoot and etc. And then the grain, finally these nutrients, especially the zinc and iron or the micronutrient, transfer into the seeds. And this is where we identify this is the bottleneck for nutrient, to increase the micronutrient density in a rice or wheat grain. And when we follow the pathway, the key genetic component associated with the zinc acquisition, and those are the key genes, especially the ZIP1 and ZIP3, and zinc uptake, and the zinc transport, some osnus, NAS gene, and also these genes are involved in zinc transport, and osnus 2 and ZIP8 and ZIP4 like genes are involved in the loading into the grain. These are we characterize this gene, and we have seen consistently and play a very important role in a zinc acquisition and transport and loading into the grain. And other important factor is that zinc, the uptake and transport and loading, determined and facilitated by complexes. There is a complex with zinc, some amino acids, and for example, nicotinamide and histidine and cysteine, and these are some of the amino acids complex with the help of transport. And if you take the Na, play a key role in zinc transport. And also the chelating agents like the phytochemicals and deoxymutagenic acid, they also play a major role in the nutrient transport across the roots and the shoot and to the grain. And there are other malate and citrate and small peptides, and phytic acid also play a negative role in a nutrient transport, especially zinc and iron transport. So as I said before, the Na is the dominant zinc binding ligand in the cytosol and xylem and phloem, and the functionality is play an important role in nutrient loading into the grain. And this is a typical the wheat grain. In fact, this graph, and we stain with the DTZ, and we can clearly, for zinc, we can clearly see this is an embryonic area, and also the aileron area. You can clearly see that the redness is when we stain with the DTZ, the color change to red, and it's a very high density. Zinc is very much located here, but when you cross the cross section, even if you look at, we don't see inside there is not much zinc accumulated. Basically, most of the zinc and micronutrient located in a aileron, outer aileron layer, does not move into the inner endosperm. And this is some of the work we did. Actually, we have published, submitted to Nature Plants this work. I thought of sharing with you. And these are the cross section of wheat grain. And you can clearly see the middle part. If this is the crease area, and the genes associated with the zinc loading into the grain, those are the genes. And what we noticed, what we observe is that genes located in the crease region are highly expressed, especially here, the YSL3 and YSL6 and ZIP genes are highly expressed. And this is the first time we document this story. And this is a very promising data. And for the CRISPR-Cas9 is like tools can be used to edit some of the genes to get the high expression in this region, and we are working on right now, and to get the more zinc, more iron into the inner endosperm. I think the technology, once we establish this technology, can be applied to rice and other cereal crops as well. And this work is submitted to Nature Plants. And further, and we were backed by this information by X-ray synchrotron data, and this is the unique facility. It's located in Australia, and so what we have done is we have mapped whole grain ironome, and this is a kind of this is again wheat grain, and we can see the zinc is it's a green color, and red color is a zinc, and manganese is a green color, and phosphorus is a blue color, and so we can clearly see how the distribution of these nutrients. And again, we identify from this data, from this analysis, this is the shoot root primordia, and this is a shoot primordia. It's a very unique. We can identify the shoot, root, primordia, and many of organelles through the nutrient, the distribution pattern. So this is a very unique, and at the same time, when you look at this area, this is, we think, very unique, play a most important role into the zinc and iron loading into the grain. And we identify some of the genes located here, as I said, explained before, are differentially expressed, play a key role, highly expressed, and play a key role in uploading, moving zinc and iron from outside into the grain inner endosperm. This work also published. And one more thing I want to highlight is that across the membrane, this crease membrane, we can clearly see a zinc and iron accumulation here, and across this section. And these two different varieties. One is a gold mark, is a zinc inefficient variety. This is a zinc efficient variety. And inefficient variety accumulate more zinc in an outer crease area compared to the zinc efficient variety PBW43. This is also very important because this tells us there is a large genetic component and in zinc transferring into the inner endosperm. And I quickly go into the next slide because time is running out. And we also work on rice to biofortificate, to improve the iron. We want to get iron into rice. This is again electron microscopy, scan electron microscopy picture, and this is the husk, and this is a inner endosperm region. And what we see when we have a cross section, we can see large network of pores. So we thought we can capitalize this pore structure to improve the micronutrient through this pore structures. And here you can see, and what we see inside the grain, and it is all the pores are very much connected to the crease area. And this fundamental configuration of starch granules create new opportunity for physical micronutrient fortification in the future. And some of the work, one of our students' work here, we can see here that the iron vacuum drying with various solvent, and this work, like getting closer to the patent, and we can use that vacuum techniques and solvent-based vacuuming to improve the micronutrient in the grain in the endosperm that can facilitate to the inner pore system. That is, I think, kind of first time we have discovered that the structures. And overall, through this process, so we want to have the iron concentration somewhere 40 to 48 milligram per kilogram, right? If you take as rice flour, and through this process, we were able to these various treatments, and in fact, we were able to get into 60 ppm, 60 milligram per kilogram kilogram iron in a rice flour. This is, I think, is one of the major breakthrough. And finally, what we are working on, we are also working on the chemifortification. We are also main emphasis on the biofortification and genetics. So using high throughput phenotyping and genotyping and gene mapping and transcriptomic, using transcriptomic tool and proteomics, metabolomics, and all bioinformatics integrating, we are working, identifying the traits and genes and QTLs to get to improve the zinc concentration in a wheat and rice system. And in the same time, we are working on the genetic engineering. This is, in fact, we are working on the CRISPR-Cas9. That is not considered as a GM crop. If we are successful, we should be able to get the genetically CRISPR-Cas9 engineered rice and wheat in the near future with a high density of micronutrient. And in summary, I want to say that the presence of significant genetic diversity in grain zinc content in the whole spectrum wheat and rice we have seen, and this is a good tools to improve the grain micronutrients in the future. And we also have established molecular mechanism and identified the key bottleneck of how nutrient load into the grain, and that can be really utilized for the breeding as well as the genetic engineering to get the more micronutrient, more zinc into the inner grain and the sperm. And we also have seen the crease size and shape and of the structure, the anatomical features. This can be modified through understanding genetic variations, and we have seen some cultivars and varieties have a large crease area that tend to accumulate more micronutrients, and those can be utilized to improve the grain micronutrient density. And basically what we want to say is our research paved the way to address the worldwide micronutrient deficiency. If we have a really good funding, we should be tackle this issue without delay. This is my team, and a team is David Patterson from Australian Synchrotron. He's a good collaborator, being long time, and Professor Hiroshi Naoki is from Toyo University, and Chandima is one of my PhD students, and Professor Peter Coviti from Queensland University. And I don't want to go all the detail. And also I want to say Heli Seniviratna, she's based in Sri Lanka, and she also working with me, PhD student. And Achini is more into the modeling part of it, Professor Achini, and Dr. Paul Miller, and again, University of Western Sydney, and Professor Robert Norton, and also Melbourne, from the University of Melbourne, Niluka Nakandala from Rona, and Niluka was a PhD student of mine as well. And also I want to thank a lot of work done outside Sri Lanka and from Australia, Japan, US. My work was supported through various funding sources. I really want to thank, and also I want to thanks all my collaborators, and I wouldn't take a lot of time. Thank you very much. If you have any question, you can ask me. Hello.
Yes, thank you, Professor Senavira, for that presentation on a timely topic. So now we can move on to the Q&A sessions. There are any questions from the audience, the live audience, or those who are joining online? I'm sure Professor Senavira will be pleased to take some questions. While the audience is trying to, the questions come from the audience. Professor Senavira, just one question really is, now you were talking a lot about the genetic diversity. So in Sri Lanka, if you consider rice, and of course iron deficiency is a big problem. So have you done any screening on the germplasm of rice for the ability to fortify iron in our rice varieties? Is there a difference?
Yes. Yes, like I have done, I think I have biggest collection of the, but still the work is progressing, and the progressing in the sense that the publishing process, and yeah, we screen a large population, and I haven't seen that diversity in anywhere else in the world, and there is a huge diversity, and iron and zinc, and that's what I say, that we can use some of this our germplasm for future breeding program, even in the international level.
That's very promising. Then also another thought that came to mind was, now we have iron soils with a lot of iron, and some of the rice breeding stations are breeding for toxicity tolerance, iron toxicity tolerance. So is there any way of converting this to, sort of utilizing this excess iron in the soils for biofortification of the rice?
Actually, like we have not looked at the iron toxicity. We have taken a separate issue, and we have, because it's a very localized, not a bigger area, and but you know, again, that has I am sure that when they accumulate more iron, and the iron toxicity itself is another big problem here. Is it reduce the growth and impact? There are a lot of other issues for the suppress the growth, and things yield are substantially reduced, and also associated with the soil toxic, like acidity, etc. Most of the iron toxic soils are acidic, and this is very much the problem in that is a problem soil space, and perhaps we can learn something from there, some fundamental the way how those nutrients taken up, load and loading into the grain, etc. And there may be good traits as well in some of the germplasm they survive in this environment. I haven't done anything that with them.
Are there any other questions for Professor Senavira? Okay, in that case, we would like to thank you very much for that very informative presentation, Professor Senavira. Thank you very much for joining us today.
Thank you. Thank you for the opportunity, and I really appreciate. Okay, have a wonderful meeting. Yeah.
So we will move to the second speaker of this session of Plant Genetic Resources and Biotechnology. She will talk about the genetic and genomic and genetic tools for breeding in food crops. Our invited guest is the Dr. Michela Trojo from Fondazione Edmund Mark, San Michele all'Adige, Italy. She's the head of the Fruit Crop Genomics and Breeding Research Unit. So it's a great pleasure to read her citation and to invite her talk. Dr. Michela Trojo obtained her PhD in ecology in 1999 from University of Parma, Italy. After earning her PhD, she completed postdoctoral research in conifer comparative mapping at the University of California of Davis, US. In 2003, she received a postdoc fellowship at the Institute of Agraria di San Michele alla Dice, later renamed as Fondazione Edmund Mark, to work on developing genetic resources in grape. Since 2008, she became a permanent research staff of the center. Her research is focused on comparative genomics among sequenced plant genomes and on developing of innovative strategies and genetic tools for associating specific alleles for phenotypes of interest. She has been a co-principal investigator for 2 Marie Curie IRSES projects funded in FP7. She's the team leader of the Fruit Breedomics project and principal investigator of 2 crowdfunding scientific research. She's the author of 83 referred ISI web publications, and her EDGE index is 38. And she's the member of editorial board of scientific journals. So I'm glad to invite Dr. Michela Trojo to deliver her talk.
Hello to everyone. Welcome to this talk. My name is Michela Trojo, and I am the head of the Fruit Crop Genomics and Breeding at Edward Mark Foundation in Italy. Let me first thank the organizer of the meeting, my former colleague Nadisha Bandara, for giving me the opportunity to talk about the research work in the genetic and genomics of fruit trees we are doing in my research institute at FREM. In case you are wondering where Foundation Edmund Mark is, we are over here in a place called San Michele all'Adige, in the north of Italy, surrounded by beautiful mountain, about nearly 5,000 miles from you. The region has the largest area covered in apple orchards in all of Italy. More than 50% of the apple produced in Italy annually are cultivated here, at the foot of the Italian Dolomites. Thanks to the fresh climate and the strong temperature variation between day and night, Trentino apples are some of the most sought after apples in both Italian and international markets. Edmund Mark Foundation is a non-profit organization with private legal status founded in 1874 on a German model which brings teaching with research, training, experimentation, consultancy, and services in the agriculture, environmental, and agri-food sector. Our resource center focuses on basic and applied research on strategic supply chains of the Trentino agrosystem, forest and alpine ecology, biodiversity evolution and conservation, effect of climate change on natural and agro ecosystem, bioeconomy, and agro biotechnology. The center has 21 technological platforms that are operated by highly qualified personnel. Each platform has a manager who is responsible for coordinating all laboratory procedure, either as services or research collaboration. Here's just an example of some of the platforms like the taxonomic, the sequencing and genotyping, plant phenotyping, plant tissue culture, volatile analysis, and many others. The center also benefits from 3 germplasm banks, the grapevine collection, which include about 3,500 accessions belonging to different species of the genus Vitis, about 1,000 accessions within the genus Malus, and 460 accessions for the berries germplasm. We do also host an active PhD program in collaboration with prestigious laboratories in Italy and abroad. More than 50 universities and private companies have collaborated with us since 2010, and we hosted more than 200 PhD students from over 100 countries. We are very open to collaboration and to all students from all over the world. So we are very happy to collaborate with the university as well. After this introduction, in my talk today, I will give you a brief description of the state of the art of genomics in fruit trees. I will show you a few successful examples of their application, and I will conclude with future perspectives. The sequence of the first plant genome, Arabidopsis, was completed and published at the end of 2000. As you can see from this slide, for the next 10 years, very few genomes were published. With the advent of next generation sequencing technologies and the reduction in sequencing cost, hundreds of genomes have then assembled and published. The time frame in the previous slide covers 10 years. In this slide, 1 year only. A representation of the increased sequencing efforts of the last few years can also be summarized in this slide that shows the plant genome sequence during the last 12 months only. Sooner, every plant and animal genome species will have their genome sequence. At my center, we have been involved in many sequencing projects, which started with the sequencing of the first heterozygous genome, the cultivated ripened Pinot Noir, in 2007, followed by a more challenging genome assembling of a highly heterozygous but also recently duplicated genome of the cultivated apple, Golden Delicious, which was done in 2010. Since then, we participated to the sequencing of the pear, strawberry, raspberry, walnut, and more recently, the lemon genome. The advent of third generation long-read sequencing has further enabled complex genome to be sequenced and assembled at the chromosome scale level. We are also moving toward the genome of the pangenome of the species Malus domestica, with hundreds of cultivars sequenced at high coverage, but also toward the pangenome of the Malus genus, with a number of wild species sequenced and de novo assembled. Assembling highly heterozygous genome is still a big scientific challenge. Assembling a genome which is both highly heterozygous and also recently duplicated into an haploid consensus sequence presents a great challenge, since the problem of heterozygosity and paralogy compound one another. To improve the diploid gene of apple and pear, we use the two double haploid produced at INRA in France. Both double haploid were found as an haploid plant that has been duplicated in vitro. The great difficulty here is presented by the problem of distinguishing the slight differences between the homologous chromosome from the slight differences between paralogous genomic loci. To perform the de novo assembly of apple double haploid genome, we combine three different technologies: short read sequencing, long read sequencing, and optical mapping. Compared to the previous assembly of the diploid Golden Delicious, we arrive at estimated genome size for two reasons. First, we were able to consider all bubble structures to result from differences between paralogous loci instead of differences between the homologous chromosome. Second, with the use of long reads, we were able to collapse imperfect repeats instead of assembling them in separated contigs. Contigs were then scaffolded using Illumina-made pair reads, and this a chromosome scale assembly was obtained using the binano optical map and a high-density genetic map. The results from Busco analysis indicate that our apple genome annotation is the most complete, despite having the lowest number of predicted genes. Busco analysis provides quantitative measure for the assessment of genome assembly, gene set, and transcriptome completeness, which is based on evolutionary informed expectation of gene content from near universal single copy orthologs selected from the orthologous database. Similarly to the Apple genome assembly, the paired double haploid genome assembly workflow began with the de novo assembly of content from the PacBio long reads using CANU. The Illumina data instead was assembled on its own using the Bruegin graph-based assembler SOAP de novo 2. This assembly was used in various ways during the course of the paired genome project for further scaffold validation and for training the ab initio gene predictors. Assembled content was further joined into scaffold using a combination of binano optical mapping data, high-C chromatin conformation capture data, and genetic maps. Same for the Apple double haploid, avoiding the over-assembly that is due to the separation of the haplotype for highly heterozygous region, we obtain a more accurate estimation of the gene model. We estimated the genome size of the Apple double haploid to be 650 million bases, which suggested that the double haploid genome may be smaller than the heterozygous golden delicious line, which was recently estimated to be 700 million. However, the double haploid tree looks similar to the heterozygous golden delicious and include the similar tree architecture, flowering time, and fruit appearance. It is possible that through the consecutive step of selfing, haploid development and chromosome doubling, some minor parts of the genome may have been lost or rearranged. Thus, it's possible that some of the genome sequence might be missing in the double haploid assembly. We are currently investigating this point, and we started a new project in order to answer to this question. A big challenge when assembling a highly heterozygous genome is to separate the two haplotypes. We recently managed to assemble the genome of citrus into two distinct haplotypes using Illumina short-read, Oxford nanopore, and the Illumina resequencing data from the maternal grandparents and paternal genomes. Mapping of the content to the pedigree in the identification of the most similar help to assemble the genome into a primary and alternative sequence. Now, going behind the reference genome, one of the main challenges in agricultural genetics is to assess and use the tremendous genetic variation present in germplasm collection and in the wild. Now that we have a reference genome for every species of interest, and with the reduction in cost of Illumina short-read sequencing, it's becoming economically feasible to resequence many accessions and discover the genetic variability, so point mutation, insertion deletion, transversion, copy number variation. But it is not yet computationally feasible to apply to a high number of individuals. Next generation sequence technology has proven to be exceptional tools for discovery, validation, and assessment of genetic markers. And thus, this has facilitated the development of genome-wise marker and array-based genotyping platform. Many methods are now available for genome-wide genotyping. So there are array-based genotyping methods that require a prior genotypic knowledge. They can provide different level of multiplexing and rapid high-density genome scan, a robust allele calling with high core rate. They are very reproducible. There is a low cost per data point. However, we have to consider a design bias and no flexibility of the platform. Reduced representational sequencing instead does not require a prior genomic knowledge. genotyping are done at the same time. This method reduces the proportion of the genome targeted sequencing so that each marker can be sequenced by a coverage with limited resources. The main advantages of this method are there is no discovery bias, they are flexible, and the costs are reduced. However, they hold a number of genotyping errors due to allele dropouts and missing data. And there is also a labor-intensive library preparation and the higher complexity in data analysis. From now on, I will discuss the use of SNP array that we have developed in collaboration with different groups based on both Illumina Infinium and Affymetrix Axion technology, starting from short reads Illumina sequencing data from many section of cultivar within each species. Each array development started with a common pipeline that goes from the alignment of the sequencing data to the reference genome, SNP calling, and the filtering of the most reliable SNPs. But the choice of the sequencing panel, which means the number of diverse sections, and the density of the array was specific to each project goal. In this slide, I'm just listing a number of arrays that we have developed for apple, pear, walnut, peach, and many other species. To develop a high-resolution linkage map for QTL and pedigree-based mapping, a low-density SNP array is sufficient, but the sequencing panel should represent the parents of or the major founders of the specific breeding programs. For genome-wide genotyping of germplasm collection for genome-wide association study, the density of the array depends on the linkage disequilibrium. The decay of the linkage equilibrium has been shown to dramatically between species, and this is often due to differences in breeding system. Selfing reduces opportunity for recombination. In outcrosser, linkage equilibrium generally breaks down more rapidly. Outbreeding species generally exhibit limited linkage equilibrium, and consequently, a very large number of markers are required for effective whole genome association genetic scan. The array can be used for validation of the breeding material as true to type and to detect polyploid and aneuploid, for genomic selection and pedigree reconstruction for pedigree breeding by design. In these cases, a low density array is sufficient, but an informative set of SNP should be included in the array that usually derive from a previous effort of genotyping. For apple, the Illumina 20K SNP array was used to build an accurate integrated linkage map based on 21 segregating population and a total of 1,600 individuals. An innovative mapping approach was used that considered both a focal point strategy and the background strategies plus data integration across families prior to mapping construction. This approach has led to the mapping of 15,000 SNP with a very precise mapping position. This map was successfully used to anchor the apple genome, but also to understand the pattern of linkage equilibrium and recombination frequencies, which is the basis for plant breeding. In this slide, you can see a graphical representation of the location of SNP marker on the physical map on the x-axis, compared to their position on the integrated genetic map on the y-axis for all chromosomes. Each marker is plotted in the color of the chromosome to which it has been genetically mapped to. This analysis is showing that there were very little discrepancy between the physical and the genetic map, with more than 95% of the map marker that were found to be located at their respective positions. The array was also used for fine mapping of apples carb-resistant genes and to develop markers to assist traditional breeding. As you can see in this slide, a number of resistant gene loci has been genetically mapped and are currently used for marker-assisted breeding. For two of these loci, we identified the two major candidate-resistant genes that have been cloned and used for transformation and are currently in validation. For both loci, we have developed and screened two back libraries obtained for the genotype that brings the resistance, and we have sequenced a few backs using PacBio sequencing. This is the work that also Nadisha Bandara has carried out during her PhD with us. Nowadays, with a reduction in sequencing costs, it is not necessary to develop expensive back libraries to screen and fine map resistant genes, but the recombinant individual can be selected and directly sequenced to identify the resistance allele. In apple, linkage disequilibrium decays quite fast, being an outbreeding species that generally exhibit limited disequilibrium, and consequently, a very large number of markers are required for effective whole genome association genetic scans. We therefore decided to design a much denser chip, the Affinities 487 SNP array, which also cover a very large genetic diversity, which encompass European and Russian germoplasm and includes some Iranian, Tunisian, and US cultivar. We implemented a large genotype genome-wide association study at the European level using an association panel of more than 1,100 different apple genotypes distributed over 6 locations and phenotype for a number of phenological traits. Highly significant genomic regions were identified on the top of chromosome 9 for the flowering period and on chromosome 3, 10, and 16 for the ripening period, consistently with already published studies. We are now analyzing other important breeding traits. Reconstruction of pedigrees is also a fundamental importance in breeding program. Apple cultivars are interrelated by recent and distant common ancestors. Many old cultivars still exist in germplasm collection, as SNP arrays have been used to identify pedigree relationship with high precision. This finding has been successfully implemented in breeding program studies. For apple, more than 800 apple cultivars and breeding selection were SNP genotype using the 20K SNP array. The data allow validation of historic pedigree records, tracing parentages of over 50 individuals for which pedigree records were lacking or false. This included major founding cultivar worldwide, for instance, Delicious, as well as ancestor from cultivar of specific interest, for instance, Enterprise and Antonovka. An updated Onecris pedigree showing inheritance of undesirable soft scaled allele at linkage group 2 QTL is represented in this slide. The use of marker information pedigrees increases the power of pedigree-based analysis-based QTL discovery. This also facilitates breeder when composing germplasm set for pedigree-based studies. This information is also very useful in clarifying the origin and mode of action of discover QTL alleles and to extrapolate the QTL result to other genetically linked individuals, and therefore to move toward a breeding by design approach. Now, I'm moving to the conclusion. As you have seen from my talk, our current understanding of the genetic diversity is based on a limited and largely domesticated gene pool, while it's limited within the wild compartment. We therefore need to bring new traits associated with resilience, adaptability to climate change, resistant pests and disease, and new interesting quality traits, for instance, associated with aroma. All these traits are brought from the wild accession and species. For this reason, we have to move from a single genome toward a consensus genome per genus, with the full genome sequencing of wild species and wild accession relevant for integration and breeding. This is the concept of the pangenome. And the concept of a single genome for a species towards that of the consensus genome per genus, whereby the genome of the entire species can be captured and variation among the different accessions properly annotated, including genomic specific loci that might be absent in the reference domesticated genome. This information will be fundamental to assist breeders to incorporate wild-related material into the cultivated gene pool. We need also more understanding of the epigenetic variation to expand the material for prime breeding and for a correct germplasm management. I have a long list of colleagues to thank for all this work that I listed here, but I would like to have a special thanks for my colleague Luca Bianco, an excellent bioinformatician who performed a lot of the work I presented here today. And thank you all for the attention.
Thank you, Michela, for the very informative talk about the genome sequence of tree crops. So the session is now open to the question and answering. The audience, the on-site and the online, if you have any questions, you can forward to Michela. Okay, Michela, I would like to raise a small question from you. Since you have talked about the apple genome sequencing and then the citrus, I know so many land races and the varieties are available in the apple and then the citrus, as well as all the other species that you have built. So if you want to select one variety as to start with the sequencing, what is the basis that what are the things that you need to consider?
Sorry, Nadisha, can you just repeat a few words, because I don't know, I could not hear very well. Sorry. You mean you asked me if I want to sequence a variety where I should start with? No.
Yes,
sorry.
Yes. If you want to start a sequencing plant species having so many varieties, how do you select which variety or which wild type that you need to pick?
Yes, this is what actually the pedigree reconstruction is what is going to help to do that, because of course if the focus of the sequencing is to using a breeding program, what we should start is from the major founder of the pedigrees. This is actually the reason why we have chosen golden delicious in our first sequencing effort. But now we are going to sequence also sequence at high coverage and de novo assembly, which is feasible now. We are going to do that for the parental of our breeding program. This will allow you to start with a breeding by design approach and while reducing then the number of cross in the future. Of course, we are working with an outcross species, which and is not self is self auto incompatible. So we need to find some strategies to reduce the breeding effort in this case. And also among the wild species, we have been selecting a number of species that has been shown to be the source of domestication. For instance, Malus silversiae, Malus silvestris, Malus micromalus, and this species has been, we are planning to sequence, to do a de novo assembly to avoid the bias of a reference genome, because by assembling a genome using a reference-based method, you will lose information that probably come from the wild species, which is the most useful now for us. You know, at this moment, I think probably in the next few years, our aim is to sequence all the parental of our breeding program.
Okay, thank you, Michaela. Are there any questions to ask from Michaela?
I have a question.
Hi. Yes, go ahead.
Hi. Incredible job you are doing. I mean, I really love this kind of enormous amount of data that you fall down from your chair. I do understand how much it is, but at the same time, I have a question about. I remember when I was a kid, there were so many apple variants. They have all disappeared from the market. Now you have like 5, maybe 6 that are sold. When you are doing now the genome of these variants, do you only select the ones that are commercially most viable today, or all these incredible variants? I remember some that have the taste of banana, some that were the taste of citrus, and some that were this size. I've never seen that now.
Yeah, no, you're right.
And we save them.
Yeah, no, exactly. Right now, so the breeding has brought as really one of the limitations during the last breeding program was to reduce the variability and to only use a few sections for breeding. This is what we have also now in our breeding program based on our previous breeders, because the idea is to select the same parents because you are sure they bring some quality parameter. But now what we must do is to go back to the wild. Of course, not to the real wild, because going into a breeding program like Nepal, it takes 20 years. So we need to find some already high quality variety, but that bring new traits. For instance, aroma has been lost, and do I agree with you? So in our breeding program now is to reduce the number of parental which come from the same cultivar, and to bring new variety into this. So this is of course what is necessary. In the past, the gene pool has been reduced, and now we need to increase it a bit again. But with the knowledge of the genome, it's possible to select already among a number of sections that are bred yearly, to select those that have a limited, so they bring new trait, but as a limited genome from the wild. So what we are also developing, this is why we need a reference, like you say, pan-genome for the wild species, because we can select the region that comes from the wild, and we can then do a negative selection into our seedlings. So just keep the new trait and select the seedling that has less portion of the wild. So this is what we, yeah, sorry.
And then a question I should know the answer, but I don't. I know about the rice, and I know about the potato world center where they keep, banks. Is there an apple world collection of apples? Because as I know, it's like 30,000 that existed before.
No, we do have. There are large. Yes, there are large collection. Actually, we had a European project where we basically genotype all this collection. We have now data for 20,000 individuals Of course, there are a lot of identical, because the number of real cultivar perhaps is 3,000 or more. But we are collecting them and also evaluating them among different environments. So we'll build some reference population that are growing in different environments to evaluate them across different climates and.
I mean, a last question. I remember that the first time I went to Nairobi, the most expensive fruit you could get in the market was apples. There was almost no, and I said that's the cheapest for us in Sweden.
Yeah.
So can they? I mean, apples grow well in tropical and semi-tropical places.
So, well, the quality won't be that great because what we have seen. So the best quality we have here in our region, Trentino, is because we have a climate which is cold during the night and warm during the day. So you need this difference for apple, for these trees. So you need to activate the flowering and also the maturation of the fruit. So what we are now with the climate change, what we are doing, we are improving apple that require a less chilling during the night and day, so so we are working on this. So I'm not sure the quality of the apple will be that great in this climate. Yeah.
Okay.
Thank you for the question.
Yeah, I had many more, but
yeah, you can write me if you are interested. I'm collaborating with the Swedish, and they have a collection of apple, and I'm working with this Uppsala.
Yeah.
So you.
I'm here.
Okay.
Okay, thank you.
Thank you to your question.
Okay, are there any questions? Okay, in the absence of questions, let's thank Dr. Michela Drojo for her valuable time spending with us here in ICF-MLS 2022. Thank you, Michela.
Thank you, Nadisha. And nice to see you again.
Me too. It's glad to see you.
Thank you.
After finishing these two invited guest talks, we will mark the end of the Plant Genetic Resources and Biotechnology Symposium. So we will join for the next session.
Now have a short tea break for 15 minutes and we'll join again for the poster sessions. That is the second poster session for the conference.
Yeah, just to agree the guideline, something like that.
unaware.
Now commence the second poster session for the conference. I would like to introduce the chairpersons, Dr. Kusita Vikramasinghe, Senior Lecturer, Faculty of Medicine, University of California, and Dr. Vasanthi Subasinghe, Head Department of Biochemistry and Clinical Chemistry at the Faculty of Medicine, University of California.
Good day. Today I'm going to present my poster presentation on development of DNA-based accurate identification method for agarwood produced by endemic threatened Grunopis valla. Myself Salachani Priyankan and my supervisor name is Dr. Nadisha Luke Bandara. In 2012, there was a news published in the social media saying that there was an endemic plant of Sri Lanka which has the capacity to produce agarwood. So it is Grunopis valla. So after that, it was overexploited and smuggled for its agarwood. So the government introduced 3 more Aquilaria crops for the commercial purpose to meet the aroma, the agarwood demand in the aroma market. Still, the threat remains for the Grunopis valla. So, because the Grunopis valla and the Aquilaria species are morphologically hard to distinguish, so there is a need for a scientific tool for the identification. So, our objective is to develop DNA-based accurate identification method to identify agarwood produced by threatened endemic Grunopis valla. So basically, the Grunopis valla sample was collected from Madhukama and it was processed till the Sanger sequencing. And in the meantime, the barcode region that we selected for this study is ITS, MEDCAP, and tRNF. So the 3 introduced Aquilarias are Aquilaria malacensis, Aquilaria crasana, and Aquilaria subintegra. The 3 Aquilaria species DNA sequence were downloaded from the NCBI and it was shown in the selected sequence was shown in the table 1. So what we did was the Aquilaria sequence and the obtained Grunopis valla sequence were compared using the multiple sequence alignment tool, and to observe the variation was recorded, and the primer was developed using the primer 3 input, and the quality of the primer was checked in the integrated DNA technology, and the primer covering region was blasted to analyze the variation in difference. So a total number of 53 single nucleotide polymorphism was observed, 42 was observed in ITS region, 8 was observed in tRNF region, and 4 was observed in the MEDCAP region. So 2 kinds of primer was developed. One primer was covering the variable regions and one primer was developed within the variable region. And the primer covering region was blasted using the BLASTN programs, and 2 criteria was selected for the variation analysis: the percentage of identity and the expect value. A total number of 7 primer were developed, 5 for the ITS region, 1 each for the tRNF region and MEDCAP region. In conclusion, the result shows that the ITS barcode region is the most suitable candidate for the differentiation of Grunopis valla from Aquilaria. However, there should be a lot of wet lab experiment has to be done to ensure that the newly developed molecular markers have the capacity to distinguish Grunopis valla from other Aquilaria species. As a result, the newly developed molecular marker in the near future is valuable to prevent the unlawful agarwood trafficking from Sri Lanka. Thank you.
Can you hear me?
Yes, I can hear you.
So you are yet to proceed with the wet lab experiment, but usually how much of the time does this, when you put this into application, how long does it usually take? Let's say you want to detect a real sample. What's the timeline for detection with a barcoding system like this?
So actually we wanted to do it as it's a rapid identification, as in the sense like normally it will take around like 2 to 3 weeks when you inspect an agave in the customs. So what we wanted to do is we wanted to do a PCR and then we had to run it for the gel. It will take around like within 6 hours. So that's what we wanted to limit that. For an example, if we wanted to check 2 samples, the wood samples, wood blocks. So sending to the from the customs, it will take more than 3 weeks to 4 weeks. It will even if it is not the agave, it has to be stored until it been clear from the customs. So I think we probably, if it's work, it will take around like 6 hours.
So can you kindly explain the gel that you have on the poster? I wasn't clear about it. So that's the
gel was like it's 1% agarose gel, because like we didn't do it within specific, because like these are the 3 extracted DNA. We didn't go for that. We couldn't able to get that agave. So it has to be done. It has to be checked in the agave specifically from the wood chips or something like that.
Right. So the extracted DNA was from the plant itself?
Yeah, plant leaves.
Okay. Okay. Right. Thank you.
Sorry for the background.
Okay, then thank you very much. Good work.
Have a good day. Thank you.
Right. We can go for the next present, I believe, post 16.
Good morning, all of you. I am Nilmini Vijayanayakar, a postgraduate student from University of Rohana. Today I am going to present my research project on effect of aqueous extracts to cinnamon bark on hyperglycemia in diabetes-induced visceral models. As you already know, diabetes mellitus is a very common disease in Sri Lanka. Nowadays, a significant amount of world population is suffering from diabetes. Therefore, diabetes is called as the epidemic of the century. If there are effective drugs for the reduction of hyperglycemia, scientists are focusing on finding more and more efficient drugs with less side effects. Innovating herbal nutraceuticals and functional foods which can be used to reduce hyperglycemia is a current research trend in Sri Lanka. Cinnamon has known for centuries as a potential herb which can be used to decrease the high blood glucose levels of people. So, the objective of this research is to determine the effect of cinnamon aqueous extract in diabetes-induced viscerals. For that, I took 24 viscerals and divided them into 4 groups. Among them, 3 groups were induced diabetes using streptozotocin. After that, group 1 was treated with cinnamon and group 2 was treated with metformin for 42 days. Group 3 was used as the negative control, while Group 4 consists of diabetes non-treated rats. Then the fasting blood glucose was measured in the baseline and after 4 weeks of the study. After that, AST, LT, and serum creatinine was determined after 42 days of the study. According to the results, there was a significant reduction in fasting blood sugar levels in cinnamon-treated rats. You can see the graphical representation of the obtained data from all 4 groups. There were no significant difference between other groups, but there is a significant difference in cinnamon-treated rats. Renal and liver function tests have revealed that there was no adverse effects happened during the cinnamon treatment. All the values were within the physiological reference ranges of healthy individuals. Therefore, we concluded that cinnamon bark aqueous extract have potential to be developed as an anti-diabetic drug or nutraceutical. Ethical clearance for this research was obtained from Ethical Review Committee, Faculty of Medicine, University of Rome. This project was funded by the AHEAD DOR project. Thank you so much for your attention.
Can you hear me?
Yes, sir, I can hear you.
Okay. Can you go to the slide where you had the serum creatinine value, I suppose, kidney function evaluation?
In this poster, I think you can see, sir.
Yes, I got it. So, if I'm correctly reading it, the liver enzymes were relatively well controlled, but the serum creatinine values are a little bit higher in the cinnamon treated group compared to the metformin. Now, is that going to be a concern because you have only treated for 42 days and for diabetes you'll have to treat for a very long time? So is it something that is known that there might be an effect on the kidneys when you take cinnamon in therapeutic levels?
Sir, we are focusing on doing this for KIM1 and NGAL molecules also. After that, we are going to decide whether those molecules also affect
seriously looking into the kidneys for that.
Yes.
Right. Do you know from the previous studies how long is there a sort of a tapering off of the effect, or is this cinnamon effect, is it sustainable for in the long term? Have you come across any feature like that?
No, sir. There are very few studies for long term. A lot of studies were about 3 to 2 months. There is no long-term studies till now.
I have one question. Maybe I didn't hear it properly. Did you say there was no significant reduction in fasting blood glucose level for the metformin group as well? Or because I heard it something like that.
In statistically, madam, there is no significant difference between the metformin group, but clinically the diabetes levels has reduced. I think because of the sample size is low, the significant difference between metformin arm couldn't be detected. But clinically, diabetes has reduced, hyperglycemia has reduced in metformin arm too.
Just one quick clarification. So when you give streptozotocin, the beta cells are destroyed, right? Am I correct?
Yes, sir, yes.
So in that sense, is it reasonable to try metformin? Because my understanding was for metformin to work, you may need some sort of a beta cell activity. Is it used in the literature in this context after streptozotocin?
Yes, sir. I have referred. Our streptozotocin values are much lower than lower. So some beta cells are
Some residual activities
remaining.
Okay. So it should be fine. Okay.
Yes.
Thank you.
Yes. Thank you very much, sir.
I got it.
Ladies and gentlemen, I'm Fabiola with my supervisor Uminda Pereira and Heshani Mudalike. I have successfully carried out protein ligand site-specific docking for plasmapsin 2, a malaria protein receptor, and identified potent phytochemicals which can act as ligands and their binding sites. Malaria is an infectious disease caused by Plasmodium species and is transmitted by Anopheles mosquitoes. Mostly, the tropical and subtropical parts of the world are at great risk. Additionally, malaria resistance have been reported in recent years. Hence, there is a great risk of re-establishing disease. This indicates the necessity of finding new drug targets to treat malaria effectively. Hence, the chosen protein was 1LF4, which is a malaria protease enzyme, and the structure of it was downloaded from Protein Data Bank. The active sites were determined using the CASP web server. Similarly, the ligand structure was downloaded from PubChem, and their ADMET properties was analyzed using SwissADME web server. During preparation of proteins, the protein underwent removal of water molecules and addition of Coleman charges, whereas the second file was saved in the PDB-QT format. Similarly, the STA point of the ligand was also converted to PDB-QT format using open babel subset. Thereafter, the active sites of the protein was selected and the grid box was placed in a manner such that it contained only the area that had active sites. Thereafter, the autogrid execution was performed and the docking execution was also performed, where the docking parameters was changed to the number of G-runs with 50 and the population size with 300. Meanwhile, Lamakian algorithm was used to execute the docking process. After the generation of docking process, the results were basically analyzed using binding energy, inhibition constant, H1 and amino acid interaction. So, with the binding, obtain the lowest binding energy. According to thermodynamics, the lower the binding energy, the more stable the complex is. Meanwhile, inhibition constant of serpentine also lower compared to other phytochemicals. The best pose of 1LF4 and serpentine was visualized using Biology Studio, and the ligand binding sites were obtained from products. While the other properties of the serpentines also indicate it as a potential ligand conducted by Ravi and Raghunathan, 1LF4 with alloying and microdontin did not produce effective results as serpentine. So, significantly, serpentine was found to be a potential ligand towards 1LF4, and it could effectively extract from a perennial shrub belonging to Asian countries. But experiments such as ELISA-based assays are vital in improving its efficacy towards treating malaria. Meanwhile, I would enhance my research with further validation such as redocking and using Chandra plot. So, with these points, I would like to conclude my presentation and also to convey sincere gratitude towards the organizers of this event, and thank you to all for listening.
Good evening. Can you hear me?
Yes, we can hear you. Is there a particular reason why you chose this plant? Maybe I missed it from your presentation. Is there any prior?
Yes.
Yeah.
Because the malaria is most commonly seen in Asian countries and African countries. So this plant can be easily extracted from Asian countries. So it will be like a quick remedy to treat malaria.
You mean it's used in like traditional medicine or?
They have found that it has anti-malarial properties, but they haven't started any research yet.
Okay.
So this will be a great advantage.
As like when you say they have found, has it been published or is it in?
Yeah, they have published. So from that, I got an idea to choose this plant, and this plant also showed a better binding energy compared to the rest of the phytochemicals that have been tested.
So it looks like, if I'm correct, it's a very hydrophobic molecule. Am I correct?
As for the moment, we considered only hydrogen bonds. Yeah.
No, I'm more worried about, let's say, even if it's successful, how about the delivery of the drug into the body? That's why I was a little bit concerned. And so let's say you get these values in silico from how, if you think about a real world example, how well do they translate into real world drugs? What is the chance that you get something working on in silico? What is the probability that it's going to work on the real world?
I would also do the control experiments with the FDA drugs approved now. So while comparing those things, we can get an idea.
Okay, thank you.
Welcome.
Have you ever wondered how pharma companies like AstraZeneca and Pfizer make therapeutic drugs? Well, one of the methods they use is structure-based drug design. Here, the structure of the protein is attacked using a ligand which will be developed into a potential drug. In order to find the best fit ligand, it is pivotal that all possible ligands are screened. But it is almost impossible to do it manually as the number is mammoth. This is where bioinformatic computational techniques come in. And one of the most widely used techniques is protein ligand docking. I'm Dravidian Languages, and my research is ligand docking for the identification of therapeutic ligands against the Nipah virus attachment glycoprotein using AutoDock, conducted under the supervision of Ms. Heshani Mudalike and Mr. Ominda Pereira. Nipah virus is a biosafety level 4 zoonotic pathogen belonging to the Paraxinidae family and Henipavirus genus. In 2018, the World Health Organization listed it as a potential epidemic-causing virus requiring urgent attention. The most recent outbreak was recorded in India, Kerala 2018, where out of 18 people infected, only 1 survived. The natural reservoirs of the Nipah virus are fruit bats belonging to the Pterophis genus. They are endemic to Southeast and South Asia, including Sri Lanka. Transmission among humans occurs through contaminated body fluids. Symptoms of the Nipah virus infection are acute febrile encephalitis and severe respiratory diseases. The fatality rate is approximately 75%, but to this day, there are no licensed treatments available. Currently, research on treatment options are focused on developing immunotherapeutic antibodies, repurposing FDA-approved drugs, molecular docking studies, and immunoinformatic techniques to create a vaccine. Materials utilized for the study were a computer with a reliable network connection, web tools, and software utilized for protein ligand docking. Here, blind docking technique was employed where no prior knowledge of the active sites of the protein were known. For docking, FDA-approved drugs for standardization and phytochemicals as new therapeutics were chosen. The 3D structure of each ligand was retrieved from PubChem in STF format and converted to PDB format using OpenBarbell. The 3D structure of the glycoprotein was retrieved from RCSB PDB in PDB format. In AutoDock, the output created following docking was used to analyze the binding energy, inhibitory constant, docking pose, and hydrogen interactions which were acquired from the respective software. Lower binding energy demonstrated higher stability of a docked complex. Lower Ki represented the integrity of the ligand to inhibit the protein. Higher hydrogen interactions proportionated with higher resistance to bond breakage within the docked complex. Thereby, the best ligands were FDA-approved drugs tolvaptan and conivaptan, and phytochemicals demethoxycurcumin and neoandrographolide. Lysine 560, valine 507 were observed to be frequently involved in forming amino acid interactions. Downstream, it is proposed to utilize the phytochemicals identified as leads for antiviral drug development against Nipah virus, as plant-based antivirals depict less toxicity adverse effects. which can be extracted from plants endemic to South Asia, such as Andrographis paniculata and Curcuma longa. Thank you.
Thank you. What was the basis for your claim that the plant-based... Oh, can you hear me? I'm sorry.
Yes, I can hear you.
Yes. I mean, what is the basis for you to say that the plant extracted antivirals will be less toxic than, let's say, FDA approved like a single drug?
Because the FDA approved drug, for example, it's been used for a particular reason. In this case, conivaptan and tolvaptan are vasopressin antagonists.
Right.
Definitely, it's going to be a side effect if we use it, but we can use it to sort of like differentiate it and make some sort of chemical change in using for as a treatment for Nipah virus. So against that, using phytochemicals that have been known to show antiviral, only antiviral activities is much more productive.
So this particular extract that you use, let me check. Sorry, maybe I missed it. This was an extract. No, what was the drug? I'm sorry, I missed that one. So this particular phytochemical, now it should have a similar binding to the glycoprotein, right? The attachment glycoprotein. So in that sense, don't you think there might be, let's say, the particular FDA approved drug has a certain side effects. If you have a biosimile to that, wouldn't you expect some similar side effects?
They are not similar structures. It's just that they bind to the same pocket. They might not bind to the same exact position, but within the pocket itself.
Right.
So they don't have the exact same binding energies, the binding amino acid interactions, and the hydrogen interactions.
Okay. Thank you.
Yeah.
All right. Thank you very much.
I'm Nishan Madhusankar from Department of Biochemistry, Faculty of Medicine, Sabaragama University of Sri Lanka. Let me share my research findings on scientific investigation on Sri Lankan polyherbal drug, Nilagiri Pandama, for its antioxidant activity, polyphenol content, and TCMS analysis. Did any of you hear about miracles of Sri Lankan traditional medicine or Western medicine? In fact, some patients who are told to be not cured by Western medicine are cured by simple treatments of Sri Lankan traditional medicine, and we have witnessed plenty of those stories. But the awareness of the scientific basis of those therapeutic herbal formulae are very limited in the literature and therefore among the public. To bridge this gap, we need to perform research as to back the related hypotheses with valid data. Finding mechanisms of action of these polyherbal formulations in Sri Lankan traditional medicine haven't received much attention in the scientific community. For our study, we chose a very important and sound Sri Lankan traditional drug, Nilagiri Pandama. Nilagiri Pandama is known to be very effective when treating abscesses, wounds, and cancers. Personal communications find that this drug has a potent therapeutic ability. As the first step, we investigated the antioxidant activity, polyphenol content, and important phytochemicals of Nilagiri Pandama. First, we analyzed aqueous extracts of Nilagiri Pandama using bioassays such as Fordinsia Caratao assay and DPPH acid. Also, we analyzed ethanol extracts of Nilagiri Pandama using Ecms Fingerprinting technique. We found considerable amounts of polyphenol content compared to gallic acid and significant amount of radical scavenging activity compared to ascorbic acid. Also, GCMS study revealed very important pharmacological compounds such as phenol derivatives and acetylene 2-ol. Therefore, a lot of works that need to be done filling the gap before we know how much this drug is healing the human body. We will carry further studies in vitro and in vivo studies to validate this polyherbal drug, Nilgiri phantom. Finally, our effort is to contribute to expedite the drug discovery process using advanced techniques in Sri Lanka. If we can apply these strategies to more drug discovery programs, who knows how many millions of dollars we could save. Thank you very much for your time.
Interesting study. I have a question now. The traditional or folk medicine generally approaches as a holistic manner, and then when you look at this Nilgiri phantom, I suppose it's a mixture of several ingredients, right? So how would
yes, madam. Yes, madam.
Translating that into a identifying a particular ingredient would reflect the effect of effectivity of the drug. Do you follow what I'm saying? Say now.
Yes, madam. I can hear you clearly.
Yeah. If it is
actually, madam, most.
Go ahead. Yeah.
Yes, madam. Yes, madam. Actually, madam, most of these polyherbal drugs working in a synergistic manner. So, in order to find the active ingredients, we need to clearly purify these phytochemicals, madam. So, we specifically look for the phytochemicals. What are the responsible for this much of antioxidant activity?
No, my concern was also now say that what is the guarantee that the active ingredient itself is actually acting, or that the mixture itself has effect? Because what we have noticed a lot of that traditional medicine, when we try to translate it into the Western medicine, because of the difference in the approaches, sometimes that when you separate the ingredients, or when you try to identify the active ingredients, that it doesn't actually give the expected outcome. Have you think about that aspect of it? That was my question.
Yes, madam. Actually, that's actually a question for me also. Actually, madam, I think we can't actually look for the only one active ingredient. So we need. I think, madam, we can't actually separate only one active ingredient since this working in synergistic manner. Yes, madam. So
very quick one. Is this, did you say this for a topical application, or is it for a systemic application? Is it to be on the skin? Or?
This is a systemic applications. Actually, this drug given by oral, with water.
Okay.
Did you hear about the kasaya thing?
Right, right.
Yes, sir.
Okay. Thank you.
Have you looked at the stability of these antioxidant species after it goes through digestion, absorption process and all? Have you
actually, madam, this is our first study regarding these polyherbal drugs. Actually, madam, why we choose this drug is this doctor has a record. So he actually, yes, he gives a Lepanagulia drug. So it's considered as a anti-cancer drug, and they are proceeding with animal studies now, madam. So we are screening for these drugs also now.
Thank
you.
Thank you very much, madam. Thank you very much, sir.
Hi, I'm Uma Pradeshri Rajagopalan, presenting my study on molecular docking that reveal natural benzophenols and xanthones from Garcinia salonica as Wnt and H pathway inhibitors in breast cancer stem cells. Cancer stem cells are a rare population that reside in malignant tumor that prime cancer pathogenesis. Although chemotherapy induces tumor regression, recurrence and metastasis have been evidenced in multiple cancers. Cancer stem cells play a major role in tumor initiation, progression, metastasis, cancer, multi-drug resistant, radiation therapy resistant, and cancer relapse. Dysregulation of Wnt and hedgehog signaling pathways were evident in cancer stem cell survival. Therefore, targeting cancer stem cells through signaling pathway would be a better treatment strategy. We identified that the 4 compounds from Sri Lankan endemic plant Garcinia selanica suppress breast cancer stem cell proliferation in vitro setting. However, their molecular targets have not been studied. The present study investigates the molecular target of the 4 compounds in Wnt and hedgehog pathways using in silico setting. For this purpose, compounds were drawn in ChemSketch and optimized in Avogadro, and the ligands were prepared using Chimera. Then the ligands were docked with protein, autodocked Vina. The binding pose analysis and the drug likeness were analyzed using online servers. For this purpose, online servers, results indicates that gasol linear xanthone 14-deoxygasol hits beta-catenin, whereas xanthone hits SMO. This beta-catenin TCF interaction needs Lys 435 and arginine 469 that were blocked by nitrolinear xanthone I and gasol, whereas arginine 515 were blocked by 14-deoxygasol. Pathogenic activation of SMO was reported in breast cancer. Among the compounds, only xanthone I showed the binding affinity towards SMO, which, as there are very few compounds that target hedgehog pathway, it is necessary to critically analyze the ability of this compound. Drug gets eliminated before clinical trial, except 14-deoxygasol, the other isolated compound not crossing the blood-brain barrier. On oral administration, all the compounds were predicted to be absorbed by human intestine and penetrate colorectal tissue with lower oral toxicity and no carcinogenicity. Drug likeness of these compounds satisfies Egan's model and Berber's rules. Although garcinol and deoxygasol violated the 2 rule of 5 Lipinski's rule, they cannot be neglected from potential drug candidates, as there are 30% of FDA drugs to violate Lipinski's rule of 5. In concluding, 4 isolated compounds can be improved suitably to produce promising therapeutic agent to target cancer stem cells. Thank you.
I have a question just regarding the process of the optimization. So how long does it actually take to optimize one of those drugs using the in silico method to identify the binding properties and all that in time-wise?
Time-wise, within 2 days
you can do it. Right. Okay. So that's a relatively rapid thing to do. Now, when you do that, I mean, do you also consider how feasible it is to actually separate the active ingredient from the extract? Now, these 4 extracts, have they been previously separated using chromatographic method? Or?
It's almost 2.5 years for the separation. Of pure compounds using bioactivity guided studies, and to develop the nanospheres, I even spent 6 months because they are very slow growth rate. Compared to that, this is very
fast. Okay, I just wanted to know the timelines basically.
Yeah, actually.
So when you look at the binding, for an example like this in one pathway, do you also consider the potential of developing resistance in the pathway if you take a drug? Because sometimes that can happen when you treat one pathway, the resistance can is there some information you can get on the likelihood of, I don't know, I'm asking you, likelihood of developing resistance for a pathway, for an example?
Yeah.
Is it something you can get from an in silico experiment?
No, actually, it's very hard.
Yeah.
But I can do that. We can at least get an idea. Like if it's not going to go through, or it is digestible, or if we like a protein resistant, you can expect they are going to be resistant developers.
Can that also predict what happens in the liver?
Yeah, because liver actually it's very complex organ. It's very complex organ. So that you should go to in vivo studies for that.
Yeah. Okay. Thank you.
Thank you so much.
Okay, you can start.
Is that a video? Yeah, now the recording. Yeah, it's a recording.
Here we are going to present our poster titled Comparison of Rat Immunoresponses to Pathogenic Leptospira zero was prevalent in Sri Lanka, and the presenting author is Gangani Pede. Leptospirosis is a zoonotic disease which is caused by spirochetes of genus Leptospira, and it is distributed worldwide and it is endemic in Sri Lanka. Although there are several diagnosis methods available for diagnosis of leptospirosis, currently there is no antigen-based detection test available for diagnosis of leptospirosis at an early stage. So, in order to establish a diagnosis method, it is very important to identify immunogen profiles of the leptospira. During our study, that's the immunes using two different zero. Circulatory prevalent zero was in country, zero of Becquerel, zero of phylogenes, and immune response was assessed using indirect in-house ELISA. IgM response against zero of Becquerel compared to zero of phylogene was significantly higher from day 7 onwards, except phase 35 and day 56, which is reflected by OD at 1 to 100 serum dilution as well as IgM titers. IgG response against zero of Becquerel compared to zero of phylogenes was significantly higher from day 7 onwards, and highest immune response was observed in day 63, while zero of Becquerel indicate serum antibody titer of 12,800, while zero of phylogene indicate serum antibody titer of 1,600. Serum sample with high antibody response was selected and used for immunoblotting in order to identify immunoreactive antigens. Immunoblotting with homologous red antiserum indicate 6 immunoreactive antigen with respect to phylogenes and 4 immunoreactive antigen with respect to Becquerel. This was subjected to immunoblotting using heterologous red antiserum, and results indicate 2 immunoreactive antigen with molecular weight 158 and 100 kilodalton, which indicate cross-reactivity with respect to zero of phylogenes and zero of Becquerel. This immunoresponse, immunogenic profile, as well as immunocross-reactivity is very useful in identifying target antigen in development of immunodiagnosis test. I would like to acknowledge grant ICGPCRP LK1701 for its financial support, and I would like to acknowledge Director and staff of IBMBB for the support.
So the idea is that you basically try to get an immune response and then see what are the proteins possibly that are actually giving rise to this immune response. I'm not very conversant in immunology. Yeah. So once you identify, then you can think about potential whether to develop an antibody against that. That's the plan. Am I correct? Yeah.
And basically immune-reactive, cross-reactive
proteins. Okay. So once let's say you have a band, and once you identify, how do you actually characterize the protein? How do you identify it? You have to do a...
Actually, we are doing a CS page, and it contains like so many proteins. Right. And then we are doing immune mapping. Right. From that, we identify immune-reactive protein.
Right.
And from that, keeping that CS gel along with the immunoglobulin profile, we can identify this is the immune-reactive band, and we can crush that gel and we can
isolate that. But you don't want to really identify exactly what the protein is, but you want to separate it out.
Yes.
So that you can generate the antibodies using that one. I saw that there is a significant variation sometimes in the titer within that very short period. Is it a natural phenomena, or is it?
It's basically with respect to IgM response.
Yeah, because it's an acute one. So this is the time that it takes for the acute response to be built up, or
actually, I will indicate the immunization schedule. When it is immunized, the IgM responses increase. And in between the days, it decreases, and again when immunized, it increases.
And then the IgG should continue. Okay, thank you. All right. More challenge.
Hi everyone, my name is J. Celestin Monezero. I'm going to present a study conducted in IBMBB, University of Colombo, entitled Effect of Mannonia pinnata on Functional Changes of Endothelial Cells Related with Dengue virus Serotype 3. This study has been supervised by Professor Shiro Mahanduneti, Dr. Narmada Fernando, Ms. Roshinila Nawila, and Dr. Swan Hapwalachichi. So dengue virus have been reported to infect endothelial cells, causing their dysfunction and lead to severity in a disease. This rationalizes the therapeutic targeting of endothelial cells. On the other hand, Mannonia pinnata is a rare medicinal plant which is commonly used to treat fever in traditional system of medicine in Sri Lanka. So the objective for this study was to investigate the effect of aqueous plant extract of Mannonia pinnata on the interaction between dengue virus Serotype 3 and endothelial cells inducing any change. So to reach this objective, a calorimetric assay for assessing metabolic activity, MTT assay, was performed to assess the effect of DNB3 on the metabolic activity of endothelial cells and to determine the potential protection of endothelial cells in dengue infection through 3 different ways. A, the pretreatment of DNB3 with AP Mannonia pinnata via its contact with endothelial cells. B, the sequential treatment of endothelial cells with DNB3 followed by AP of Mannonia pinnata. And C, the sequential treatment of endothelial cells with AP of Mannonia pinnata followed by DNB3. As a result, a significant reduction of endothelial cell viability of 49.8% was recorded following the interaction between endothelial cells and DNB3 as opposed to that in absence of DNB3. Then, with addition of Mannonia pinnata, significant protection of endothelial cells was observed at almost all concentration of AP Mannonia pinnata in the 3 treatment strategies. Interestingly, the highest or increased significant protection of endothelial cells was observed during the pretreatment of DNB3 with AP Mannonia pinnata prior interaction with endothelial cells, with the highest protection at 15.6 and 31.3 microgram per ml. So this suggests that the plant extracts act directly by inactivating the pathogenic activity of the virus. To sum up, the study suggests that the AP of Mannonia pinnata exhibits an antiviral effect against DND3 by protecting the metabolic activity of endothelial cells. So further studies are needed to understand the underlying mechanism of the direct inhibitory activity of Mannonia pinnata against the dengue virus and to characterize the active compound. So this study was supported by the Association of Commonwealth Universities and the IBMBB University of Colombo. Thank you for your kind attention.
Thank you. Can you kindly go to the essay? Was it an MTT essay? I'm sorry, I went so far. I could not really follow. I'm sorry. So the results that you have put on the graph, is it an MTT essay or I'm not sure? Couldn't follow. I'm sorry.
Thank you. The result here are from MTT assay.
MTT assay. So you basically, when you increase the concentration of the extract, right, the viability has basically decreased. Am I correct? I'm just trying to figure out whether I missed out something.
Yeah, at the beginning, it starts to increase, but when it increases more, it starts to decrease again.
Right. So once you add the pre-treatment, basically the second graph is the protection. Okay. Okay. Yeah. The first one is basically the titration of the virus.
Of the virus.
Ah, right. Okay. Sorry. Took a minute for me to understand. Okay.
Yes.
Okay. So your conclusion is by looking at these three, the pre-incubation of the virus with the drug, basically it's a more protective effect than therapeutic effect. Is that your conclusion?
Yes. The direct contact with endothelial cells. Yeah, the virus and the plant, it increases the protection of the endothelial cells.
Okay. Is there a merit in using like a leakage assay rather than a viability for something like this? Or will it give a similar result if you use some sort of a leakage assay?
Here we only use that for viability, the MTT assay.
Okay.
For proliferation. Yeah.
Right. Okay. Thank you.
Thank you.
A very good day to all of you. I would like to present about an interesting research project on synthesis of silver nanoparticles using plant-derived phytochemicals. Most of the plant extracts used in traditional medicines possess inherent antimicrobial activity. One of the promising ways to promote their efficacy is to incorporate them into nanostructures. In this study, silver nanoparticles were synthesized using aqueous extracts of Linnea coromandelica and Pothos scandens. In Ayurvedic medicine, these plants are used in treating wounds. The bark of Linnea coromandelica leaves and stems of Pothos scandens were dried and pulverized. 10 grams of Linnea coromandelica and Pothos scandens were sonicated at 44 kHz for 30 minutes in H2O in separate. Different concentrations of the aqueous extracts were mixed with aqueous silver nitrate in separate to synthesize Linnea coromandelica coated and Pothos scandens coated silver nanoparticles. Several different conditions were employed and yields were optimized based on UV spectral data. Homogenization at 10,000 RPM under UV light was found to provide highest silver nanoparticles yield. The figures show the UV visible spectra overlay during the synthesis of Linnea coromandelica and Porthosiscandens coated silver nanoparticles at different time intervals. Silver nanoparticles were characterized using particle size analyzer and atomic force microscopy. Atomic force microscopy revealed the presence of spherical Linnea coromandelica silver nanoparticles and Porthosiscandens silver nanoparticles of the size ranges.
Recording in progress.
A very good day to all of you. I would like to present about an interesting research project on synthesis of silver nanoparticles using plant-derived phytochemicals. Most of the plant extracts used in traditional medicines possess inherent antimicrobial activity. One of the promising ways to promote their efficacy is to incorporate them into nanostructures. In this study, silver nanoparticles were synthesized using aqueous extracts of Linnea coromandelica and Porthosiscandens. In Ayurvedic medicine, these plants are used in treating wounds. Stem bark of Linnea coromandelica leaves and stems of Porthosiscandens were dried and pulverized. 10 grams of Linnea coromandelica and Porthosiscandens were sonicated at 44 kHz for 30 minutes in H2O in separate. Different concentrations of the aqueous extracts were mixed with aqueous silver nitrate in separate to synthesize Linnea coromandelica coated and Porthosiscandens coated silver nanoparticles. Several different conditions were employed and yields were optimized based on UV spectral data. Homogenization at 10,000 RPM under UV light was found to provide highest silver nanoparticles yield. The figures show the UV visible spectra overlay during the synthesis of Linnea coromandelica and Porthosiscandens coated silver nanoparticles at different time intervals. Silver nanoparticles were characterized using particle size analyzer and atomic force microscopy. Atomic force microscopy revealed the presence of spherical Linnea coromandelica silver nanoparticles and Porthosiscandens silver nanoparticles of the size ranges 100 to 300 nanometer and 200 to 400 nanometer respectively. In conclusion, the present study revealed two novel methods of phytochemicals-assisted synthesis of silver nanoparticles. Thank you very much.
Hello, I am Madhushi Rajpaksha from Rajarati University of Sri Lanka. Our topic is antioxidant, antibacterial and antifungal activities of flowers of hibiscus species. First of all, I would like to give a brief overview of our study. Hibiscus is an ornamental plant that belongs to the family Malvaceae, which has been shown to exhibit some biochemical properties due to the presence of various active natural compounds. The flowers are used for medicine, cosmetics, food and beverage production in some countries, but very limited in Sri Lanka. The main objectives of this study are to determine the antioxidant level, IC50 value, total phenolic content, antibacterial and antifungal effect of hibiscus flowers in Sri Lanka, and also to assess the potential of hibiscus flowers for therapeutic purposes. For this study, 11 varieties of hibiscus flowers were collected from Anuradhapura area. From these 11 samples, 10 samples are hibiscus rosa sinensis, except MRJ. MRJ is classified under the species of Hibiscus schizopetalus. The fresh flower petals were oven dried and the active compounds of hibiscus flowers were extracted using methanol and dichloromethane and concentrated using rotavapor. The antioxidant activity and ISO50 values were determined by DPPH method. Police Geocalculus method was used to test total phenolic content of hibiscus flowers. In addition, antibacterial and antifungal activity was analyzed by this diffusion method. This study has proven that the maximum amount of antioxidant activity and total phenolic content was observed from Hibiscus rosa sinensis and minimum amount from Hibiscus schizopetalus flower. The antibacterial activity of hibiscus flowers were determined from the microbes Escherichia coli, Salmonella typhi, Staphylococcus aureus, Pseudomonas aeruginosa, and antifungal activity was determined by using Candida albicans. This graph has highlighted that the flower Hibiscus rosa sinensis having the highest antibacterial and antifungal activity and Hibiscus schizopetalus flower having the lowest antibacterial and antifungal activity. On the final note, both Hibiscus rosa sinensis and Hibiscus schizopetalus have shown antioxidant activity, antibacterial and antifungal activity. Therefore, Hibiscus rosa sinensis have shown promising biochemical activity than Hibiscus schizopetalus. So further studies are required to explore possibility of using it for therapeutic purposes. Thank you.
Can you hear us now?
Yes, I can hear you.
So to get those numbers, how many samples actually did you analyze per flower, let's say, like type?
I analyzed in 11 flowers.
No, I mean the value. So for an example, the inhibition
I
percent.
analyzed 11 flowers. Sorry, sir.
Was it in duplicate, triplicate analysis or
Sir, I did it duplicates.
duplicate? Okay.
I did duplicates.
Because except for the, if I'm correct, the antifungal activity, the others are probably not statistically significantly different. Am I correct? Because I can see the values, but they're probably not
No, but
statistically
I
significant.
don't think she used statistics. Did you use any? Did you use any statistical analysis when comparing?
Yes, sir. No, ma'am. I didn't use it. No, I didn't.
For the time being, these are the average values, ne? Right. So I think it's better, perhaps, particular. Yes, go ahead.
Yes, sir.
Better, particularly for that antifungal, which is very standing out. So you probably, and I think it might not be very accurate to say that this one will have the least one, because the the range is not that large, except for that antifungal, which is very interesting. I think that has a very standing out effect.
But
Yeah, so I think you should perhaps do a few more if you want to really claim that this has the highest activity and this has the lowest activity. That is my feeling, because they are very similar.
Okay, sir.
Okay.
Yes, sir.
Right. Thank you.
Thank you.
The other person is there. There's a person to ask.
Not there. No problem.
Thank you, Dr. Kusita and Dr. Vasanthi, all the way from Faculty of Medicine, University of Colombo, colleagues of Professor Prasanna Gallen, our director. So thank you for spending your valuable time with us. You have done a great job. Okay, hope you will join us next time also.
Thank you.
Thank you.
No, that's fine.
Next, session 7, Natural Products for Therapeutics, award winner, Pratita Tene. Again, for session 1, evaluation of poster communications, the winner is again Pratita Tene. Next, session 2, evaluation of poster communications. Award goes to Priyankan Selacham. Thank you, madam. This brings us to the close of the awarding ceremony and also to the end of the 3rd International Conference on Frontiers in Molecular Life Science. We hope you had 2 days of enjoyment, fun, and most importantly, knowledgeable outcomes out of it. Thank you, everyone.